Morning to one and all present here, respected delegates and eminent personalities gathered here. The important thing is to not stop questioning. Curiosity has its own reason for existing. These are the words of the great scientist Albert Einstein. Man is living in an era of science and technology. From the moment we rise from the bed till the moment we go back to bed, we can see the help of these two invisible handmaids that serve us so faithfully. Science is for humanity. As you all know, the pandemic had struck us so hard that we were not able to conduct this event for the past 2 years in our college physically. But now, the wait is over. May I take this opportunity to welcome you all to the prestigious 21st K.V. Thomas Endowment Seminar and 4th International Symposium on New Trends in Applied Chemistry 2023, organized by the Postgraduate and Research Department of Chemistry, Sacred Heart College, Tevara. I request everyone to raise for the college anthem.
Stopping by the shimmering shores of time, musing on the timeless truth sublime. Seek aside a gleam on flaming wings. Sacred Heart, the soul enchanted sings. Blessed are we on this praising shore, blissfully gleaning pearls of lore. Pilgrims on these pastures green, seeking joy into lands unseen. Sacred hearts, abode of light, shine on bright, abiding hearts. Sacred heart, shrine of light, shine your beams, make us one at heart. Children of the heart, we join our hands to love, to learn, to grow together. Children of the heart, we join our souls to dare to dream, to live for the other. Master benign, anchor divine, hallowed be thy home, amen. Grant us to bless, grant us love, all that proclaim thy grandeur and love. Sacred hearts, abode of light, shine on bright, abiding hearts. Sacred heart, shrine of light, shine your beams, make us one at heart. Make us one at heart. Make us one at heart.
I'm extremely delighted to call upon Dr. Franklin John, Head of the Department of Chemistry, Sacred Heart College, Tevara, and General Convener of NTAC-2023 to welcome the gathering. Sir, please.
Good morning. Most respected Honorable Nobel Laureate Professor Ada Yonath, Honorable Vice Chancellor of MG University Professor Sabu Thomas, Professor K.V. Thomas, former Union Minister and representative of Kerala State to Central Government, Professor V.N. Rajshekharan Pillai, ex-Vice Chairman of University Grants Commission, most beloved Professor Itzhak Tor, Professor Tamuki, Professor Rosa, Professor Dr. A. Srinivasan, our college manager, Reverend Father Paulos Kidangan, CMI, Principal Reverend Dr. Josh John, CMI, Organizing Secretary of the Symposium, Dr. Ramakrishnan S., distinguished speakers, invited guests, academicians, research scholars, and esteemed faculty members. 7th February 2023 will be written in the history of our 75-year-old department and college in golden letters. Yes, we are greatly privileged to have one of the pioneers in science, one of the pioneers in ribosome structural elucidation, the most respected Nobel laureate, Professor Ada Yonath, amidst us. I express our heartfelt gratitude and welcome you to Secretariat College, Kochi. Welcome, ma'am. We are greatly honored by the presence of our Vice Chancellor of our MG University, Professor Dr. Sabu Thomas, who supports us in all our ventures and is with us whenever we come up with any program, whether it is academic or non-academic. Welcome, sir, for our symposium. I would like to welcome Professor K.V. Thomas, on whose name the Endowment Trust started 20 years back, and he supports us enormously whenever we are arranging this program annually. Welcome, sir. Our college manager, Father Paulos Kidangan, CMI, is with us. Welcome, Father. Our head of the institution, Principal Father Dr. John CMI, is with us. Welcome to this function, Father. Professor Dr. V.N. Davishekaran Pillai, as I heard in the morning, this is the third time he's coming for this particular symposium. And he traveled all the way from Mumbai yesterday night. Thank you for being with us. Welcome, dear Professor V.N. Davishekaran Pillai. Coordinator of our symposium, Dr. Ramakrishnan S., who untiringly worked for this event to happen. Welcome, Dr. Ramakrishnan. I also would like to mention few names who are on the audience. We are honored by the presence of Professor Itzhak Tor, the magician of organic chemistry, the great man in nucleotide chemistry. Welcome, dear Professor. We have with us the great professor in photochemistry of biological systems, Professor Tameoki from Hokkaido University. Professor Derval Rosa from Brazil is with us. Welcome, dear Professor. I would also like to mention and welcome Dr. Hagi Yonath, daughter of Professor Ada Yonath. Welcome, ma'am. I would also like to mention Professor Dr. A. Srinivasan from NICER Bhubaneswar, who came as a resource person for our symposium. This is the 21st edition of our National Endowment Seminar in the name of our former head of the department and former Union Minister, Professor K.V. Thomas. I respectfully welcome all the dignitaries. We do have a definite purpose for our symposium. Our primary aim is to motivate the academic community and the students. We are greatly indebted to all who took pain, traveled a long distance. Words are not enough to express our gratitude as you traveled from other side of the globe, especially Professor Tor, Professor Rosa. Thank you so much. We have a long list of distinguished speakers from various scientific institutions inside India. I extend a warm welcome to them and consider their presence to be a great honor. Our department is working behind this event for months. I greet every one of the faculty members and staff members, friends, colleagues, deans and heads of the departments of other departments, a warm welcome. Participants, especially research scholars and faculty members from different parts of India, are making this program a meaningful one. Thank you so much and welcome to this program. I take this opportunity to greet the sponsors of our symposium, American Chemical Society, Royal Society of Chemistry, Elsevier Publications, along with the central funding agencies like SERB. I warmly welcome my colleagues and the event coordinator, Dr. Ramakrishnan S. Once again, his enthusiasm and devotion have been evident since the inception of this concept. I applaud him for that. I wish all the delegates a pleasant stay and welcome you again. I remain. Thank you.
Thank you, sir. With immense pleasure, I extend a gracious welcome to Reverend Father Paulos Kidangan, CMI, Manager, Sacred Heart College, Tevara, to deliver the presidential address.
Most Honorable Nobel Laureate and key speaker of the day, Professor Dr. Ada Yonath, Honorable Vice Chancellor of Mahatma Gandhi University, Dr. Sabu Thomas, Professor K.V. Thomas, former Union Minister and Cabinet Member of Kerala, Professor V.N. Rajashekaran Pillai, ex-Vice Chairman of UGC, Professor Dr. Rosa, Professor Dr. Tamoki, Professor Dr. Yatik Tor, Dr. Srinivasan, Principal Reverend Dr. Jose John CMI, Dr. Franklin, Head of the Department Chemistry, Dr. Ramakrishnan S., Organizing Secretary, Reverend Dr. Sebastian Borsar, distinguished professors from around the world, faculty members, academic luminaries, and my dear friends. Today, when I stand before you, I feel greatly honored and I treat it as a rare privilege to address this elite gathering on the occasion of the inauguration of 23rd Professor K.V. Thomas Endowment Seminar, which is now an international symposium on the topic New Trends in Applied Chemistry, NTAC-2023. This time, we have fortunate enough to have Professor Ada Yonath, who is a renowned scholar in the domain. Professor K.V. Thomas, fondly called as Thomas Marshall, and Professor is one of the greatest alumni of Sacred Heart College. During his political career, he ensured that Hartians form a part of his life, and as such was his home par excellence by all means. He ensured that the best is delivered to us by all means, and he tirelessly catered to all our needs in men and material. As manager of Sacred Heart institutions, we are sure that we wouldn't have achieved this much without his constant and reassuring support and encouragement. Professor Thomas was there in all moments of our joy and sorrow, and by instituting this endowment, he made it sure that we are getting the best in the form of world-famous academic exposure that transforms SH into a hub of learning and discourse. I congratulate the Department of Chemistry for picking up the threads of this highly versatile endeavor and taking it forward so successfully in all these years without astounding success. On my personal behalf, I express my heartfelt gratitude to Professor Ada Yonath, Professor Sabu Thomas, Dr. Rosa, Dr. Yatik Tor, Professor Tamoki, and Dr. Srinivasan for inspiring with us your visit. I hope this symposium will play a key role in bringing all researchers together to discuss current areas of research and reflect upon the advances in scientific area. Thank you all.
Thank you, Father. On this auspicious occasion, it is a privilege to have Honorable Vice Chancellor of Mahatma Gandhi University, Professor Dr. Sabu Thomas, for the inaugural ceremony. With immense pleasure, I invite Professor Dr. Sabu Thomas for inaugurating NTAC-2023 by watering the plant. This bonsai plant, which is an Indian laurel, also known as Malayan banyan, was planted in our college 7 years ago. I request all the delegates to join with Professor Dr. Sabu Thomas for this green inauguration. I request all the dignitaries to stay back on the dais as we are heading for the release of our proceedings. May I kindly invite Professor Dr. Adai Yonath to release the proceedings and Professor Dr. Sabu Thomas to hand over the same to Professor Dr. Adai Yonath. I would also like to invite Dr. Thomasakutti Jose and Dr. Shamna M., editors of the proceedings, to join for the same. Thank you all. I request all the dignitaries to take their seats. I am extremely privileged to welcome Professor Dr. Sabu Thomas, Vice Chancellor of Mahatma Gandhi University, to deliver the inaugural address. Sir, please.
Very good morning to all of you. The president of today's meeting, Reverend Father Paulus Kidangan, CMI manager, Nobel laureate Professor Adai Yonath, Professor K.V. Thomas, Professor Rasharan Pillai, Dr. Franklin John, Reverend Dr. Jose John, CMI, Principal of the College, Dr. Ramakrishnan, and all my international colleagues from different parts of the world. Professor Hossa from Brazil, Professor Cho from United States, Professor Chamoki from Japan, Professor Srinivasan, Nicer, my good friends, Professor Biju, Professor Maiti, and Professor Suresh will be joining us probably in the afternoon. Dr. Hagi Yonath, and all my friends, colleagues, students. You know, as the Vice Chancellor of Mahatma Gandhi University, I'm really excited today because this college has been consistently doing very well in academics. Let us give a big clap to the college. And I'm glad that the college is organizing the 21st Professor K.V. Thomas Endowment Seminar and 4th International Symposium on Trends in Applied Chemistry. My special congratulations to the Department of Chemistry in organizing such a beautiful function. And I'm glad that Professor Ada Yonath is with us today, which I could actually coordinate the meeting. Professor Ada Yonath has been visiting India for quite some time. This is her 25th visit. And she likes India. And she has been interacting with academics and students of India for a long time. She came to Mahatma Gandhi University and interacted with our faculty members and students, and we had a wonderful time. Let us give a big clap to Professor Ada for his exciting trip to India, interacting with us. Professor K.V. Thomas, an academic turned politician, has shown great interest for academics of this college, academic work of this college. And Professor Thomas has been doing excellent work, philanthropic work, for elevating the academic standard of this institution for a long time. And this is probably the third time I'm attending Professor K.V. Thomas endowment lecture and seminar. And this time the Department of Chemistry is organizing an international symposium as part of it. I remember when Professor K.V. Thomas was Central Minister, we could bring him to Mahatma Gandhi University. And I remember he actually opened my international conference, India-US International Conference on polymers for packaging. That time, Professor Thomas was the Central Minister of Food. And he has been giving us a lot of support. Right now, Professor Thomas has taken up a new assignment in Delhi, and we wish him all the best and good success, Professor Thomas. Dear friends, colleagues, conference symposia are extremely important. These give us a lot of opportunity to interact with the rest of the world. I remember when I was a PhD student at the Indian Institute of Technology back in 84, 87, my advisor used to send me lots of conference symposia that gave me a lot of opportunities to interact with the rest of the world. I remember when I was a visiting scientist in North America, my advisor used to go for all the SES meetings, Professor Robert Prudhomme. And Professor Prudhomme used to tell me, Sabu, if you go to SES meetings and if you attend all the sessions very meticulously, you understand the latest advancement in the subject. Therefore, conference and meeting symposia give us a lot of opportunities to interact, to meet the stalwarts in the field. Since I joined Mahatma Gandhi University, we took a statistics. I joined sometime in 87. Professor Pillai was the director of the School of Chemical Sciences. And when we took a statistics, we found that Mahatma Gandhi University has organized very close to 500 international conferences in all the schools. And we found that there were more than 10,000 people from different parts of the world actually visited our campus. And my own field, in polymer science and nanoscience, we organize a large number of meetings and symposia. So you name any country in the world, I have at least 10 academics as my good collaborators. Therefore, conference and meeting and symposia has a lot of advantages. We have signed MOUs with more than 200 universities across the globe. And we have very active cooperation with many institutes. Therefore, I request the chemistry department keep up this tempo and organize meetings, symposia. This is extremely useful for the students. I request all the students to interact with the leading speakers in this conference. I'm glad that the Department of Chemistry is bringing lots of people to this campus. So I wish all the best for this meeting. With all your permission, I declare that the 21st Professor K.V. Thomas Endowment Seminar and 4th International Symposium on New Trends in Applied Chemistry have been inaugurated. And I wish all the best for the conference. And I find that this conference is extremely interdisciplinary, because when you talk about applied chemistry, you deal with all branches of chemistry: medicinal chemistry, polymer chemistry, pharmaceutical chemistry, materials chemistry. My colleague from Japan is a brilliant materials chemistry. So this is truly an interdisciplinary conference. And I wish all the best for the conference. Thank you so much.
Thank you, sir, for your enlightening words. Next, I would like to invite Professor Dr. V.N. Rajashekharan Pillai, Honorable Vice Chancellor of Somaya Vidyavihar University and Provost of Somaya Vidyavihar and Somaya Ayurvihar under the Somaya Trust, to felicitate the occasion. Sir, please.
Chief guest of today's function, distinguished protein crystallographer, Professor Ada Yonath, President of this function, Father Paulos Kadangan, CMI Manager, Professor K.V. Thomas, formerly Professor of Chemistry of this great institution, and afterwards a very prominent political figure in the country, former Union Minister, Professor Sabu Thomas, the Vice Chancellor of Mahatma Gandhi University, Kottayam, to which this college is affiliated, Dr. Franklin and Dr. Ramakrishnan, who are the coordinators of this international seminar, distinguished experts from outside the country and also from our country, dear young students who are participating in this two-day international conference, and other distinguished invitees. Daughter of Ada Yonath, she is also a guest with us. I recognize her presence as well. First of all, let me congratulate the SH College Devara, the management and the organizers of this conference for hosting in this institution a very important conference on the occasion of the Professor K.V. Thomas Endowment Lecture Series. I had occasion to visit this college several times. When I visited chemistry department, when Professor Gopalakrishnan, Professor Devasya, such people were teaching in the chemistry department as an examiner. And from there, after I joined the Mahatma Gandhi University in 1983, in the very inception, I used to interact with the institution. And then, of course, I gave one of the Professor K.V. Thomas Endowment Lecture also. This is an institution established in 1944. And there is a lot of scope for taking initiatives and giving models to other institutions by this college. I remember this is a college accredited by National Assessment and Accreditation Council in 2000. I was heading the National Assessment and Accreditation Council at that time. It has organized very significant activities in science, in all other areas also. And I am very confident that institutions of this kind should become universities in the near future. Of course, I know that this is an autonomous college, twice accredited, and promoting the independent initiatives in academics. It is very important to provide complete autonomy to institutions. The National Education Policy strongly emphasizes the importance of providing complete autonomy to the academics. And this college could be a model for providing or getting such autonomy, and then that could emulate many other institutions in the Kerala state to become centers of excellence. Professor Ada Yonath is giving the inaugural plenary lecture here. She's a woman of science. Her trials and tribulations to become a great scientist, it is, I mean, probably it's very good, particularly for young girl students, to read her biography. It is very, very important to read her biography. I remember I hosted her when I was heading the science and technology in the state, in Trivandrum, along with a crystallographic conference. Then she visited to inaugurate the startup, 100 startups in Somaiya Vidya Vihar University in 2018, to inaugurate chemical startups, chemical and biological startups. And her presence in this conference, her address in this conference can really motivate the young students to come up. It is not only that you hear her lecture, but you should just find out how she achieved this with a MSc chemistry and then going to ribosomes and all that, the achievement. And it is also very important. By bringing such people of eminence into the institutions, this is the thing which we are achieving. And let me congratulate the college, Dr. Franklin, and all his colleagues for doing this wonderful job of getting together eminent scientists and the Nobel laureate, and also the giving the students and youngsters an opportunity to listen to them. Thank you very much.
Thank you, sir. I invite Reverend Dr. George Jones, CMI, Principal of Sacred Heart College, Tevara, to felicitate the occasion.
A warm good morning to one and all. This is a great honor for SH College Tevara to have this 21st Professor K.V. Thomas Endowment Seminar and 4th International Symposium on New Trends in Applied Chemistry. The reason behind this great joy is none other than the presence of Nobel laureate Professor Ada Yonath and several eminent scientists from different parts of the globe. And it is being organized by the Department of Chemistry, a symbol of coordination and collaboration among themselves and with other departments of this college. The motto of our college is Cor Rectum Inquirit Scientia. It's in Latin. Its English translation may be like this: a righteous heart seeks after wisdom. So this is a moment, a continued journey in the path of wisdom seeking. As Professor Sabu Thomas rightly said, and also Professor Rajashekharan mentioned, Sacred Heart College from its very inception in 1944 strived to build this interconnectedness among various departments. We have 20 departments here, different programs, and always there is this connectedness between the departments and subjects. So this seminar itself, as Professor Vice Chancellor mentioned, it is an interdisciplinary subject, biochemistry and molecular biology, as they are closely interconnected and interdependent fields of study. The deliberations here will definitely enrich each and every participant, especially young, vibrant seekers of wisdom students, how these various branches of science can be interconnected, especially when it comes to research and innovations. So the deliberations will be of a very, very high and advanced, I hope, and I believe. It will definitely open up the hearts and minds of young students here so that they can have new light on which how to focus their studies or how to orient their future. And I rightly appreciate the words mentioned by all these great men on this college, about this college. And I humbly request each and every participant, student participant especially, to interact with these great eminent professors and scientists here. That will be the most advantage that you will get from this symposium or seminar. And I do believe that as these great men, great scientists, including a Nobel laureate, they are so humble. And they will be very happy to interact with you and also to answer your questions and queries. And they will be happy to help you and guide you in your future career and research or whatever be. I place on record all the eminent personalities for being here, and I wish all the very best for this conference and seminar. And I should specially mention Professor K.V. Thomas, because he is instrumental in establishing this endowment seminar several years back. But this year itself, he had been here for more than 7 or 8 times in person to organize, to discuss, and to give guidance for the smooth conduct of this symposium. And I congratulate Professor K.V. Thomas for his new assignment in Delhi, and also I thank Professor Ada E. Yonath for being with us at SH College Thavara, Cochin on this 7th day of February 2023 and making it as a ever memorable one. May the Sacred Heart of Jesus bless us all. Thank you very much.
Thank you, Father. Now I invite former Union Minister and the current State Representative to Central Government, Professor K.V. Thomas, to deliver the felicitation. Sir, please.
Nobel laureate, Dr. Ada Yonath, Vice Chancellor of the Mahatma Gandhi University, Professor Sabu Thomas, former Chairman of the UGC and Vice Chancellor of MD University, Father Paulos Kidangan, Manager of the College, Father Jose John CMI, Principal, eminent scientists who have come from outside India and from India, the scholars and students who are participating in this seminar. As you are aware, in 1947, as Pandit Jawaharlal Nehru, the first Prime Minister of India, said, a new nation is coming into existence. And just 3 years before, this institution came into existence. It was a new challenge by the CMI fathers. And fortunately, my eldest brother, Joseph, was the first student in the first batch of zoology botany combination. I happened to come to Secretariat College. I am 14 years younger to my elder brother, but he insisted that I should study in Sacred Arts. CMI Fathers, Carmelite Mary Immaculate, is an Indian congregation. Many of the Christian congregations are foreign origin. But CMI congregation was started, given shape by Saint Chavara Kuriakos, who has initiated the overall development of education in Kerala. He is the priest who said, along with every church, there should be an educational institution. So he is the person who started education for the development of the common man. Now I came to the college when I was 14 years old, and we are all staying in hostels run by the fathers, and they were very strict. Nowadays, no, for the last so many years, I get up at 4:30 in the morning because my wardens, fathers, who used to ring the bell at 4:30, we should have go to the bathroom, washing room, then bathing and etc. Come back at 5:30, go for the prayer. So there was a system. And I still follow that system. I am going to be 77. This college has produced men of literary letters, in the political field, scientists, and eminent personalities. So whenever I come to this institution, I am proud of this institution. And Kerala, as well as India, is moving steps by step in the primary education as well as the higher education. India is one of the growing nations in the world, giving leadership. Our Prime Minister Narendra Modi is the head of the 20 G countries, and all the meetings are taking place. So I think this conference today, where Nobel laureate Ada Yonath and other eminent scientists are participating, will give a new boost to Kerala and will give a new path to India. We are all happy that you have all come over here, God's own country. I was the tourism minister of Kerala for a quite long time. And Kerala is one of the well-known destinations in the tourism development. So I request and welcome all of you to spend some time. And I think tomorrow you are coming to my village, Kumbalangi, which is the model tourism village in the country. You can spend some time in the evening with the villagers and see how we have developed in the tourism sector. So once again, I welcome Nobel laureate Ada Yonath, her daughter, or other eminent scientists to Kochi, to my village, Kumbalangi, and to my institution, Secretary College.
Thank you, sir. Now, let me call upon Dr. Ramakrishnan S., Organizing Secretary of NTAC-2023, to propose the vote of thanks.
Respected dignitaries on and off the dais, I am both humbled and honored to be given the opportunity to express my sincere gratitude to all present here on behalf of the Department of Chemistry and my own college. The respected President, Father Paulos Kedangan CMI, our manager, is always a constant source of inspiration and support to all of us. I extend our gratitude to him for his presence and blessings. Thank you, sir. Nobel laureate Dr. Ada Yonath deserves our boundless gratitude not only for kindly consenting to be with us today, but also for her invaluable contributions to science. I thank her from the bottom of my heart for her inspiring presence and her release of proceedings. Thank you. Thank you. Our chief guest, Professor Sabu Thomas, Vice Chancellor of Mahatma Gandhi University, has graced this occasion. We are grateful for his presence here and his tireless efforts to foster research of the highest order in the university. Thank you, sir. This seminar and symposium would not have been possible without the support of our former head of the department and former colleague, Professor K.V. Thomas. This annual academic event is a testimony to his passion as a teacher, which as vibrant as his commitment as a public figure. I extend our warmest thanks to you, sir. Let me thank Professor Itshok Tor, Professor Tamoki Nobioki, Professor Dervel dos Santos Rosa, Professor Srinivasan, and all the other plenary speakers for graciously accepting our invitation to deliver lectures at this event. Surely their talk will illuminate and inspire all of us. Thank you. Our principal, Reverend Dr. Jose John CMI's keen leadership is the key to organize an event of this stature, sir. So I extend our heartfelt thanks to you, Father. Thank you. Our young head of the department, Dr. Franklin John, has demonstrated exceptional leadership and organizational ability in making this event possible. Thank you, sir. And on behalf of the Department of Chemistry and College, we sincerely wish all the participants fruitful 2 days here. Thank you. Thank you very much.
Thank you, sir. With this, we have come to the end of the inaugural ceremony of NTAC 2023. I request Professor Adai Yuna to stay back on the dais for the Nobel lecture, and all the other dignitaries can take their seats in the front rows. Chemistry is not merely a subject. It is an amalgam of observations, experiments, inferences, serendipity, convolutions, realizations, questions, answers, research, and exploration. The chemistry department of Sacred Heart College was established in 1944 with the intention of providing the best education to all those who have the passion to learn chemistry. K.V. Thomas Endowment Seminar is the most prestigious event organized by the Postgraduate and Research Department of Chemistry. This is the 21st K.V. Thomas Endowment Seminar and 4th edition of NTAC. This year, the event is even more graced by the presence of Professor Dr. Adai Yonath, Nobel Prize winner in Chemistry in 2009. Now, we are moving on to the most awaited session.
Sorry, I forgot to express my sincere thanks to Professor V.N. Rajashekharan Pillai, sir. I am extremely sorry, sir. Okay, sir, we are grateful for his presence. Thank you, sir.
Just a second, I have to check.
Now, we are going to have a Nobel lecture by Professor Dr. Ada Yonath, who won the Nobel Prize for Studies of the Structure and Function of Ribosome in the year 2009. The session will be on the topic from origin of life to next generation RNA-based therapeutics. The session will be chaired by Professor Dr. Sabu Thomas, Honorable Vice Chancellor of Mahatma Gandhi University. It's my pleasure to introduce him to the gathering. Professor Dr. Sabu Thomas is the founder director of International and Inter-University Center for Nanoscience and Nanotechnology of Mahatma Gandhi University. He has 5 Indian patents to his credits. He is cited in the list of most productive researchers in India, in which he holds 5th position. Professor Dr. Sabu Thomas has been listed in world's top 2% scientist by Stanford University, USA. He has collaboration with 45 institutes around the world. He has more than 1,000 publications. He is also listed in the most cited researchers in material science and engineering by Elsevier Scopus data. He is also the former Dean of Technology and Applied Sciences and Syndicate member. Some of his major scientific field of interest are polymer blends, fiber-filled polymer composite, particulate-filled polymer composite, and aging and degradation. He has received many awards and prizes. Recent awards are National Researcher Award DST Nanomission 2020, Award for Outstanding Contribution by Mahatma Gandhi University, and Dr. APJ Abdul Kalam Award for Scientific Excellence. I request Professor Dr. Sabu Thomas to please chair the session.
Dear colleagues, I'm extremely delighted to chair today's talk of Professor Ada Yonath. Let me formally introduce Professor Ada Yonath. Professor Dr. Ada Yonath is a Nobel laureate, crystallographer, best known for her pioneering work on the structure of ribosomes. She is the current director of the Helen and Milton Kimmelman Center for Biomolecular Structure and Assembly of the Weizmann Institute of Science, Israel. In 2009, Professor Yonath received the Nobel Prize in Chemistry along with Professor Venkataraman Ramakrishnan and Professor Thomas A. Stites of her studies on the structure and function of the ribosomes, becoming the first Israeli woman to win the Nobel Prize out of 10 Israeli Nobel laureates. The first woman from the Middle East to win a Nobel Prize in the sciences, and the first woman in 45 years to win the Nobel Prize for chemistry. Her research has also been keen to understanding how antibiotics work. Let me also tell very briefly about the early career of Professor Yonath. In 1968, Professor Yonath completed a PhD in X-ray crystallography from the Weizmann Institute, Israel. Professor Yonath accepted postdoctoral position at Carnegie Mellon University, United States, 1969, MIT, United States, 1970. She was a chairperson and a senior scientist in the Department of Structural Chemistry at the Weizmann Institute of Science in Israel. She held the director position at Maser Center for Structural Biology, Weizmann Institute. She worked as a visiting professor in Max Planck Institute for Molecular Genetics, Berlin, Germany, Universidad Austral de Chile, University of Chicago, Dental School of University of Alabama, United States. She is a member of the prestigious professional organizations like Israeli Academy of Sciences and Humanities, Royal Society of London, European Biological Organization. Apart from Nobel Prize in Chemistry, 2009, she was the first recipient of several awards, including Albert Einstein World Award of Science, Distinguished Science Award, Galapagos, 2013, the Röntgen Medal, Germany, 2015, India's Prime Minister Gold Medal for Outstanding Scientific Contribution in 2011 and 2017. She received the Honorary Doctorate of Science from University of New York at Albany, USA, University of Strasbourg, France, University of Southern California, United States, Warwick University, UK, and many other esteemed institutions in the world. She was a member of the various education advisory committee in Israel and principal users of Synchrotron Radiation ESRF France, APS Argonne National Laboratory United States, Cornell High Energy Synchrotron Source United States. The Scientific Advisory Board of the UN Secretary General, Mr. Ban Ki-moon, she is currently a member of the Scientific Advisory Board of the Health Science Platform at Tangcheng University, China. She is an active member of the Advisory Board for Sustainable Development Goals, UN headquarters. Professor Ada, this is your 25th visit to India. We are all excited to listen to you. Let us give a big clap to Professor Yonath. And Professor Yonath, we are all looking forward to listening to you. Thank you.
Do you have something here that is not so slippery?
Yeah,
Thank you.
yeah, perfect.
No, it's not perfect. Can you put off some of the lights? Can you put off some of the lights? What are you doing? Oh, please don't play with my computer.
Excuse me.
So good morning, everybody, and I'm very thankful for being able to tell you about our studies and to thank you for all these wonderful words you said about me. Not all of them are really correct or very important. And I want to talk about what we have been done about ribosomes. Of course, not everything, but I picked up the more exciting chemical aspects of the work. And I want to talk about all what we can. And we are not able to say much, but whatever we can about origin of life, from it to genetic diseases. So first, just to remind you what the ribosome is doing. So the green thing is the ribosome. It's made of two subunits. Small subunit is reading the instructions from the DNA. Actually, the instructions from the DNA are not brought to the ribosome. What is brought is messenger RNA, mRNA. Now everybody knows what is mRNA, owing to the new products. Some of them are for vaccination of COVID-19. So mRNA is coming. mRNA is made of nucleic acids. Each nucleic acid has an anti-codon loop, which essentially tells which amino acid will come in. So they are getting into the ribosome and incorporating with the tRNAs that are bound on the other side to amino acids or to the growing protein, of course. So there are three sites, A, P, and E. A is for the new tRNAs that will come in, and once it stands here, it can combine with the growing chain on the tRNA at the P site. Then it goes out, becomes a protein at the end, and after the bond between the two amino acids is being made, the P site tRNA immigrates to the E site, the exit site, and goes out. That's all what the ribosome does. Provides reading and writing framework for the genetic code of the newly born proteins. So ribosomes are very widely spread in all cells. And in liver cells, for instance, just as an example, they can contain up to 6 million ribosomes, 6 million ribosomes per cell. In other cells, there are smaller numbers, but still very large ones in millions. Even in bacteria cells, 50 to 80,000 ribosomes can exist. So the first thing we did, and it took 20 years to get there, is to see how ribosome actions. And I hope it works, even here. Yeah, it worked yesterday, so it should be okay. What you see here, this line, this very faint line is messenger RNA, which reaches the first part of the ribosome, the small ribosomal particle, goes into its path. There is a specific path for it, which is monitored by initiation factors. What you see here is a bacteria. The initiation factor is this little piece in blue at the end, which make sure that the messenger came into its correct place. Why this is achieved, the first amino acid can be brought by the first tRNA with another initiation factor, this light blue initiation factor number 2. In bacteria, there are 3 initiation factors. So initiation factor brought it in and made it bound correctly with the amino acid. You see this little dot in blue? This is the size of amino acid compared to the whole ribosome. Once everything is fine, the factors are leaving and the large subunit can come and accommodate itself by bridges with the small one. And now the ribosome is ready to work. We look at it from the outer side. Amino acids are coming in with factors, elongation factors. The ribosome helps in, out of the large subunit, the ends of the large subunit. Now we took away the large subunit, and you can see only the small one and how things are happening. So the tRNAs are moving and being read, and the peptide bond is being made on the other side of the tRNAs. You can see here just the peptide bond and the tunnel for which the newly born protein will come out. Maybe later on I will tell you stories about the tunnel, but now you can see how the ribosome works. Now you can see the whole ribosome again and how it continues. And the protein becomes larger and larger and coming out and folds correctly. And then other factors coming, recycling and erasing, and the ribosome can be separated to the 2 subunits. The newly born protein can go and do what it needs, and the whole system is waiting for the next job to do. That's all. I want to tell you that we made this movie with 2 young scientists, computer scientists, and they were so happy with the result that they started a new company, Z Animation, just on the accounting of this, and this our second movie that I will show you in a minute. So here you can see the components of the bacterial ribosomes. I want to say that in higher organisms, the 2 components exist. Also, the main regions of function are conserved. What is happening is that they become larger. It's many more pieces, but not at the active sites. So the small one on the left and the the large one on the right, they're both made of ribosomal RNA, which is their main component, and many, many ribosomal proteins. Each of them is shown here in a different color. And just to remind you again, the small subunit is responsible for decoding and the large for peptide bond formation. So if we look at it at the side, from the side of active ribosome, small subunit on top, large on the smaller one, they are connected by their own interactions, but also by tRNA, which is reading the code here and producing the protein here like that. So the main active parts, active regions are this one and this one. The reading, the decoding center, and where peptide bond is being made, which is called PTC, peptidyl transferase center. From the other side, it looks like that, small subunit, large. The reading happens here, and the bond is made here. And I want to focus on how the bond is being made. I think it's interesting anyway, but here for sure, because this is the chemistry. So this is the large subunit, here is the peptide bond formation, the PTC, and it looks like this. What I will keep from now on is the A site, where the new amino acid is coming in blue, and the P site is after the bond is being made is in green. So you can see here, this is the way it looks, with two pieces of, or two end pieces of the amino acids. Or you can see it here, and if I can show you the very important piece, this is the same in all ribosomes, no matter if it's bacterial, if it's eukaryotic, if it's whatever. It's made in two pieces, which are at this point completely same, completely similar in all organisms. So just to remind you what is tRNA, we looked at it earlier, but I want to focus now on this part. So here is the decoding part. This is the main feature of tRNA, which is very highly conserved from bacteria to elephants, if you want, or to us. And the end of it, which is fully conserved everywhere, always CA, cytosine adenine, and amino acid. Always CA amino acid, and this is what binds to the PTC. So if we look at its position more carefully, we see this is the position of the CA amino acid, of the P, of the A and the P site. And when we look at it, it is directly into the tunnel, the ribosome exit tunnel, and it has a semi-symmetry. Actually, it's almost full symmetry, but because everything is here left or right-handed structure, it is only semi. So if we look at the path that the A site tRNA has to make until it goes to the P site, you can see here how it moves from blue, the A, to green, the P, and the interactions that it makes, or the contacts that it makes meanwhile, while going from one to the second with the amino acid, with the tRNA, sorry, at the border of the tunnel of motion. When this is, again, fully conserved. So we proposed that because it's fully conserved, and because it is existing in all ribosomes, and because it didn't change from the smallest ribosome to the biggest known today, we propose, owing to its high conservation, that this semi-symmetrical region indicates that its existence is beyond environmental conditions. It means it was before evolution. This is our proposal that was not wrongly shown. And if you want to look at it in one summarized slide, so this is the large subunit, this is a position. It looks like this from top and like this from the side. And it is universal. And just to show you some of the universality, we superpose the main chain of the two parts of a bacteria, of halobacteria, of very robust bacteria, in yeast, in how you call it, parasites, and in human. It's exactly the same. Once we drew one of the on the other, we could not distinguish between them. So if there is a protoribosome, why shouldn't build it? Shouldn't build it in the lab? So we talk about this part, this blue. I hope that you can see. This bluish part, or cyan part, of the whole large subunit RNA map, two-dimensional map. And I highlight here the pieces that are similar in all ribosomes at the P site and at the A site. And this all belongs to this part. So when we constructed it in the lab in order to show that it really exists, we cut it here and here and here and here because the ribosome grew with evolution, not this part. So when we constructed it, we could make changes in the contact between the cut pieces, what we call mutations. With time, we could play with it, although it's much less important. What's important are the conserved sites. And we got, when we built it, we got the structures that actually we expected. You can see it here with the substrates, also here with the substrates from a different viewpoint, and we are looking at these two parts of the PTC, which means that the semi-two-fold symmetry within the asymmetrical contemporary ribosomes is the way ribosomes started, or the way proteins started to be made. So with years, we spent on it many years, almost 15 years, but after about 10 years, an Indian postdoc came to our group, joined us. Her name is Tanaya Bose, and she was able to make all these compounds, all these what we call proto-ribosomes, because they are before ribosomes. All these that are shown here in the table, I hope you can see in this column, and looked which ones of them are stable. And many of them are stable. Actually, all what I'm showing here are stable as proto-ribosomes. So next thing she tried is to see which of these proto-ribosomes can make peptide bonds, because this is actually why they are there. And you can see here, oops, sorry. Oh, I'm sorry. You can see here, these are making peptide bonds that are highlighted. It means 5 out of the 13 proto-ribosomes that we had. which shows that dimerization is not sufficient for peptide bond formation, but peptide bond formation is what we are looking for, what evolved later to the whole ribosome. So this, the fact that not all of them are suitable may explain why ribosomes developed in a specific way, and not all proto-ribosomes led to fruitful ribosomes. So how do we suggest nature went from the proto-ribosome to the modern ribosomes? So we think that throughout the sequence of each component of the central region has been genetically optimized for its functional task. And this is indicating that function is superior to sequence. Not every sequence can make function. Function needs more, needs from the chemical point of view, positioning in the way that a bond can be made. And this explains the lack of sequence symmetry in the symmetrical region. It means many sequences could be there, and those that survived are those that could make peptide bonds. Now, once we talked about Tanaya Bose's achievement in making peptide bonds, let's go to other subject of this lecture, and these are the antibiotics. It's clear that because of the fundamental role played by the ribosomes, many antibiotics target them. Actually, almost half of the known antibiotics, the clinically useful antibiotics, target protein biosynthesis, and mostly by paralyzing the ribosome. Not all, some are interfering with messenger reading, but I'm talking now about the function of the ribosome. So the question that we asked was, how do the tiny ribosomes, tiny antibiotics paralyze the giant ribosome? For antibiotics, it's really giant, because bacterial ribosomes, which is the target of the antibiotics, have a molecular weight of 2.5 million Daltons, whereas the antibiotics are less than 1,000, between 500 to 1,000. So all are really very clever. They target the functional sites of the ribosome. For instance, here you can see the PTC. You see a section through the ribosome with tRNA in the P site, growing proteins coming out, and this is the PTC. And the first one that was brought into usage as an antibiotic was not as the first antibiotic, but the first ribosome antibiotic was erythromycin that targets the top part of the tunnel just next to the PTC. So if we look at all the others, we can see that they are targeting the decoding, the hinging, the bonding, and the tunneling. I will not talk about all of them, but I just want to show you that all these antibiotics that are written on all these sites are targeting very important, very functional sites of the ribosomes, the bacterial ribosomes. Let's just look at those that bind to the top parts of the tunnel, which are macrolides, and erythromycin is representing them. This is the red thing here. The tunnel goes from the board towards you. We look at it from top, and the ribosome here is in green. So we made a little clip together with the same two students. And it's fantastic. We worked, maybe I will stop here just as it. We worked almost 20 years to understand how the ribosome is built, to determine its structure, and another 3 months to understand how the antibiotics or some antibiotics paralyze it. So let's look here at edeine. Edeine is binding to the small subunit. It was discovered in Poland, and look how clever and how little and how important. Actually, edeine is hardly being used because it doesn't distinguish between the patient and the bacteria, the parasite. The second one that you will see is targeting the A site, and it's called tetracycline. You can see this. Sorry? Sorry? It sits in the P site and doesn't let the second tRNA bind. The third one that you will see is the one that we looked at earlier at the entrance to the tunnel, erythromycin. Sits exactly here. And the last one that I want to show you is really very clever antibiotics. Look, it mimics nature. It goes exactly where nature needs it, into the bone. So these are the antibiotics that are known, and what's also known and very disturbing is the resistance to antibiotics, which is one of the most severe problems in modern medicine. Can we combat resistance to antibiotics? I'm not sure, but I want to show you what we are doing. Because most of the pharma companies stopped looking, stopped searching for new antibiotics, owing to the expected resistance and the low price. So we did not think about price and so on. We thought about antibiotics. I want to, first of all, to show you the increase of drug resistance on the left. So we look here at 3 bacteria, 3 disease-connected bacteria. And this is the number of new antibiotics. Here we look at the increase in 12 years. And here we look at the approved new antibiotics in 30 years, from 1984 to 2014. That's all. 7 new antibiotics. So this is why we are trying to make new antibiotics. We are not taking the way that companies are using, that changing the macrolides, improving the macrolides, but we are designing new generation environmentally friendly antibiotics. And for this, we have modest contributions, but I want to tell you about. So we determine the structure, the ribosome structure of from a pathogen and from non-pathogenic. The non-pathogenic here shown in gray, and the pathogen pieces that are different in cyan. The rest is the same. So you can see that there are several different pieces, and we think, like, let's focus on one of them, that it can be a potential binding site to new antibiotics. Actually, in Staph aureus, which is a pathogen, we could put all these potential ones, and we made for each of them a matching molecule. You can see here even in more structural way, but I won't go into detail. We could make a species-specific targeting compounds that you can see here, and some of them did stop ribosome work. So we are working now on producing them in a way that human beings will be able to use it. The last few minutes are for another point that we are working now on, and these are genetic ribosomal diseases. So 25 of the cancers, and a very important disease that is a blood-related disease, DBA, have different mutations in ribosomes. It's not clear if the different mutations cause the disease or came in the disease, but they are there. For instance, in DBA, which is an anemia, 97 different mutations were found in DBA of ill people. Still, it's not clear when it comes in, if at all they are expressed. We know them only genetically. Actually, with the aid of the lab of my daughter here, Hagit Yonat, but there are many. Here there is a presentation of cancer and DBA, and you can see this 25%, very, very highly located in specific ribosomal proteins. And you have here the list, which is really of less important, but what we are trying is to find out which of these mutations, if it is expressed, may change the surface of the ribosome. You can see here the ribosomal proteins that have mutations in both sides of the ribosome. They're mostly on the surface, so we expect that they will contribute to the surface either directly or through the RNA that is next to them, the brown piece, and then we will be able to make antibiotics for them. So the targeting is based on the identification of specific structural changes. It's too long to read, but this is the platform that we are trying to produce now. Before I finish, I want to show you the group members, not all of them, and most of them have left, only the current ones are shown in this little suns. Tanaya Bose, that I already announced, that is senior postdoc, she just finished postdoc, is shown here. And on the genetic diseases works Disha, who is also an Indian, and Rajan, that came only a few weeks ago and is still not in the picture. So thank you very much. And my head is full of ribosomes.
Thank you, Sada, for your exciting talk. It's open for questions and comments. Please, yeah. Yes. Okay, thank you.
A quick question, Ada. You talked about the proto-ribosome, so in making peptides. Kinetically, how fast or slower it is compared to a native, say, bacterial ribosome in terms of amino acids per minute or seconds?
It's much slower, much slower in the lab, but maybe in nature found ways to make it faster, like found ways to make the ribosome work very, very fast. But part of the whole idea of the proto-ribosome is that it was slower. And only those that could become faster, they had there was a reason for their winning the game. Yes, please.
So ribosome is very special. So we may call it a king of the catalytic polymer. So it transfers the information of the messenger RNA to totally different polymer of amino acid, right? So could you find any quite special structure for the specially functioning ribose.
Well,
Are
we
there
think that this pocket that we call the protoribosome is special. I don't know about anything like this, but maybe I don't know enough.
I see. So the information transfer must be very precise, right? So are there any special structure for maintaining such preciseness?
Yeah, but this is not in the ribosome, it's before the ribosome. It's where messenger RNA is being produced according to the DNA. This has to be very precise and also relatively quick, depending on the body needs, but it's usually quick.
I see.
It's much more important than the ribosome itself that accommodates every sequence and can make peptide bonds. Ribosome is less selective. The selectivity is more in the first step.
Okay, thank you very much.
Ma'am, you talked about oncoribosome concept. Oncoribosomes,
Yes.
when it becomes cancerous, but ribosome, you also told that it is relatively conserved. So what might be the triggering factor to convert a ribosome into an oncoribosome? You have mentioned that it is relatively conserved.
I don't do this. Nature
No.
does it.
Okay, what might be the triggering
I'm not
factor?
interested in making more diseases, but I'm interested in understanding or treating non-diseases. And oncoribosomes, it's actually not very well defined name, because some say that it's ribosomes that mutations were found in their component. Some say that these mutations happened while there was cancer, and we are not in the situation to be able to answer this. Actually, it's not known whether these mutations are being expressed.
Okay, ma'am.
What is known is DNA mutations in the genes.
Yeah. Thank you.
So we think if we understand if this is expressed and the solution can be this or that, maybe we can add information which is medically useful.
Ma'am, what about the feasibility and expense of conducting drug discovery experiments based on these ribosome targeting compared to this conventional protein targeting? That means
Commercial protein what?
that means normally we are now most of the research is drug discovery experiments conducted on the basis of conventional this after protein crystallization compared to
Protein crystallization, you talk.
So compared to that experiments, what about the expense and feasibility of these ribosomal conducting these experiments?
Okay, so we determined the first structures actually crystallographically. We crystallized the ribosomes. We found that ribosomes that can be crystallized. This was the main problem in the beginning. We found those that can be, that are so strong, so complete that we could crystallize them. We used, in the beginning, we used mainly ribosomes under pressure that try to keep together. But now it's not needed. Now there is electron microscopy. Ribosomes are fully made for it, let's call it like this. They are large enough, and the main structure is known. So we look, not only we, the whole world is looking for differences, and we do it in cryo-electron microscopy. So the first structure came after 20 years, the second after 5 or 6, and the people that copied from us or repeated us, let's say it nicer, took them about 8, 9 years. Now it takes a few months.
Okay, thank you.
Very interesting. From PhD students and master students, please, questions to Professor Yonah. Dr. Jonas, I have a question. You also did a lot of synchrotron measurements to look
Yeah.
at the crystallization of
Yeah, this was in the beginning, part of the 20 years.
part of the yes.
The first part and the important part. The part that afterwards, for instance, we designed the cryocrystallography, that everybody laughed at us when we did it, and within half a year it became all over the world. So we did this too, and I'm very proud of it, but it's not needed now for ribosomes. It's needed for proteins, smaller proteins.
More questions? Yes. Yes. Please give a mic.
So you haven't shown us much of the structures of the test antibiotics that you made, but it seems to me from the table that they could have been short oligonucleotides. I might be wrong.
Yeah, of course, they might be wrong. Those that are completely wrong did not make dimers.
Okay.
Once we got dimers before that they are at least partially wrong, let's tell them to make a peptide bond. So from the 13 that we had as dimers, 5 made peptide bonds.
Okay,
Which means the dimerization is not sufficient for peptide bond formation, and that we don't really understand
Right.
which will do what.
And you used actually puromycin, it seems to me, which is an antibiotic on its own, to test this peptide
Yeah,
bond
we
formation.
did use puromycin.
Yeah.
We did.
Which
This was
is-
actually the test.
So
We
you
tested the. We used at least at one side a material that will stop the
Right.
growth, so the newly born chain will not run away, and we then will lose
Exactly.
it.
That would be interesting actually to screen for actually new puromycin analogs. So
Yeah.
I'll
Yes.
think about it.
Yes,
Thank you.
it's a really good idea. Thank you.
Thank you.
Professor Ada, are you also looking at the defects in ribosome biogenesis?
We assume that if there are differences in ribosome in ribosome sequence, it means in structure there will be a different biogenesis. But as long as we don't know whether we do or do not have the differences, we are working on it now. We don't know much about the biogenesis.
Yes. Please give the mic. Okay, okay.
You mentioned that the PTC is targeted by the antibiotics. So you also mentioned that PTC is mostly conserved across the species. So how does this antibiotic does it attack human cells? Only attack bacteria?
I'm sorry, but there are two different aspects. Antibiotics are not on cancer, are on diseases that are infectious diseases. Cancer is a new topic that we look into, and from the structural point of view, not from treating it, not yet from treating it. So antibiotics are good for a different type of diseases, infectious diseases, or maybe those that are connected to known differences in the ribosome. In bacteria, not in human. So I learned, if to continue this, I learned that all ribosomes work the same way. Who cares if it's bacteria or human? That's correct for how the ribosome works, but not how the ribosome is ill. Then the illnesses are different, or how the patient is ill. Then the differences in ribosomes between different levels in evolution are meaningful.
Professor Anna, are you also working with the pharma companies to develop environmentally friendly antibiotics?
If you find a pharma company that agrees to go into it, I'll be very happy to collaborate. But they all tell me, antibiotics, they're cheapies, they don't make lots of money, resistance will come before we have something, we are not interested, even in India. Usually they say it in India because there are so many diseases, maybe, but even the Indians are careful about this. And I look at it more scientifically, less profitably.
Still some questions with students. You know, there are no more questions. Let us give a big clap to
Thank you for inviting me. Although I was not very hopeful.
Thank you, ma'am, for the enlightening talk. Let's give a hands of claps once again to the ma'am. I invite Professor Dr. Sabu Thomas to hand over the memento to Professor Dr. Ada Ionath as a token of love. I also request Professor Dr. Ada Ionath to kindly receive the same.
Just a second.
Thank you, ma'am. Thank you, sir. Now I invite our beloved principal, Reverend Father Dr. Jose John CMI, to present the memento to Professor Dr. Sabu Thomas as a token of love. Thank you, Father. Thank you, sir. First session of NTAC-2023 has come to an end. Thank you, everyone, for your patient listening. Now, there is a group photo session, and I request all the delegates and participants to gather at the basketball court. Volunteers will lead you to the venue. After that, there will be a short tea break. It is arranged in the Marion Hall, and I request you all to gather back at sharp 11:30 in the Malicious Hall for the Plenary Lecture 1 by Professor Dr. A. Srinivasan. Thank you. And also a gentle reminder, during the lunch break, all the oral presentation
Thank you very much for joining us here in this 21st Professor K.V. Thomas Endowment Seminar and 4th International Symposium on New Trends in Applied Chemistry 2023. Now, we are moving on to our first plenary lecture by Professor Dr. A. Srinivasan on the topic Porphyrinoids with Polycyclic Aromatic Unit. And for me, this is certainly a distinct honor to introduce the chair of this session, Dr. Joshi Joseph. Dr. Joshi Joseph is the principal scientist at CSIR NIST Trivandrum and associate professor at ACSIR. He's the recipient of the prestigious Ramanujan Fellowship in 2012 from DST India and Raman Research Fellowship 2020 from CSIR Government of India. He is a life member of Chemical Research Society of India, Materials Research Society of India, and a member of Royal Society of Chemistry. His current research interests include biomolecular recognition, nucleic acid chemistry, and design of functional organic materials. With this, I welcome Dr. Joshi Joseph to chair the session. So please.
Thank you. Good morning, all. At the outset, let me thank the organizers for giving me an opportunity to visit this college and attend this conference. This is my first time in SH College, Devara. It's my pleasure to introduce my senior colleague and a friend, Professor A. Srinivasan, who is the speaker for the next session. Professor Srinivasan is currently a professor at NICER Bhubaneswar, and he has his bachelor's from Madurai Kamaraj University and master's from Bharatidasan University. After that, he did his PhD with Professor T.K. Chandrasekharan, IIT Kanpur. And after his PhD, he was a JSPS fellow in Kyoto University and Kyushu University. And in 2005, he returned to India and accepted a senior scientist position at CSIR NIST Thiruvananthapuram and spent 4 years there. And when the NICER is formed, he moved to NICER as an associate professor in 2009 and later became a professor in 2013. And he served 2 times as the chair of School of Chemical Sciences, NICER, and Dean R&T of NICER. And he works on tetrapyrrole-based macrocycles and basically photochemical application, especially photodynamic therapy, sensing and related applications. And he is publishing very well, more than 70 research articles, and completed more than 10 PhD students so far. And I should say that many of his initial students, like who are in CSIR NIST Thiruvananthapuram, are faculties in various colleges and universities in Kerala. And of course, Professor Ramakrishnan, who is organizing this, organizing secretary of this conference, also is a student of Professor Srinivasan. And he's a recipient of CRSI Bronze Medal and several other awards. And with this brief introduction, let me invite Professor A. Srinivasan to deliver his talk.
This is up, this is down. This is up, this is down, right? Let them set the slide. In the meantime, this is a great opportunity for me to thank the organizers, particularly the chair of the department,
Dr. Franklin John and Dr. Ramakrishnan, who has taken full effort to set this conference in a full mode. And yeah, this is the first visit to Ernakulam and first visit to the Tevara College. And of course, good to see you, good to see Professor Josie Joseph and Professor Akat Biju. And they were all, yeah, we were there in Institute Trivandrum, now we were there in different areas actually. Yes. And of course, Ramakrishnan was there as the first student of mine, and he was recommended by Mahesh Hariharan, who is currently a faculty member at ISET Trivandrum. And for me, this Tevara College is Sacred Heart College. Yes, it is, in principle, it is Bardwan University of Kerala. Yes, Bardwan University, you can find so many faculties across India. And now here also we can see there are so many people who have gone out, particularly Biju is here and Mahesh Hariharan. I have given only 2 examples. There are so many other faculties which I heard from Ramakrishnan yesterday, but most of them are from, very happy to hear from that. They are all from Bardwan University of Kerala, that is Sacred Heart College, Tevara. And I hope there are so many younger budding people are here, and then they will be definitely be the faculty member somewhere or the other inside India. With this, I will start my, yes, yeah. No, wait is point, okay. Okay. Yes, the title of my talk is this is Porphyrins with a polycyclic aromatic units. Mainly we worked in the area of porphyrin chemistry. And this is, I will take you through a very simple story so that you need not necessary to worry about what is going on exactly. It is a very, very simple story, and how we have come out with how we address the problem, and how we follow it up, and then how we will address the issues, and then finally find out the solution for the people. Let us come back to the ground state, because so far we were busy with Professor Yonath lecture, and we were all in the excited mode with the ribosome. Now come back to the ground state. Let us see what is going on in the ground reality. We can find it up and follow it up for that. Yes. These are the porphyrinoids which I am going to discuss you today, and particularly this is the porphyrin framework. One can see a 4-pyrrole unit connected with 4 methane carbon atoms, and if you remove one of the mesocarbon here, there are 4 mesocarbons here. Number 1, 2, 3 and 4. There are 4 mesocarbon atoms here and 4 pyrrole units are there. And if you remove one of the mesocarbon here, one can end up with the contracted porphyrinoids called a carole, and one can introduce, mimic the structure something like a carole, and one can also alternate the biphenyl framework, and one can also increase one of the mesocarbon here. If you remove one of the mesocarbon, you can end up carole, and you increase one of the mesocarbon, one can end up with the homoporphyrin, the simplest expanded porphyrin. Yeah, simplest contracted porphyrin and simplest homoporphyrin. And today we will address all these things. I am taking you all through a movie story. I am taking you all to 2007, what we have started in Institute Trivandrum, and this is what we started with, and this is not a porphyrin. This is a unit called calyx pyrrole, where instead of a mesocarbon, where conjugated mesocarbon, now it is a sp3 hybridized mesocarbon, and there is no conjugation within the molecule. Okay, what we thought of initially, where with my first student, Professor Ramakrishnan, we started introducing something like a ferrocene framework, so that this molecule is mainly used for This is mainly used for sensing property by using anions. So by mimicking the sensing property, whether we can introduce the electrochemical unit, something like the ferrocene, and find out whether that can usually bind with the anion or not. How easy to find it out. Okay. This is how we started the synthesis. We have started with a simple ferrocene, and then with the TMEDA we got the diol, and then with the diol we can get the pyrrole, connected pyrrole one can convert with, and finally one can get the macrocycle. So whatever the macrocycle which we have proposed, it is finally we got the macrocycle what we have discussed. And we also got the crystal structure, and happy to publish the paper in 2007. So now we have introduced the not a conjugated derivative, and we thought of introducing a conjugated one within the system, because this is a ferrocene unit. One can see there is the connection between this ferrocene unit and the rest of the framework. One can follow it up whether the conjugation is there within the connected system. Connected means there is the conjugation between the ferrocene unit and the part of the porphyrin framework, whether the conjugation one can establish or not. That is the idea. So the chemistry not ends there. We started with the 2007, and we have published the paper in OL, and one of the reviewer, one of the competitor who was closely working on this area of research, and he has nicely mentioned that the work was published from Ramakrishnan and Srinivasan, who has worked in this area. So he was generously has given the credit, but at the same time he has introduced the partial conjugated system by introducing a thiophene macrocycle within the system. Yeah, there is no conjugations. Here you can see there is no conjugation, but this framework one can see from pyrrole to another pyrrole, one can see a thorough conjugation within the system. And in the meantime, we have also synthesized the macrocycle, something like this, whatever the system which he has introduced, and we have come out with the shorter version of that. We have introduced the similar system, and we also got the corresponding expanded derivative. And now the final communication from this molecule, we thought of introducing a fully conjugated one. Yeah, fully conjugated one can come out with something like this. Yeah, conjugation between the ferrocene unit and the rest of the pyrrole framework, whether we can introduce the conjugation or not. We started something like that, and where the reaction did not go. To our surprise, the reaction did not work. Okay. And we also tried with the other synthetic methodology, whether a simple ferrocene and then take the dipyromethan, whatever I discussed in the previous slide, and do the reaction, whether one can end up with the final macrocycle, and still we could not be able to achieve. And to adding more salt to the wound, the guy who has followed up our work, and he has established the synthesis of ferrocene macrocycle, and then introduced the fully conjugated one, and then thorough conjugation between the ferrocene unit and the rest of the framework of the porphyrin framework. So what we could not be able to achieve, there is the guy who achieved the thing. And in adding more salt to the wound, he has not only established the ferrocene, and he has also come out with the rutinosine. So we could not be able to, what we could not be able to achieve, which he has done the things. Okay. Now the story not ends there. The story continues from 2015. Now I am starting with the 2015, what we have done. And this is 2015, the same group which he has started the similar kind of methodology, and wanted to introduce something like a phenanthrene unit in the macrocyclic framework, and then got the macrocycle something like this. One can see the fully conjugated system in the macrocycle. Now the same period which we were also working in the similar kind of the system. Let us see what kind of the system which we are working on a biphenyl unit. There there is a simple difference between these two. See here there is the additional unit is connected here. Because of the phenanthrene unit, there is the additional framework, it is there, and here that is not there. That is the only difference. But the structure is more or less similar, which he has proposed. And in addition, we also have the bipyridal unit. And we also have the other molecule, which is, once you reshuffle the biphenyl framework, one can end up with the meta derivative and as well as the ortho derivative here. So there are possibilities one can come out with. So 2013 onwards, I am busy with the administrative side. That is, if being a youngest institute, you have to run around pillar to post for all the activities. So, and I could not able to concentrate, but the student which he has synthesized everything, and he is trying to push me left and right, telling that, sir, which we have already synthesized and the guy already published the paper, and are we going to miss the bus again? The basic question he asked. So let us see whether we are going to miss the bus or we are in the right direction. Okay. So let us start with the biphenyl one, what we are talking about. This is, I will also move on to the bipyridal one, and if the time permits, I will look at the meta ortho-connected macrocycle. So this is how we started for the synthesis of this macrocycle. So fortunately, we don't have this kind of the system. We have only a system with porphyrin, and then there is the system with the benzene connected system. That is the only macrocycle which is there in the literature. And in addition, we have seen the previous slide, the phenanthrene framework. Now, what we have done, we have the bipyrrole unit here, and then we just replaced with the biphenyl unit. And this is how the synthesis goes on. We have started with the diformyl derivative with the Grignard reaction. One can end up with the diol, and one can end up with the dipyramidine, something like this. And do the reaction with the pentafluorobenzyl, one can get the macrocycle. What we have proposed here, that is reflected here. And we have also got the respective copper complex. Let us see this. This is the indication for whether we are in the right direction or not. Where the electronic absorption spectral analysis is clear and straightforward, we are in the right direction. Where the macrocycle, whatever we have synthesized, we are in the right direction. Let us see what and all we have done. We have analyzed the sample, and we got the proper clean NMR for the macrocycle as well as the copper complex. Now the copper complex, it is in principle, it is either the 2 plus or 3 plus. In principle, if it is showing a clear cut NMR, in principle it is a copper 3 plus or a copper 1 plus, any one of these two combinations. And it shows the clear cut NMR, it may be a copper 3 plus or copper 1 plus. And where we have the possibility that there are 3 anionic part, 1 CH, 1 CH, and 1 NH is there. So that are 3 hydrogen atoms are going out. Obviously, that will be a copper 3 plus instead of a copper 1 plus. And we have also got the crystal structure for the molecule as well as the complex what we have come out with, and where the macrocycle is more or less flat. Don't worry about what is going on exactly here. Whatever we have synthesized, finally we have achieved by crystal structure analysis. And the geometry around the metal center is a square planar. Let us see what we have come out with. The macrocycle, in principle, benzene is an aromatic unit. So far which we have come across, benzene is an aromatic unit. But once it mixes with the macrocycle, it loses its aromaticity. Though it has its own aromaticity, and when it mixes with the rest of the framework, it loses its aromaticity. Because the conjugation, this is more of a sp2 character here and more of a sp2 character here. And here there is the conjugation from here to here. You can see the thorough conjugation in this area. There will be a conjugation, and when it mixes with the biphenyl unit, the conjugation interrupts. So the aromatic macrocycle now turned into a non-aromatic one. And the similar trend was also reflected from copper 3 plus. And what else we have come out with? Yes, yeah, we have when we have started with the work, we have submitted all these things. Okay, we have done the simple copper by phenyl containing macrocycle, Carroll complex with Carroll macrocycle, which we have come out with, and we have introduced a copper complex. It is everything is a brand new molecule, so definitely it is worth publishing in a very good journal. Okay, when we send it to the reviewer, then the reviewer started asking question, whatever the complex which you have come out with, it is not a copper 3+. He told very clearly, you have to reconsider your things, whether it may be a copper 2+ or a copper 2+ cationic form or an anionic form. Any one of these two forms in principle. And he also suggested these are the papers which is already known, already published, and you have to recheck your copper 2+. Fortunately, he did not reject the manuscript. So he has put all sorts of objections, and then he asked us to revisit whether that is the copper 2+ or copper 3+. And for his evidence, which he has given also all the details which he has come out with, because there is the hydrogen here, that is the hydrogen here, when it goes to the low temperature or the high temperature, it becomes broad actually. And the similar trend, whatever the variable temperature NMR, once you check, we can see there is the variation in the variable temperature NMR analysis, and he has concluded that there is the copper cation somewhere with the one of the pyrrole unit. And the other guy, the last guy, whatever he has come out with, there is the mesocarbon, whatever it is there, that is while going down the temperature, it is disappear, and in principle it is a copper 2+ cation, not a copper 3+. So this is the regular trend for the copper simple Carroll complex for copper 3. Now the question is we have to address. Okay, he has given all his evidence, whatever he has come out with, whatever the reviewer asked, and now it is our position to address the issue, whether it is a copper 2+ or a copper 3+. That is the conclusion which we have come out with. And to try to come out with the copper 3+, which we have tried all the analysis. These are the molecule which is known. There are particularly a Carroll complex which is known, and there are a series of complexes which is already reported in the literature. And where the settling angle, what you can see, it is more of 15 degree from starting from 75 degree to, of course, one can go up to 15 degree. So there is the guy which is there at the mesocarbon, which is here, and in principle that is behaving like a carbocation, or it is behaving like a cationic form or anionic form, and behaving like a radical kind of thing. That is what the suggestions to identify the issue. And there are to support our evidence, there are two more porphyrin unit which our native group which they have published, and their report is which I have discussed. I have mentioned it here, and here one can see the bottom two are the result, whatever we have come out with, it is all matching with the already known complexes. All are copper 3+. These two are also a copper 3+, and whatever we have come out with a copper 3+, and the result is more of matches with the porphyrinic one. In principle, it is a copper 3+, and it is not a copper 2+, a cationic form or anionic form, or not either in the radical form. Okay, that is the first evidence which we have come out with. And in addition, we have done the variable temperature analysis, which I have shown the previous slide, one of the previous slide, which I showed very clearly that once you do the variable temperature, you can see the broadening of the signal, and we don't see any of the broadening of the signal by varying the solvent from toluene D8 or DMSO D6. In all the cases, the spectra is very clear. So there is nothing happening within the molecule, but in principle it is a copper 3+ and not a copper 2+, a radical form. Okay. Then the last evidence is from the EPR analysis, if at all, if it is, whatever the evidences which earlier the people which they have reported, they have shown only through the NMR analysis, but they have never reported anything with the EPR spectral analysis. And for our evidence, if it is a copper 2+ radical cation or a radical form, in principle one could see the signal, but we did not see any of those things. So in principle, it is in the toluene mode or in the solid state mode, the signal is very flat. So in principle, it is EPR inactive. The molecule is NMR active, and whatever we have come out with NMR analysis, EPR analysis, and based on the calculations where the people have come out with from the crystal analysis that matches with the copper 3 plus, not a copper 2 radical cation. So obviously the molecule, the macrocycle with the complex what we have proposed, it is a organo copper 3 complex, and finally which we have submitted, and the reviewer get accepted and finally puts the paper in the good journal. So in addition, where we were debating for copper 2 plus and copper 3 plus, and from our institute there is another information has come from it is not only from copper 2 radical cation, it is also possible to go up to copper 4 plus. There are evidences where the people have come out with. So we were debating for copper 3, and the possibility at the end of the day, after 2015, after our paper, there is the paper published about copper 4 plus. So there is the internal institute itself, there is the competition for this molecule. Okay, this is about the story of the first complex which I have come out with. And let us move on to the second one. Now I move on from biphenyl to bipyridyl. Let us see what we can come out with the bipyridyl. Whether we are following the same story what we have come out with in the case of the biphenyl, and now we have replaced with the bipyridyl. Let us see what bipyridyl one can do. So these are the macrocycles which is known in the literature. These are the macrocycles which is known in the literature. One can introduce a phenanthrene unit in the macrocyclic framework, like phenanthrene one can introduce, and where the pyridyl unit one can introduce, and one can introduce a simplest porphyrinite something like this, and one can introduce a Carroll derivative something like this. And what we have come out with, and instead of bipyrrole, and we have introduced a bipyridyl unit, where in the case of the previous case which we have introduced a biphenyl unit, and here we have introduced the bipyridyl unit. That is the only difference. Let us see how it goes. This is the bipyrrole is replaced by the biphenyl. And the synthetic methodology is more or less similar. Okay, there is no problem about the synthesis. Okay, we have taken the diformyl derivative and then corresponding diol derivative and the dipyrimethane and get the macrocycle, and also one can do the coordination chemistry what we have come across with the copper 3 plus in the previous section. And with this we are of course the electronic spectral analysis, we are in the right direction, and unlike the biphenyl case, and one could see a fluorescence spectra, excited emission state, a fluorescence spectra one could observe from the analysis. And we also done the NMR. NMR is very clear and paka, what we have discussed in the previous case. Here also the NMR is clear and pretty strong, and it confirms that the macrocycle and the final complex what we have proposed is clear. Let us see what else we have come out with. We got the crystal structure, absolutely no problem, like previous case. And the geometry is a square pyramidal in this case. And we also got the bipyridyl one, though it is aromatic and mixes with the system, it becomes a non-aromatic, like the biphenyl Carroll. Let us see, this is finally we got the crystal structure of one of the complex, the zinc complex, whatever I am talking about. We got the crystal structure something like this, and the distance. Let us see the distance between this. There is a distance between the system of pentafluorophenyl unit at the mesocarbon, and which is connected with the one of the phenyl unit of the other framework. It is almost like a hexagonal structure which is combined together actually. Okay, this is of a rough structure. Once you reshuffle the things, one one can end up with the macrocycle, one can end up with the complex structure, something like this. Yes, simple supramolecular assembly one can come out with. And one can donate during February 14 lovers day, one can donate such a kind of the studded one to their lovers. This is something like that which we have come out with. Let us see whether we can come out with something from this. Whatever the complex structure which we have proposed, whether we can come out with something from this. Here it is possible to find out the adjacent distance, alternative distance, opposite distance, and also one can find out the overall volume of the complex. So once I shift all these things to the right hand side, one can see the distance between the two zinc atoms. Two zinc ions, whatever we can see, either the adjacent one and alternative one or the opposite one can come out with. And also one can find out the overall volume of the crystal structure. It is 1563 Armstrong. So is that sufficient enough? Okay. Now the question is that sufficient enough for pushing a very good paper? In principle, that the standard what we have come out with, it is not as sufficient as far as we are looking forward. So what else which we can come across from this? This is, we started with the sensing experiment. We have started with the bipyridal derivative, do the reactions, a simple analysis with the perchlorate salt of zinc. You can do the bind with the zinc perchlorate, and one can do the binding experiment, whatever we can come out with. Here, the fluorescence intensity, it is gradually going up. Upon adding a zinc perchlorate salt, gradual increase of the salt, one can see the gradual increase in the fluorescence intensity. And here, it binds exclusively with the zinc 2+, not with any other metal ions. And there are so many metal ions we mixed with, finally it finds exclusively with zinc 2+. And also it shows the binding with the 1:1 binding mode with the macrocycle. And also we tried with the competitive experiments whether it binds with any other metal ions, and mixing with the other metal ions, and it binds exclusively only with the zinc 2+. And we also found out the binding constant and the detection limit, which is mentioned in the slide. And finally, we also got from the electronic spectral analysis, also we got the similar kind of observation what we have come across in the previous slide. And finally, we got the crystal structure of the complex, what we have proposed with the sensing experiment that we have come across. So let us conclude this part. This is the macrocycle where the people have come out with, and where with the simple phenyl unit, with the zinc 2+ salt, there is the gradual decrease in the fluorescence intensity, there is a gradual increase in the fluorescence intensity in this case, and they have kept the name as CHEF, Claisen-induced enhanced fluorescence, Claisen-based enhanced fluorescence, CEF. And what we thought of, it is CEF is a very unique name, because just like entering the way we call the chef and then order 2 dosa, 2 idli, so something, the chef is a very good name. So what we thought of changing the name, whether we change chef from something else. So what are the possible names which we can come out with? Okay, for changing the name, there is the name which the people which we have come out with. There is the AIE or AIEE. That is in principle, a pyrene unit wants it to overlay one among other, and finally initially it is a fluorescence emitting material, and once it becomes a solid state, it loses its emission intensity. And where they have introduced, to contradict the statement, the people have introduced some non-emissive material at a solution state and mixes the water molecule, and one can end up with the emissive material at the end of the day. So in solution it may not be a colorful material, but in the solid it is a colorful material. So they have introduced the name aggregation induced emission or aggregation induced enhanced emission. So this is to mimic similar kind of the area. This is the macrocycle what we have come out with, and we have also done the similar kind of experiment. We could see the emission intensity which is going up. So this is more or less same as that of the aggregation induced enhanced emission. By adding a gradual addition of the water molecule, one could see the gradual increase in the emission intensity. So to mimic that part, and what we have come across, which we have changed the name from CHEF to CIEE, Claisen induced enhanced emission. So once you add the metal salt, the emission intensity goes up. So we have tried to change the name, what they have proposed earlier, CHEF to CIEE to mimic AIEE. But so far it has not been thoroughly accepted by the people, but still they are able to follow only with the CHEF. But however, absolutely no issue. It is get accepted in a good journal. That is the wonderful result from this observation. Okay, so far I talk about two porphyrinites, and it's the simplest porphyrin which we have come across. And now I move on slightly from a simple porphyrinite to the expanded porphyrin. Okay, I am introducing a simple, in the porphyrin framework, I am introducing additional carbon atom so that you can get the expanded derivative. Let us see what we have come across. This is the porphyrinite called, I am moving on to the third story. Okay, this is the porphyrinite called homoporphyrins. Let us see, this is the expanded porphyrin, what I am talking about, called homoporphyrin, a smallest expanded porphyrins. What we have come out with, a simple porphyrin framework, and we are adding one more mesocarbon here. So everything is same. Whatever we are talking about, everything is same except this area where you cannot, there is the slight extension in the conjugation, adding one more carbon atom. If you remove one carbon atom, one can end up with the contracted derivative. If you add one more carbon atom, one can end up with the expanded derivative. This is the simplest expanded porphyrinite. Let us see what we have come out with. The first one is a 18 pi system. It follows the Huckel 4n plus 2 pi aromatic character. But you add one more carbon atom here, it loses its aromaticity. It is become a non-aromatic character or anti-aromatic character. From 18 pi to 20 pi, it is a non-aromatic or a anti-aromatic character. Let us see whether we are able to achieve the non-aromatic character or the anti-aromatic character at the end of the talk. So the system, the people started such kind of the work from 1960s. Whatever the system which they have come out with, there is no conjugation within the system like what I have started in the first part. There is no conjugation here, the conjugation interrupted here, and similarly there is the conjugation interrupted here. And of course they have introduced a conjugated molecule, what we have come across, what we have proposed at the top. Okay, we have, but whatever the molecule which they have introduced, which is quite unstable and not able to separatable. Okay, these molecules are unstable. Up to 1967 to almost 2015, the molecule is not possible to synthesize. Okay, let us see. This is the first expanded porphyrin, simplest expanded porphyrin. A competitor from my senior from IIT Bombay, which he has introduced the simplest expanded derivative. Fortunately he has not introduced the pyrrole unit. What he has come out with, he has introduced the furan derivative or the thiophene derivative in the macrocyclic framework. And for our favorable system, and both the molecules are non-aromatic. Okay, it is not a anti-aromatic or aromatic molecule. It is the macrocycle at the end of the day. It is a 20 pi system and it is a non-aromatic macrocycle. Let us see whether we are able to achieve the pyrrole-based macrocycle, whether we can come out with or not. Okay, we are starting with the simple synthesis for this macrocycle. We have taken a diol framework and then a dipyroethane framework, not a dipyromethane, where in the starting itself we have introduced the ethane bridge here, and do the reaction with the paratoluene sulfonic acid, one can end up with the macrocycle something like this, and also one can do the coordination chemistry with this. I am reiterating again here the coordination chemistry, particularly with the rhodium salt. In principle, it binds only with these two units, and not with this system, or not with the system something like this. But in principle, here it is coordinating between the dipyroethane unit framework and then dipyromethane methane unit framework. It is neither coordinating this side, not coordinating the other side, but it is coordinating in between. Okay? So in principle, when you do the analysis, we always think that it always coordinates with the bottom mode, not with the top mode or not with the side mode. That is the conclusion where the reports were known. Okay. So how it is connected with the side, I will come back to you. This is the electronic spectral analysis. The spectral analysis is clear and straightforward. So the macrocycle is clear and straightforward. And we have also done the variable temperature analysis, and where one could see the NH signal, it is this NH signal, it is initially at around 6.2 ppm, it is going down to 5.5 ppm. Usually if it is a non-aromatic molecule, it is more than 10 ppm, and here we are observing in between. If it is a fully aromatic, usually at around -2 ppm. So it is neither aromatic nor non-aromatic or anti-aromatic. Usually it is at the 10 ppm, that is not appearing here. Okay. Then we have done the respective rhodium complex, and the rhodium complex also it shows the clear-cut variable temperature analysis, which is going from the upfield region, going from the downfield to the NH signal, whatever we are observing, that is going to the upfield region. So in principle, it is neither a non-aromatic or anti-aromatic or a aromatic molecule. So what else is this? Okay. So this is we are successful enough to get the crystal structure of both the things, and finally we are able to get the Mobius aromatic character of this macrocycle, and finally it is the Mobius aromaticity shows -6.8, and in this case it is -6.9. The molecule is Mobius aromatic. It should be in the negative point, and we could be able to see if the value is close to 0, the macrocycle is aromatic. Mobius non-aromatic. The value is positive, and the macrocycle will be a Mobius anti-aromatic or anti-aromatic. If the value is negative, the macrocycle is aromatic, Mobius aromatic. So it is different from what we have come across so far. It is either aromatic, the Huckel aromaticity now overruled here. A Huckel aromaticity overruled here, and one can end up with the different kind of aromaticity called a Mobius aromatic system. So this is the where the previous cases the people introduced whatever they have come out with, it is a non-aromatic macrocycle, and here we have come out with the anti-aromatic system. And yes, and I will also highlight why this is on the why it is neither in the bottom side nor at the top side. We have done the theoretical calculation for all these molecules, for all these metal complexes, and finally energy minimized before the crystal structure. We have done all the theory, and finally the theoreticians clearly predict it is neither connected with the bottom or not connected with the top, it is connected with in between. So theoretician has given the clear conclusion. And the final crystal structure also support what the theoretician. In principle, earlier we used to get the result and then send it to the theoreticians and then ask them to get the opinion, or able to whatever we are able to come out with, whether you can match with the result or not. But this is the other way around. What the theoreticians predicted, and finally it is reflected from the experimental structure. Getting the theory support, it is not that easy. For this molecule, it's supported thoroughly. And of course, that is also, finally it is through. Let us see, for another 2-3 minutes, I would like to spend only on the system, where the sumaragdenium based system, where I have talked about a phenyl and then predyl separately. Phenyl system separately, predyl system separately. And here I am introducing the phenyl and predyl together. And we have done the coordination chemistry with the platinum salt. And of course, this is for the routine analysis what we have come out with. And we have also got the respective coordination chemistry with the platinum 2 as well as the rhodium 1. And the rhodium 1 is straightforward, what I have discussed earlier. Usually it is on the bottom side. Here it is clear. So this is what I am trying to convey in the previous section. It is at the bottom part, it is coordinates. And on the platinum side, which is now there is one of the pyrrole, it's not connected, and one of the phenyl unit is also not connected. And here one could see the clear-cut format. One can come out with the system something like this. It is a typical organometallic platinum 2 complex, and also it is a typical unsymmetrical pincer type complex. A pincer type complex in the macrocyclic core. And this is how which we have come out with, however, which we could not able to do the catalysis so far, which I am not expertise in this area, catalysis particularly. Okay, let us move on. Finally, this is the overall summary what I have come across. And I have discussed the biphenyl carol, bipredyl carol, and then the phenyl predyl carol, and of course these two are expanded, these three are all expanded derivative which I could not able to cover, and of course there is the article published here. Finally, I would like to take this opportunity to thank my students, and mostly this is done by Adi Narayana, and this is the photo taken from NIST Trivandrum. Just opposite outside the auditorium, which is the existing auditorium. At the front side, they usually write RRL NIST Trivandrum, that is there in the front, which I have deleted the things actually. Watching. Then this is Professor Ramakrishnan. He was in 2009. Okay, and we have Dr. Anju here, Dr. Derry here, Ajit Thomas is here, and then we have Gauri here, Salini here, and of course, as rightly pointed out by Josie Joseph, most of them are faculty member at various colleges in Trivandrum. Sorry, various colleges in Kerala. And of course, we have Dr. Adi Narayana, which he has involved for this biphenyl and piperidal work, and Mainak Das, and then Anju, mostly of a homoporphyrin work which they have come out with. And we have some of the crystal structures solved by our scientific officer, and of course strong support from theoreticians, whoever, whatever I am talking about, it is fully supported from NIST Trivandrum. CS Suresh from NIST Trivandrum, and I also take this opportunity to thank the funding agency. NICER, it is generously supported by the DIE, and also thank ACRB DST for the funding. Thank you very much.
Thank you very much for that.
Happy to answer.
For that very nice story. And the seminar is open for discussion now. I encourage students to ask questions.
Sir, very nice work.
Yeah, thank you.
I have a doubt, like you had done zinc metallation, right?
Yes.
You had used zinc perchlorate.
Yes.
Is there any specific reason why you went for that instead of zinc acetate or zinc chloride?
Yes, by zinc acetate also we have the crystal structure which I have shown. Okay. For the analysis purpose, usually they use the perchlorate salt only For perchlorate salt, it is available for across the first row transition metals.
Okay.
So we have to compare with the perchlorate salt for all the transition metals. From there you can decide.
Okay.
Yes.
So the free base was emissive or non-emissive?
Free basis was mildly emissive. It is not that
Mildly.
emissive.
So only upon adding zinc
Only
it was-
adding zinc, it is going up.
Oh,
Yes.
Sir, and one more doubt.
Ha.
Like you had shown the Nix value, right?
Yes.
One in without bracket and one inside the bracket you have
Yes,
put,
yes.
I didn't get it. Like, is it?
One could see the center of the macrocycle. What is the Nix value?
Yes.
That is what I have projected. That is minus 6.1 something like that, which I have mentioned. And there are some values of the individual units also I highlighted. So that is for the individual pyrrole, and then there are 4 pyrrole units which are there. That values also I have mentioned it in the value.
Okay,
so the center one is the final decision.
Sir, inside the bracket and outside it you had mentioned. So is there any difference for that? Like for even the center of the macrocycle and the pyrrole unit?
That is the deviation from the plan actually. It is 6.96 bracket 0 something like
yes,
that.
deviation.
Yes. How much it is deviated from the value.
Thank you,
Yes.
sir.
Yes.
So it was nice and informative talk. So
Yes.
in your talk, first part you have discussed about the copper-based porphyrin structure.
Yes.
There actually that when molecule is going to that copper 2 to copper 3 to copper 4, there is any optical changes there, like color change?
Copper 3, because this is biphenyl work actually. Biphenyl work, if you are having a simple carol with the pyrrole unit, then you can compare copper 2, copper 3, copper 4, one can compare. And here the system is not like a pyrrole based. Not all the 4 pyrroles are not a pyrrole based. There are 2 of them are a biphenyl and 2 of them are a pyrrole. So we cannot compare this one with the simple carol. In principle, yes. A simple carol one could be able to possibly follow it up 2 plus, 3 plus and 4 plus if at all if it is existing. That is what the 4 plus paper is highlighted. Yes.
Thank you, sir.
Yes.
Thank you, sir, for the
Yeah.
nice talk.
Yes.
My doubt is based on the synthesis of macrocycle. You are
Yes.
synthesizing macrocycle by 2 bifunctional units.
Yes.
So is there any possibility of formation of polymers or 2 plus 2 or 3 plus 3
Yes,
macrocycle?
in principle, yes. Polymer is possible, but whatever the yield which I have shown, it is less than 10%. The basic question always the people ask question.
Yes.
Okay, why you are taking such a amount of the material and finally you will end up with only 10%. Getting the 10% is on the, as a porphyrin chemist, one will always be happy. But as an organic chemist, they will not accept. Okay, so they will always, they will be very happy with the energy excess is increasing from 65 to 66 or 65 to 70. They will be very happy. But they never talk about this synthesis part actually. Particularly this mostly of become a polymeric material at the end of the day. Yes, whatever the pyrrole we are starting with, this becomes a polymer.
Okay, sir.
Yes.
Thank you.
Any other questions from the students?
Good to hear from you. Yes.
Any questions?
Thank you for
Yes.
your talk.
Yes, thank you.
And I would like to know some curious about the application of this
Yes.
type of compounds.
Yes.
And I would like to hear about if you have one idea, if this type of compounds is
Yes.
possible to use it in some solid polymers like membrane.
Yes,
and to remove some
Yes,
heavy
yes,
metals
yes.
in
Yes,
water
yes.
treatment.
Yes.
So I would like to hear about it, something, your opinion. Okay, thank you.
Yes, yes. Such a work which we have not done. In principle, I can clearly I give my opinion so far. See, I'm more of interested on the synthesis of the macrocycle and find simple applications whether that can be useful. Like that is the only thing. But beyond the capacity which I have to rely on the collaborators whether they can come out with. Particularly the pincer type complex which I have come out with, and in our institute itself there are so many organic chemists, and most of them are working in catalysis.
Okay.
And still I could not able to approach any one of the persons to execute the work actually. So that is the one which we are looking forward, and particularly your membrane part, whatever you are talking about so far, which we have not explored. Yes, we have to look for a right collaborator so that at least we can establish such a things. Yes.
Thank you very much.
As far as I am concerned, I am not an expertise in that particular area.
Thank you very much. And I have one question more.
Yes.
Okay, please. What about the toxicity? And do you hear about this possibility and about the toxicity? Do you have some word about it? No.
No, because the porphyrin based, there is one macrocycle which is mainly used for a cancer therapy. Protoporphyrin, there is a macrocycle which is still the people use it for the PDT applications.
Okay, thank you.
And in principle, I don't think it is a toxic material. Yes.
Thank you very much.
Yes. Photophrin.
So this most of your porphyrinoids have this pentafluorophenyl substituents. Is there any particular reason for introducing that?
Yeah. For a NMR analysis, pentafluoro is better because you don't see any signal. Yes, yeah. For 1H NMR, you don't see. Yes, yeah. That will make it, because it is a huge molecule, and then there are phenyl units, and then you have the pyrrole units. All are falling in the same region. So in order to avoid such a things, we have chosen the pentafluoro. And the next one is, and also it gives the supramolecular assembly kind of thing. Yes, there is no secret with the pentafluoro. Yes, make the story simple, which we have come out with the pentafluoro.
Will it help crystallization or?
Yes, absolutely no problem. Yes, for NMR analysis, of course, the crystal analysis, this pentafluoro is very useful.
Okay.
Yes, and also we can get expanded macrocycles. Yes.
Okay, if there are no more questions, please join me in thanking Professor Srinivasan for this wonderful talk.
Thank you.
That was an enlightening session, and I thank Professor Dr. A. Srinivasan for the lecture, and I convey my sincere gratitude to Dr. Joshi Joseph for chairing the session. Now, I request Dr. Joshi Joseph to kindly hand over the memento to Dr. A. Srinivasan as a token of love, and I welcome Professor to kindly receive the same. Next, I call upon Dr. Grace Thomas to present the memento as a token of love to Dr. Joshi Joseph. There is an announcement. During the lunch break, all the oral presenters are requested to hand over their presentations in pen drive to the technical team sitting in the left of the hall near the podium to Dr. ABTG and Dr. Jason M. Che. Those who are participating in poster presentation competition can hang their posters in the premises of Department of Chemistry in the first floor according to your convenience before 5 p.m. The evaluators will be there at 5 o'clock in the evening. For any queries, approach volunteers. So with this, there is an end to our morning sessions. Now I request all the delegates and participants to gather at Marion Hall for the flavorsome lunch. Volunteers will lead you to the venue. All the participants are kindly requested to take their lunch coupon along with you. All the participants and delegates are requested to gather at Malicious Hall at sharp 1:30 p.m. so that we can continue our sessions without delay. Thank you.
having a sumptuous lunch. Hope all of you enjoyed it. I'm extremely overwhelmed to welcome you all back to the second plenary session of 21st Professor K.V. Thomas Endowment Seminar and 4th International Symposium on New Trends in Applied. The talk will be led by Professor Dr. Tamoki Nobuyuki on the topic Building a Smart Molecular Robot by Combining a Motor Protein and a Molecular Photoswitch. I am profusely elated to take this opportunity to introduce our chair of this session, Dr. Aravind Kumar CT. He doesn't require an introduction as he is a well-recognized figure. Professor Aravind Kumar is the Pro-Vice Chancellor of Mahatma Gandhi University, Kottayam, and Professor of School of Environmental Sciences. Some of his major research areas include environmental chemistry, photochemistry, and polar research. He is the Honorary Director of Inter-University Instrumentation Center and the Coordinator of Sophisticated Analytical Instrument Facility. Dr. Aravind Kumar was awarded Erasmus 2019 Visiting Fellowship from European Union. He is the Chairman of Committee on Antarctic Environmental Impact Assessment and Antarctic Environmental Governance 2022. We are grateful to him for accepting our invitation and becoming the chair of this event. With immense pleasure, I invite Dr. Aravind Kumar CT to chair this session. Welcome, sir.
Yeah, thank you very much. And I am thankful to the organizers, Dr. Franklin and others for the invitation. I'm really delighted to introduce Professor Tamoki from Hokkaido University. Now a word about Hokkaido University. It is a university where they have maximum interaction with Indian universities. In fact, Hokkaido University has several collaborations with many universities, including ours. And even Government of India has an MOU with this Hokkaido University for sending people to the transportation department, where India is now developing the technology, the bullet train technology, and developing that kind of facilities. So some of the training is being given by Hokkaido University. So it's a very important university for India, and there are almost about 16 professors, Indian professors are working in the university. So Professor Tamoki obtained his PhD from the Graduate School of Science and Engineering, Shiba University, Japan, under the guidance of Professor Yamako for his work on photochromism of macrocyclic azobenzene derivatives. After completing his doctoral studies, he joined the Research Institute for Polymers and Textiles, now known as National Institute of Advanced Industrial Science and Technology, Tsukuba, Japan, as a research scientist. During 2001 to 2008, he served the institute as the group leader. During the same time, he was also a visiting professor at Yamagata University and University of Tokyo, Japan. Later in 2008, he moved to Hokkaido University as a full professor at Research Institute for Electronic Science. For 2 years, from 2017 to 2019, he was the director of Green Nanotechnology Center of Hokkaido University. His research mainly focuses on synthesizing photochromic macrocycles and photoresponsive molecular organizations and polymers. He is also interested in the synthesis of a new photoresponsive compound that activates the molecular function of motor proteins instead of ATP. He has published more than 100 articles in highly reputed international journals. With this very brief introduction, I cordially invite Professor Tamoki for his lecture. Please.
Okay, so I switched on. So may I use that microphone? Yes. Oh, okay. Thank you very much for your kind introduction. So, and I also want to thank the Dr. Ramakrishnan and the other organizing committee member for inviting me to this nice symposium. So today I want to talk about molecular photo switches. So I also show the usage of the molecular photo switches, so actualizing a kind of smartness in the non-living molecular systems. And finally, I want to discuss the possibility of photo switches to the side effect-free cancer therapy. So for a higher form of the life, including the human being and sharks, so information obtained by eyes are quite important. So this woman, without seeing anything, so continue swimming peacefully. But so after finding a swimming woman, this man-eating shark will try attack her in the next moment. So we get most of the information through eyes and process the information by brain and take next actions. But not only the higher forms of the life, but even the euglena, which is a typical unicellular form of life, can act next action depending on the provided optical information from surroundings. So as seen here, euglena can move by using the function of flagella. But the more important fact is that euglena can sense the difference in the wavelengths of the light and reflect to the next action. So initially homogeneously dispersed euglena here escaped from the blue light from the left-hand side or gathered to the green light by time. So this is proving that neither the brains nor nerves are necessary for accomplishing such smartness. So analyzing the optical information from surroundings. So my question is, what is the minimum requisites for accomplishing the smartness? And is it possible to make a molecular system without life smart enough to gather and escape from light by judging the wavelengths? So in our body, there are several moving molecules, motor proteins, so working for supporting the muscle or supporting cell division or movement of the small organisms. So there are the myosin, kinesin, and dynein. And I want to focus on a kinesin microtube system supporting the transportation of the cellular cargo in the cell or cell division. So kinesin work on a microtubule track, which is a tube-like aggregate of the round protein called tubulin. And this is the animation showing how kinesin work on a microtubule track. So this is kinesin, and this is the head part of kinesin. And at the tail part here, so cellular cargo is attached and delivered. Okay. Like other motor proteins, kinesin is ATPase, the enzyme of hydrolysis of ATP. And ATP is adenosine triphosphate. And by the reaction, so by the energy obtained by ATP hydrolysis, or the structural change obtained by the reaction, so mutual position between the kinesin motor protein and the microtubule is changed. And by repeating this reaction, so processive movement of kinesin against the microtubule is attained. So such motor function of kinesin microtubule can be visualized with the fluorescent optical microscope. So in this in vitro motility assay, we fix the kinesin taken out from the cell is physically fixed on a glass substrate surface, and the movement of microtubule, fluorescently labeled microtubule, driven by the kinesin head and the energy of ATP, can be visualized on a fluorescent optical microscope like this. So this yellow line is each microtubule. So having the diameter about 25 nanometer, and the average length of the microtubule here is about 20 micrometer. But unfortunately, so the movement of this microtubule is totally random, right? So you cannot see any smartness in this movement. So I thought, in order to make the motor proteins smarter, so it is important to add some switching function. So there are several methods to regulate the motor function of kinesin. So one method using the caged ATP, so blocked ATP. So one time off to on switching of the kinesin's activity is possible. And by using the caged inhibitor, blocked inhibitor, so one time on state to off state of the kinesin's movement is possible. So these two methods, they irradiate UV light to deblock the protecting group. So real ATP is released, real inhibitor is released by the function of UV. But unfortunately, this is just one-way reaction. And by using the thermal-responsive or electro-responsive polymer, reversible switching is possible, but in sufficient manner. So it means that by using the ever-known method, so the switching is non-reversible or incomplete. So I want to develop the reversible switching. So I try to use photoisomerization or reaction of azobenzene. Azobenzene is very typical photoisomerizing molecule, which changes the structure between trans isomer and cis isomer by the action of UV and the visible blue light. So not only the structure, so long extended trans structure and bent structure, but so property is also changed. So hydrophobic nature in trans and hydrophilic nature in cis. So my first attempt to make the switching is to introduce the azobenzene photoisomerization unit into the high energy molecule. So original high energy molecule for kinesin microtubule is ATP, right? So ATP is composed of 3 units, adenine part and ribose part, triphosphate part. So in the first high responsible high energy molecule, I introduced the azobenzene to the ribose unit at the 2' position. And in the second photoresponsive high energy molecule has no adenine or ribose. It means that this is totally non-nucleoside triphosphate. So the synthesis of the azobenzene appended ATP can be synthesized by this synthetic route, starting with adenosine. And the synthesis of non-nucleoside triphosphate, AZO-TP, is much simpler, and by this route, starting with the 2-amino azobenzene, so we could synthesize this triphosphate. So first, I'll show you the effect of this azobenzene tethered ATP. So this also works as a high energy molecule for the kinesin microsystem. Without ATP, this can drive the grinding motility of microtubule driven by kinesin fixed on the glass substrate. And after UV irradiation, we get the cis-rich state, this cis-rich state, and the velocity is increased. And by UV visible light irradiation, which revert the structure to the trans, and so velocity is reversibly changed. So almost 30% velocity is changed by UV light and visible light irradiation. But so 30% change is not sufficient. So to enhance the change of the velocity by light, so I totally changed the I redesigned the high energy molecule from the start. And finally, I found that this non-nucleoside triphosphate can support the movement of kinesin microtubule. Okay. And this is showing movement of the microtubule, not using the ATP, but AZO-TP. And not only the kinesin microtubule, so we can drive the myosin-actin system by this AZO-TP. So in this experiment, we fixed the myosin on the glass substrate, and the fluorescently labeled actin fiber is seen, observed on a fluorescent optical microscope. So this is the first non-nucleoside triphosphate, which drives the kinesin microtubule system. So next, we studied the effect of photoisomerization on the activity of AZO-TP, non-nucleoside triphosphate. So initially, it's in a trans state, and after UV light irradiation, it changed to cis, and after visible light state irradiation, so cis go back to trans. Okay, initially moving, right? Now I irradiated with UV light. Substantial decrease of the velocity is observed, and after visible light, go back to the initial velocity. So velocity can be switched just by irradiating with UV and visible for just 2 seconds. And this change can be repeated many times. And initial decrease of the velocity is about 80%. So quite substantial change is driven by the photoisomerization of AZO-TP. To know the contribution of cis isomer to the motility, so we studied a little bit in detail, so concentration dependence on the grinding velocity at the non-irradiated state, 100% trans, and photostationary state under UV light, which gives the 8% of remained trans and 92% cis. So by using this data is analyzed with Michaelis-Menten equation, and by using this case value, we got the theoretical line for the 8% of trans of this concentration, so as a black line. And the blue one is experimental result with 92% cis and 8% trans. And this experimental data almost match to the theoretical line calculated for just 8% trans. This means that cis has almost no activity in comparison with trans azobenzene. So we can explain this substantial change of the activity from trans and cis by investigating the structure of the kinesin containing the ADP this time in the X-ray crystal structure analysis. So this part is adenine. So ATP is easily hydrolyzed and the ADP is remained and makes the complex. And ADP's adenine part is sandwiched between some amino acid residue of the kinesin. So from the bottom, histidine. And from the top, so proline and arginine is interacting by pi stacking interaction and hydrophobic interaction, respectively. So we can reasonably explain that in that pocket, trans-azobenzene can be easily substituted for the adenine part because trans-azobenzene has a flat pi conjugated and hydrophobic nature like adenine, but cis-azobenzene cannot because cis-azobenzene has a round and rather polar hydrophilic nature. So that is the reason why trans-azo-TP is working for the high energy molecule efficiently, but not in the cis form. So in addition, it is well known that in the kinesin pocket, so ribose part is not so specially recognized. So that is the reason why so ATP azo, this molecule, shows just the moderate change in the activity by the trans-cis photoisomerization. So we could develop the first non-nucleoside triphosphate, which drives the kinesin microtubule motor system. And substantial change of the velocity is driven just by irradiating with UV and visible light. So we could regulate the motor function of kinesin microtubule by using the photoresponsive high energy molecule, but with incomplete on-off manner. So, so perfect switching between the on state and the complete off state is desired to actualize the smartness in the molecular system. So for that purpose, I try to design the photoresponsive inhibitor, not the energy molecule. So it is known that the C-terminus domain of kinesin has inhibition effect. So interact at the motor part to block the motor function at the motor domain when the cargo is not bound to the tail part. So in our experiment, we usually use a recombinant kinesin to exclude this inhibition effect. So in our photoresponsive inhibitor design, we try to link the peptide taken from the C-terminus of the kinesin and photoresponsive azobenzene. And I added this inhibitor as additive. This shows the concentration velocity relationship. So without azobenzene unit, so just a peptide shows the inhibition property, but no photoresponsibility. So black shows a non-irradiated state, and red is UV irradiated state. So these are no change. But by adding the azobenzene unit as a terminal, so some photoresponsibility is seen. So trans state without UV irradiation shows a much stronger inhibition effect than the UV irradiated state containing cis-rich. And then to our surprise, so reversing the amino acid sequence to this and shortening the amino acid residue enhances the change of the activity, inhibition activity. So at 2 millimolar concentration, we could completely switch from complete off state and on state reversibly. So this is showing the movie of the motility. So of course, without the inhibitor, no light effect is seen. So by irradiating with UV and visible light, so velocity is constant. So but with the photoswitchable inhibitor, azobenzene peptide, so initially stopped, completely stopped. And after UV irradiation start to move, and visible light irradiation completely stopped. So this complete on and off switching of the velocity is actualized using the photoswitchable inhibitor, azobenzene peptide. So this is showing the change of the velocity at the vertical axis. So before irradiation or after visible irradiation, no movement is seen. And after UV light irradiation containing cis, the velocity is seen. So complete off and on state is repeated many times by just irradiating with UV and visible light for 1 second each. So finally, I want to show you the smartness of this molecular system. So in this experiment, I irradiated with UV light from the top, whole area irradiated with UV light. And from the bottom, so I partially irradiated with blue light. So with so circle area, in the circle area having the 15 micrometer. Okay. So initially, just UV light irradiated. And at this part, blue light is irradiated. And as you see, so the concentration of microtubule is enriched, gradually increased. And I remove the blue light here, and the position is shifted here. And now, so concentration of microtubule here is increased. So microtubule escape from the blue UV area to blue area, so reversibly. Okay, I think this is showing the a kind of phototactic property or a kind of smartness. So with the non-living molecular system, but this is almost identical to that seen in the living euglena. So as a conclusion, it is possible for non-living molecular system to exhibit smartness, to feel the light stimulus from the outside, process the information, and make its own judgment and takes action. So the key is photo switch. So I showed you that by using the photo switches, we can almost freely regulate the motor function of the motor proteins. So from now, so I want to show you an example of the application of the photo switch into the cell, aiming at side effect-free cancer treatment. So these images are showing a cycle of mitotic cell division, which is one of the most important event in the bioprocess, and it is frequently seen in the cancer cells, right? And so during the cell division, so chromosome, here chromosome are specially visualized. Chromosome move from the surroundings to the this is called the metaphase plate at the equator. So at the equator, aligned and separated and become two cells, right? And the chromosome dynamically move, drastically move in the cell division process. So it is important. So cell division is frequently occurring in cancer cells. So selective inhibition of cell division is quite important way for the side effect-free cancer treatment. So in the cell division, there are several proteins working, and especially kinesin 7, which is also called as a centromere-associated protein E, or shortly CEMP-E. Is delivering the chromosome in a cell from the pole side, pole part, to the metaphase plate at the equator. So that is the role of CEMP-E. And this CEMP-E's function can be confirmed by observing the chromosome congregation. If the CEMP-E works correctly, so the right chromosome alignment or congregation at the metaphase plate is observed. And if the CEMP-E's function is stopped, so the misalignment of the chromosome or miscongregation of the chromosome is observed like this. In this image is here, so some chromosomes still remain at the pole side. And if this state continues, so cells undergo apoptosis and eventually die. So this is one of the non-photoresponsive inhibitor for CEMP-E. And this was known. And we try to substitute, change one of the recognition site of the non-photoresponsive CEMP-E inhibitor to the photoresponsive unit, pyrazole-azole-benzene. So this is one of the important photoswitch. And we can synthesize this as a photoswitchable CEMP-E inhibitor. So this is the synthetic route of the photoresponsive CEMP-E inhibitor 1. And we could synthesize this with the 6 steps route from the Boc-protected nitrophenylalanine. And this, we studied the photo and thermal isomerization property of this molecule with UV visible and proton NMR spectroscopy. And this shows a very nice photoisomerizability, but under the UV light, we get the cis-rich state containing 93% of cis, and under blue light at 510 nanometer, we got 86% trans-rich state. And this shows the thermal isomerization from relatively unstable cis to trans in the dark, and the lifetime of cis in the dark is 77 hours, so reasonably stable. And we see the almost fatigue-free repeated isomerization is seen. So first, so the inhibition property of the synthesized photoresponsive inhibitor for CEMP-E is studied with the ATPase assay. This figure shows the concentration and ATPase activity relationship. Without the light, black line is obtained. After UV, red line, and after visible light, blue line was obtained. And the IC50 value of the inhibition, which is the concentration showing the half of the ATPase activity, is largely changed depending on the irradiation condition. So before irradiation, 5.9 micrometer, and after UV irradiation, it changed to 120 micrometer. So at certain concentration, we can switch the ATPase activity by changing the wavelength of the light. So this movie shows the movement of the chromosome after adding our photoresponsive inhibitor to the cell. So initially, some more chromosomes are misaligned, not aligned well, and so now irradiated with UV. So chromosome tried to align to the center, and I stopped with the visible light to convert the photoswitch to trans state. Now it's in inhibition state. So no misaligned chromosomes are still here. And from here, I irradiate with UV light. So, okay, now I irradiate with UV light. Chromosome come close to the equator plate. Okay, so in this, like this manner, we could regulate the movement of the chromosome, so reversibly. So finally, so I show you the change of the fate of the cell. So if the chromosome alignment is stopped, so cell goes to the apoptosis and die. So we check the cell viability depending on the irradiation condition. So black line and blue line shows no irradiation or visible light irradiation. And the red line is after the UV light irradiation with the cis. So if we irradiate at the 1 micromolar concentration here, so most of the cell died with blue light, visible light irradiation. And most cell remain alive with UV light irradiation. So we can change the fate of the cell depending by choosing the wavelength of the light. So for some purpose, lit active inhibitor is preferred to the dark active one. So our original photoresponsive SEMP inhibitor is the dark active one. Dark active means that in the dark, as an initial state, it shows the inhibition effect. But after the irradiation, inhibition effect is diminished. And lit active inhibitor is initially non-inhibitor. So after the irradiation, it works as an inhibitor. So we could switch the dark active inhibitor to the lit active inhibitor by changing the substituent position of the parazolyl azo group from para to meta. And we designed this molecule as a lit active inhibitor and synthesized. And we confirmed the photoisomerization from trans to cis and cis to trans with UV and visible light irradiation or thermally. So this is the result with the cell or so ATPS assay. So here, black and green line shows no irradiation or visible light irradiation, showing the almost no inhibition. But after UV light irradiation, serious inhibition is seen at the 10 to 100 micromolar concentration region. So we can switch the ATPS activity by the in the presence of compound 2. And only the misalignment of the chromosome is seen with the UV light irradiation. So without irradiation or after visible light irradiation, no misalignment is seen. So the cell chromosome can be switched with the lit active inhibitor 2. Okay, so finally, I'd like to show you the possible way to use this photoresponsive inhibitor for SEMP-E for the selective anti-cancer drug. So we can directly inject the SEMP-E inhibitor, photoresponsive SEMP-E inhibitor, to the whole of the body, and we can irradiate selectively at the diseased part, like cancer, with the active light to convert the drug from inactive state to active state and kill the cancer cells. And by time, the activated drug can migrate to the healthy body, healthy cells, but at that time, so thermal isomerization or with photochemical isomerization with other wavelengths, so change to the state from active to inactive. So then, even after the migration, healthy cells can be healthy. So we can, by using this method, we can do the side effect-free cancer treatment. Okay, I want to finish my talk by introducing our main contributors to this work. So these are my former student, PhD student at my Hokkaido University. So Nishad Peru synthesized ATP and studied the photofunctional property of its kinesin 1. Now he is working at Toyo Inc. in Japan as a researcher. Sunil Kumar synthesized azopeptide and studied its inhibitory function with kinesin 1. And now he is working at a private herb company in India as a senior research scientist after doing the assistant professor at Indian College. So Amruta optimized the structure of azopeptide and studied its inhibitory function with kinesin 1. Now she is working at my laboratory at Hokkaido University as an assistant professor. And Mafi, she is in Bangladesh, so synthesized a photo-switchable SEMP inhibitor. Now she is working at the University of Texas, Austin as a postdoc research fellow. So thank you for your kind attention.
Professor, at the end you are giving a proposal. You activate that using light, and with body temperature it may come back to the inactive state. So in the actual system you worked on, so to what temperature it is coming back to the other state?
So summer isomerization.
Yeah.
So I showed you that so around 70 hours is needed to get to convert the half concentration of the Cs.
Okay,
but it is quite dependent on the structure. So I just show you the typical one.
Yeah.
So by, for example, introducing the amino group on the phenyl ring of the inhibitor, it is the lifetime of the Cs is shortened. So we can freely tune the lifetime of the Cs from several tens hours to the less than millisecond.
Okay. Thank you. It's informative.
Yeah. So I have already synthesized the amino one, and it shows less than second.
For the real validation of these experiments, I think in vitro studies are required. Which type of living cells you conduct the experiments to?
So it's a cancer cell, but so I forgot the exact cell lines.
Okay, cell lines from which organs?
Yeah, human.
Human.
Yeah,
right.
that is all the human.
Okay.
Yeah, I want to know, after your experiment, did you notice any side effect from the hemo system? You did your experiment on hemo. Did you notice any side effect? Negative side effect?
Side effect.
Yeah, negative side effect.
So you mean that adding our chemicals or irradiation? So I don't see any side effect, but if we irradiate long time with UV light, so it we see the death of the cell. But we usually irradiate just 1 second or several seconds for converting almost whole of the molecule from trans to cis. So no need to irradiate so long time. So not so visible side effect is seen. But so actually, and also the dose effect. So if we dose so high concentration, so maybe cell died. So we need to use minimum amount to the cell to avoid the side effect. Okay?
No more questions. See, how do you compare this with photodynamic therapy? Because that is a...
So I hear that photodynamic therapy is also so toxic, so singlet oxygen or something is formed by the energy transfer from the dye, right? So, but so dye molecule remain. So we cannot go out from the dark part to from dark part to the normal light room. So to avoid the side effect, so we need to remain in the dark for 10 days, 10 hours or 20 hours or something like that. But with our wavelength of the light, so it's reversible, right? So active state and inactive state can be chosen by selecting the lifetime of cis or irradiating the whole body with the light. So reversibility is the biggest difference.
Yeah, reversibility, okay.
And so our molecule, it's like a chemical effect, not like so single oxygen damage is like so physical. So it's like more I can say soft way. I think.
Okay. All right. Okay. Any more questions? No. Okay. So thank you very much. That was an excellent lecture. So let us.
Great knowledge with us, and sincere thanks to Dr. Aravind Kumar CT for chairing wholeheartedly. Now, I request Dr. Aravind Kumar to kindly present the memento of appreciation to Professor Dr. Tamoki Nobuyuki. May I kindly invite Professor Nobuyuki and Professor Aravind Kumar CT. Thank you, sirs. Now I welcome Dr. Franklin John, Head of Chemistry Department, to present a memento as a token of gratitude to Dr. Aravind Kumar CT. Thank you, sirs. A few announcements. Those who are participating in the poster presentation competition can hang their posters in the premises of Department of Chemistry on first floor before 5 p.m. The poster evaluation will start at 5 p.m. Our volunteers are happy to help you all. Also, I am extremely delighted to inform you all that we have set a wonderful cultural event which will begin at 7 p.m. I welcome you all for the event on behalf of Department of Chemistry. Thank you.
Good afternoon all. Next, we are moving on to Plenary Lecture 3 of NTAC-2023. The lecture will be delivered by Dr. Biju A.T. on N-heterocyclic Carbene catalyzed umpelongophaldehydes, Michael acceptors and aldemins. and I'm very glad to take this opportunity to introduce our chair. Dr. S. Anas is the Associate Professor of School of Chemical Sciences, Mahatma Gandhi University, Kottayam. He is the Honorary Director of Advanced Molecular Material Research Center and the Institute for Integrated Programs and Research in Basic Sciences of MG University. His areas of research includes transition metal catalyzed CH activation reactions in organic synthesis and design, synthesis, and studies of functionalized polymers. He has a large number of papers in international journals to his credit, and he is a member of many statutory bodies of MGU and QSAT. Also, he owns an Indian patent in development of novel reusable polymer-supported transition metal catalysts for coupling reactions. With extreme gratitude and pleasure, let me hand over the session to Dr. S. Anas. Welcome, sir.
Good afternoon to all. It is indeed a great pleasure and privilege for me to be here as part of this great event. Thanks to the organizers for inviting me, and especially chairing a session which is going to handle by Dr. Biju, who was a co-researcher during my PhD time in NIST Trivandrum. He was always motivating and inspirational to all of the co-researchers and the junior students. I take this opportunity to thank Dr. Biju for your wonderful time there. Coming to his introducing Biju, Dr. Biju has completed his MSc from this college in the year 2001, and after that he joined the NIST Trivandrum with Dr. Vijay Nair for carrying out his research in the area of organic chemistry, where he was working on the citron chemistry, which is citron developed from triphenyl phosphine and diazo dicarboxylate. And after completing an excellent PhD there, he moved to Taiwan, National University of Taiwan, for his first postdoctoral studies. And later, where he was working with Professor T.Y. Liu for 1 year, and after that he was awarded the prestigious Alexander von Humboldt Fellowship in Germany, and he moved to University of Münster to work with Professor Frank Glorious for 3 years, where he was working excellent chemistry in NTAC catalysis and related areas. And in the year 2011, he got a position in NCL Pune as a senior scientist, and later in 2017 he joined as a associate professor in Indian Institute of Science, Bangalore, in Organic Chemistry Division. He has got excellent track records in the publication track record, published more than 135 papers in international reputed journals, and edited 2 books and author of 9 book chapters. And also, more importantly, he has got 9 US patents to his credit. He is a recipient of several honors and awards, and including the MRSI Silver Medal and the Best Scientist Award of the NCL. So many lists. And when the organizers given me the CV, it may take nearly half an hour to read out all his awards and honors. And also he's actively involved in the editorial assignment of various journals, and he is currently the editor-in-chief of the Journal of Heterocyclic Chemistry, and also the editorial advisory board member of many journals, including chemistry, European Journal of Asian Journal of Organic Chemistry, Synlet, etc., etc. So I'm very happy and proud to invite my friend and inspiring researcher. And I would say that he's one among the leading and one among the top organic chemists of the country. You are going to witness that due to his hard work, he has now reached in that level. With this word, I invite Dr. Biju for his lecture on N-heterocyclic carbene catalyzed ampholing of aldehydes, mycloacceptates, and aldimines. Thank you.
Okay, so good afternoon to all of you. Thank you very much, Anas, for the very kind introduction. And at the outset, I would like to thank Ramakrishnan and Franklin, and also all the organizers of this NTAC 2023 conference for inviting me for giving a talk on one phase of our research on carbene catalysis. So in the next 45 minutes, I will try my best to give you a take-home message on the chemistry of N-heterocyclic carbene and how one can use this catalytically active species in forging carbon-carbon bonds. So we know that catalysis is one of the sustainable methods for forming carbon-carbon and carbon heteroatom bonds. So when one does the catalysis, we can think of wide variety of carbon-carbon and carbon heteroatom bond forming reactions. And if one uses an enantiomerically pure or chiral catalyst for a given transformation, one can think of synthesis of a wide variety of chiral molecules. And we know that there are several ways one can do catalysis, starting from the enzyme catalysis, where biomolecules act as catalyst. The second one is transition metal catalysis, where metals such as palladium, platinum, or palladium, ruthenium, rhodium, so several metal catalysts can be used to form carbon-carbon bonds. So there is a third area which all of you may be aware that this area has been recognized by Nobel Prize in 2021, the small organic molecules-based catalysis, where one can think of using organic molecules for catalyzing a given transformation. So among these organic catalysts or organocatalysis in general, there is a subclass in this vast realm of catalysis where one can use N-heterocyclic carbenes where one can use N-heterocyclic carbene as catalyst for doing carbon-carbon bond-forming reactions, where these carbenes are responsible for unconventional modes of reactivity. I hope in the next few minutes I will be able to convince you how carbenes can be used for forging bonds in an unconventional way. So before going to the details, I would like to give you just an introduction about what are carbenes and how it can act as a catalyst. So we know that carbenes are electronically sextet, very short-lived species, and because of it, it is highly reactive and are divalent species. And whenever we think about carbenes in general, the first thing which will come to our mind is carbenes are having 6 electrons in the outermost shell of carbon, or in other words, carbenes are electrophilic in nature. So typical examples are dichlorocarbene, the intermediate in a Riemann-Diemann reaction, or a dicarboethoxycarbene, so where the carbene carbon is flanked by electron withdrawing grooves, which makes the carbene center more electrophilic. On the other hand, there are a class of carbenes where the carbene carbon is flanked by heteroatoms such as oxygen, nitrogen, or sulfur, where the mesomeric effect of the heteroatom, which pushes the electron density towards the carbene center, thus making the carbene carbon more nucleophilic instead of the inherent electrophilic character. So the idea is we need to have a carbene carbon flanked by heteroatoms such as oxygen, where the mesomeric effect or the resonance effect plays a vital role in bringing this carbene carbon nucleophilic instead of the inherent electrophilic reactivity. So once this carbene carbon forms the part of a ring where the carbene center is flanked by nitrogen, sulfur, where the mesomeric effect of the heteroatom pushes the electron density towards the carbene carbon, thus making it nucleophilic. So such carbenes can be generated in solution, starting from either the imidazolium salt or the thiazolium salt or even the triazole derived salt by adding a mild base, which will abstract this most acidic proton, leading to the generation of carbenes in solution. So in most of the carbene catalyzed organocatalytic transformations, this catalytically active carbene intermediate can be generated in solution from the corresponding stable azolium precursors in the presence of a mild base. So when we think of the carbene catalysis in forming various bonds, the first and foremost application of carbenes in organocatalysis is the addition of this carbene to electrophiles such as aldehydes, Michael acceptors, or aldimines. For instance, if we consider the addition of an aldehyde, the addition of a carbene, which is nucleophilic, to an electrophilic aldehyde, which will convert the flat aldehyde to a tetrahedral intermediate like this, which could undergo a proton transfer, leading to, again, a flat system, which is a hidden enamine, as you can see, which is postulated by Ronald Breslau from Columbia as early as 1958, which is now popularly known as the Breslau intermediate. So once you add carbene to this electrophilic aldehyde, which is electrophilic at the carbonyl carbon, but the addition takes place in such a way that now, after the addition, the aldehyde carbon is nucleophilic, or in other words, it is simply an acyl anion equivalent. So in the process of addition of carbene to an aldehyde, what we have done is we have changed the polarity of an inherently electrophilic aldehyde to a nucleophilic species by adding a catalytically active species. Or this is known as the polarity reversal or inherent reversal of polarity, which is otherwise known as umpolung, and in this case it is an aldehyde. So once this acyl anion is generated from aldehyde using carbene, this acyl anion can add to a second aldehyde, forging a carbon bond in a benzoin condensation, or it can add to an activated carbon-carbon double bond to form another carbon-carbon bond, or the construction of 1,4-dicarbonyl compounds in a name reaction, which is called Stetter reaction. So this is the most important mode of reactivity of carbene. So carbenes can do reactions or catalyze reaction in an unconventional way. So in contrary to the normal polarity concept, carbene can be useful for the reversal of polarity of aldehyde in this specific case, where the reaction proceeds via the Breslau intermediate. So the Breslau intermediate was just a proposal in 1958, but it was fully characterized by Professor Berkesel and co-workers from Cologne, and now all the Breslau type intermediates can be fully characterized, and even the crystal structure of these intermediates are reported by Berkesel and co-workers. So if you look at the Stetter reaction, where the aldehyde addition to a Michael acceptor, so the carbene adds to this aldehyde, generating the nucleophilic Breslau intermediate, which is now nucleophilic at this carbon, and this nucleophilic species can add to the activated carbon-carbon double bond to form an enolate equivalent, and a subsequent proton transfer and the elimination of free carbene leads to the formation of 1,4-dicarbonyl compounds. So when we started working in this area of carbene catalysis, the benzoin reaction was known, the Stetter reaction was known. So as I said, the Stetter reaction is the addition of aldehyde to Michael acceptors, which where the carbon-carbon double bond is electron poor. However, the addition of these nucleophilic Breslau intermediates to moderately electron poor Michael acceptors such as vinyl sulfones and vinyl phosphonates were not known. So we have demonstrated the addition of a simple aldehyde, regardless of whether it is aliphatic or aromatic, to vinyl sulfones, leading to the formation of gamma ketosulfones in moderate to good yield. A thiazole-derived carbene generated from this thiazolium salt using potassium terbutoxide was key to the success of this reaction. So inspired by this, we extended this concept from vinyl sulfone to vinyl phosphonate, where a variety of aromatic aldehydes can forge a carbon-carbon bond with vinyl phosphonates, leading to the synthesis of gamma ketophosphonates in moderate yield. So Stetter reaction, where aldehyde can be added to electron poor carbon-carbon double bond. So in addition to this Stetter reaction, where an aldehyde can add to a Michael acceptor, this carbene catalyzed strategy can be combined with non-organic reactions. For instance, one can think about a cascade reaction where a steter reaction can be combined with an aldol reaction. So this was demonstrated by Songe and co-workers from Shanghai. A dialdehyde reacts with an acrylate in the presence of carbene, resulting in the formation of a tetralone derivative in high diastereoselectivity. So what happens in this reaction is the carbene adds to one of the aldehyde moiety, generating the nucleophilic Brislow intermediate as in the previous case. Once this Brislow intermediate is generated, this could add to this Michael acceptor, so the vinyl ketone in this case, and the enolate thus generated can add to the second aldehyde in a diastereoselective fashion to force this carbon-carbon bond and the synthesis of tetralone derivative. So we were inspired by this Songe report, where the steter-aldol reaction can be eventually achieved, and we thought, can we use this concept, the steter-aldol type cascade process, for the synthesis of axially chiral molecule? And we know that axially chiral molecules such as biaryls and related compounds with a carbon-carbon axis is widely known in organic synthesis, because these are core structure in many natural products and biologically active molecules. However, the axial chirality, or the CN axis, specifically carbon-nitrogen axis, is relatively unknown. So we thought, can we use this concept, the steter-aldol strategy, for the synthesis of an axially chiral or enantiomerically pure CN axially chiral molecule? So the idea was, we typically use the Songe strategy, where a dialdehyde was treated with a 2-tertiary butyl aryl malimides, where we have this carbon-carbon double bond, which is electron deficient, which can be a replacement for an acrylate, where the reaction can proceed via a steter-aldol strategy to form a carbinol like this, which contains 2 chiral centers, which are point chiral, and a CN axis, where the CN axis can be controlled by inhibiting or introducing a tertiary butyl group at the 2 position to lock the rotation. The free rotation of the bond can be restricted. And now this compound contains 2 chiral centers and an axis. So to make things simple, what we did was, in situ the carbinol was oxidized using an oxidant, so that this carbinol gets converted to this ketone, or these 2 chiral centers are basically destroyed. That means the compound features only a CN axis. So this was our idea, whether we can use this steter-aldol type strategy for constructing a succinimide derivative containing only a CN axis. Because this type of CN axis, the compounds featuring CN axis, is relatively less known in organocatalysis. So this was our idea. So with this background, Saumen Barik, a talented integrated PhD student in my group, started the optimization of reaction conditions. And finally, he found that using an amino-indinol-derived chiral N-heterocyclic carbene precursor, so as you can see, this carbene precursor is chiral, and the carbene was generated using Hunix base as a base. And after the carbinol generation, the reaction mixture was subsequently oxidized using PDC to form this succinimide derivative with a CN axis, and this product was formed in 70% yield and 90% enantioselectivity. So this reaction was found to be quite general with respect to a wide variety of malimide derivatives and symmetrically substituted dialdehydes, and in all cases the products were formed in reasonable yield and enantioselectivity. So with this interesting result on scope of this reaction, we thought, or we gave a tentative mechanism of this reaction, where this reaction proceeds via the generation of free carbene, and the free carbene adds to one of the aldehyde moiety, generating a tetrahedral intermediate, and this undergoes a proton transfer to form the nucleophilic Brislow intermediate, and now this intermediate is nucleophilic enough to add to the carbon-carbon double bond of the malimide, generating an amide enolate equivalent, a tri-substituted amide enolate equivalent, which undergoes a proton transfer to generate a tetra-substituted enolate equivalent, which is more stable likely. And now this enolate undergoes an intramolecular aldol reaction to form the desired carbinol derivative, which was subsequently oxidized using PDC to form the desired CN axially chiral molecule. We also considered the elimination of free carbene first, and then an aldol reaction where the release of free carbene generates this ketone or aldehyde intermediate, and a subsequent aldol reaction can again lead to the formation of this carbinol, which can be subsequently oxidized. So to get insight into which mechanism is operating, whether the pathway 1 or pathway 2, where the carbene is eliminated prior to the aldol or not, we seek the help of my colleague, Garima Jindal, an expert in DFT calculation. And what Garima found was the elimination of carbene first followed by an aldol reaction, or the pathway 2, is indeed stabilized by almost 5.4 kilocalories per mole, which gave us some hint that this reaction is proceeding via the pathway 2, where the carbene elimination takes place and then an intramolecular aldol reaction takes place. So with this result on the CN axially chiral molecule, we also gave some insight into the mode of enantiomer induction. So if we see the 2-tertiary-butyl phenyl malimide, if we consider the plane of the molecule through the aryl group, one can think about 2 atropotrophic phases for this molecule, where the nucleophile can add to either CA or CB. So if we consider the nucleophilic addition at CB, you get this enantiomer, or alternatively, if the nucleophile adds to CA, we get the opposite enantiomer of the product. So this is a known process, or this is known in the literature, widely known for this 2-tertiary-butyl substituted amino malimide derivative. So we believe that in our case, the nucleophilic Breslow intermediate adds from the atropotopic rephase of the molecule, which undergoes, or which further undergoes an aldol reaction to form this desired product, which was confirmed by single crystal X-ray analysis. And we feel that the atropotopic psi-phase addition of the nucleophilic Breslow intermediate to CA, leading to the opposite enantiomer of the product, is less likely because of the geometry of this chiral carbene precursor, where the bottom phase addition will be inhibited. So we also gave some insight into the stability of the formed CN bond, how stable will be the axially chiral CN bond. So we found that if you take this product, the succinimide derivative, having a CN axis and heat the product up to 90 degrees Celsius, we observe that there is no appreciable change in enantiomer selectivity, which indicates that the CN bond is almost locked, it cannot rotate. But beyond 90 degree, we found that the E value drops, and up to and almost at 150 degree Celsius, the product is racemic, which indicates that at 150 degree, the CN bond is free to rotate. And we feel that at 150 degree, the racemization means the CN bond is free to rotate. So moreover, we also determined the rotational barrier. So at what energy the CN bond can rotate. So following the procedure of Dennis-Curran, we found that the rotational barrier for the CN bond is 32.4 kcal per mole, which was obtained following this procedure of Dennis-Curran. And with the theoretical calculation, my colleague Garima found that the rotational barrier for the CN bond is almost 34 kcal per mole using DFT studies, which is more or less in agreement with the experimental value. And I should confess that all this reaction leading to atroposelective succinimide derivative works only with the tertiary butyl group at the 2 position, and it is obvious that the tertiary butyl group at the 2 position will lock the CN bond and prevent it from rotation. To get more information on this, we have prepared 2 more substrates, the methyl and isopropyl substituted substrate, and treated it with the phthalaldehyde, and the reaction works under the optimized condition in moderate yield, and as you can see, the enantiomer ratio is 50-50. That means it is just racemic mixture. That means with methyl or isopropyl substitution, the CN bond is free to rotate, and that's the reason for the fast racemization. To get more insight, what we did was the DFT calculation by Garima again to find the rotational barrier for with a methyl substituent it was 22.4 and isopropyl it was just 24, which gave a hint that the low rotational barrier for the key carbon nitrogen bond rotation leading to the racemic mixture of the product. So in short, we demonstrated a desymmetrization of simple 2-aryl or 2-tertiary butyl aryl malimide derivative for the atroposelective construction of CN acylichiral succinimide derivative featuring a high substrate group compatibility or substrate compatibility, and the products were formed in up to 95% yield and 94% enantiomer selectivity. So as I told you, this is just a desymmetrization of a 2-tertiary butyl phenyl malimide derivative. So if we look at this simple 2-aryl substituted malimide derivative, it is a symmetrical system or a symmetric molecule. And what we have demonstrated is how to break the symmetry of the system. So this molecule is symmetric, which is convincingly shown in a single peak in an HPLC pattern. So which means that this molecule is achiral and prochiral. That means you can do one operation on this molecule to make it chiral. So if you break this symmetry of this molecule by introducing a substituent at the olefinic position, now the molecule is no longer symmetric, it is chiral, and if there is no chiral source for the synthesis, this molecule will be racemic, which is confirmed by an HPLC pattern. The 50-50 HPLC pattern is an indication that this molecule is now not symmetric. So what we envisioned was, can we use this concept or the breaking the symmetry or starting with a broken symmetry substituent on the malimide, so specifically an amino substituted malimide derivative, and use of a catalytically active mode of reactivity in carbene catalysis, which is the alpha beta unsaturated acylosolium chemistry. Now this is an enamide system or specifically an enamine, and this is an electrophilic system, and we envisioned a 3+3 annulation reaction where one can think about the separation or the separation of these enantiomers of this malimide derivative using carbene catalysis. So what specifically we envisioned was, out of the 2 enantiomers of this malimide, one of the enantiomer will react with this alpha beta unsaturated acylosolium using carbene catalysis, where the opposite enantiomer will be intact, which essentially could lead to a kinetic resolution strategy. So this alpha beta unsaturated acylosolium can be generated from a wide variety of enols using carbene catalysis, and the generated Breslow intermediate can be oxidized using a 2-electron oxidant to generate this intermediate. So over the years, we used this single intermediate in carbene catalysis, the alpha beta unsaturated acylosolium intermediate. The key alpha beta unsaturated acylosolium intermediate for the synthesis of a wide variety of carbocycles and heterocycles in high enantiomerically pure form. So with this experience in the alpha beta unsaturated acylosolium chemistry, again Saumen Barik, what he found that the amino malimide react with an oxidized bromoenol using a chiral carbene, and we found that only one of the enantiomer of this malimide react with this bromoenol in a 3+3 fashion to form this dihydropyridinone derivative in reasonable yield, diastereoselectivity and enantiomer ratio, and the opposite enantiomer was unreacted, which leads to the kinetic resolution of these amino malimides, and which works up to 94% EE value. And this reaction was also found to be quite general. A wide variety of bromoenols and amino malimides are all well tolerated under the reaction conditions, and the products were formed in reasonable yield and enantioselectivity values. So the reaction proceeds again via the generation of free carbene. The free carbene adds to the aldehyde in a 1,2 fashion, generating the 1 nucleophilic Breslow intermediate, and now a debromination can lead to the electrophilic alpha beta unsaturated acylosolium intermediate, and now this enamine moiety can add to the beta position in a Michael type fashion, generating or forming the key intermediate, where it has to be noted that only one of the enantiomer of this racemic maleimide will react, leaving the other enantiomer unreacted to form this key carbon-carbon bond, and a intramolecular cyclization could result in the formation of the desired dihydropyridinone product. So what we believe is that only one of the enantiomer of this maleimide, so if we consider the maleimide species with the 2-tertiary group projecting above the plane, the nucleophilic addition from the bottom phase is feasible, the top phase is prevented by the amino-indolone moiety, and the bottom phase can be feasible, or the rephase addition is feasible, and this leads to the reactivity where the opposite enantiomer of the maleimide, where the tertiary butyl group is projecting below the plane, will not undergo any sort of addition to this alpha beta unsaturated acetylsodium, and hence this will come out as out of the catalytic cycle as an untouched and enantio enriched substrate. So in short, we demonstrated a kinetic resolution strategy for resolving this racemic maleimide to enantiomerically pure maleimide along with a dihydropyridinone substrate. So far I told you about the construction of axial chirality with a CN bond, or atropos selective synthesis of CN axially chiral bonds. So now we thought, can we extend this concept to the atropos selective synthesis of NN axially chiral molecule? This will be even challenging because the NN bond has a weaker bond energy. So the NN bond is normally a weak single bond, and this is prone for deplanarization, and the rotational barrier for the NN bond is relatively less compared to a CC or CN bond. And more important, there is not much information available for the NN axially chiral molecules in the literature. So based on these things, my student Kuruva Balana, after several NN bond containing hydrazine derivatives trials, he found this oxazine, or this type of heterocyclic molecule could be a substrate for introducing chirality leading to the NN axis. So this quinosolinone derivative, as you can see, one of the nitrogen atom part of the quinosolinone moiety, which is rigid, and the second nitrogen is connected to an ester or a carbamate indeed, and the fourth, the next NH can be functionalized using carbene catalysis using a chiral carbene. This was the idea. So Kuruva Balana smartly designed this system, and he found that using a chiral carbene, an NH functionalization, indeed an amidation reaction, resulted in the formation of this product in 94% yield and excellent enantio selectivity. So this is the first time using carbene catalysis and atropos selective synthesis of axially chiral NN bond, or specifically the quinosolinones, were prepared in good yield and enantio selectivity. So the reaction also features excellent substrate scope, substrate functional group compatibility, and in all the cases the NN axially chiral compound or NN axially chiral compounds were formed in moderate to good yield and excellent enantio selectivity. So here also the reaction proceeds via the generation of free carbene from the azolium salt. The carbene adds to the aldehyde, generating the tetrahedral intermediate, and a proton transfer leads to the alpha beta unsaturated Brislow intermediate, or the homoenolate equivalent. And now this can undergo oxidation using the Karash oxidant to form the key azolium intermediate. To that, the quinosolinone can add the nucleophilic addition through the via the NH moiety, leading to the formation of the desired product. And we also feel that the reaction proceeds again via the bottom phase addition of the quinosolinone, the psi phase addition, which is favored, and the rephase addition, the top phase addition, is inhibited by the catalyst, which is projecting the amino-indane core of the catalyst is projecting above the plane, so that the nucleophile always approaches the azolium from the bottom phase of the molecule, which leads to high enantio enrichment. So far I told you about the carbene catalysis, where polarity reversal happens from aldehyde. So the carbene addition to aldehyde, generation of Brislow intermediate, and subsequent reactivity. And I have shown you a couple of examples where we utilize this concept for the construction of axially chiral molecule. And how about carbene catalysis beyond aldehydes? or beyond a simple Breslow intermediate. Can we use the carbene catalysis for the polarity reversal of a simple Michael acceptor or a methyl vinyl ketone specifically? So this question was answered by Greg Fu and co-workers as early as 2006. One can use carbene catalysis for the polarity reversal of simple alpha-beta unsaturated ester. As you can see, the alpha-beta unsaturated ester is electrophilic at the beta position. But using carbene, one can reverse the polarity and then displace this bromide to form the exocyclic cyclopentene derivative. And this concept was extended to an intermolecular version by Matsuoka and Glorious group independently to form a tail-to-tail dimerization of methacrylate derivative. And these reactions are expected to proceed via the deoxy analog of Breslow intermediate. So the carbene adds to the beta position the proton transfer, generating a deoxy Breslow intermediate instead of a simple Breslow intermediate in the case of an aldehyde. So we thought, can we use this concept, the polarity reversal concept using carbene, and can we use it for a simple methyl vinyl ketone? And we thought, can we use it for a simple chalcone, the polarity reversal of chalcone? So what we did was a stoichiometric reaction of this chalcone with an carbene generated from the diisopropyl substituted imidazolium salt, and this reaction works very well to form this diketone derivative in 60% yield. So this is envisioned to be a deoxy Breslow intermediate which is oxidized at the enolate position or the alpha position of the ketone. So what we thought was this reaction proceeds via the addition of free carbene generated from this imidazolium to the beta position of this enol, generating a tetrahedral intermediate, and now a proton transfer could lead to the formation of this deoxy Breslow intermediate. But unfortunately, what happened is under the reaction condition, under basic condition of potassium terbutoxide, this enolate generated was trapped by dissolved oxygen present in the system to form the 1,2-diketone. Or in other words, we tried to do this reaction in the presence of air. We got a slightly improved yield of 79%, where the Breslow intermediate was subsequently oxidized by air. That indicates that this product is formed via a irreversible addition of carbene to the Michael acceptor, or this product is very stable. That means one cannot use this strategy for a catalytic transformation because the carbene is adding to the Michael acceptor, but there is no way it can come out or come back and make this catalytic circle active. So with this simple chalcone, there is no possibility of catalysis. So then we thought, can we use a different ketone? And after several trials, my student Shilpa Barik came up with a cyclopentene dione derivative with a dimethyl group. And in this particular case, when we added carbene and imous-derived carbene to this Michael acceptor, we could isolate the deoxy Breslow intermediate in 89% yield. There was no appreciable oxidation at the enolate position, where this reaction proceeds via the initial addition to generate the tetrahedral intermediate and then the proton transfer to generate the key Breslow intermediate. And this is isolable, and since there is no appreciable oxidation at the alpha position, we thought there is a possibility of catalysis. So what we did was, instead of a catalytic reaction, a stoichiometric reaction, we planned a catalytic reaction using carbene and a different electrophile. So what we tried was use of this ketone and an N-substituted icetin as an electrophile using a thiazole-derived carbene, which was optimized by Shilpa, which leads to these oxindole derivatives in very high yield and good substrate scope compatibility. So here, the reaction proceeds via the generation of the free carbene, the thiazole-derived carbene. It adds to the Michael acceptor, generating the tetrahedral intermediate, and then the deoxy Breslow intermediate, and now the deoxy Breslow intermediate adds to more electrophilic carbonyl of this icetin, generating an alkoxide, a proton transfer, and release of free carbene results in the formation of this oxindole derivative. At this stage, we also considered the possibility of a pathway where without this umpolum or the pathway without the generation of the deoxybristol intermediate. So a simple tetrahedral intermediate initially generated can add to the icetin in a Morita-Baylis-Hillman pathway, generating this alkoxide. It could undergo a proton transfer, and elimination of carbene could lead to the same product. However, the key bristol intermediate, the deoxybristol intermediate, was isolated under the reaction condition, and we have enough experiments to prove that this reaction is not proceeding via a Baylis-Hillman pathway, and we have enough experiment to conclude that this reaction eventually proceeds via the polarity reversal of this Michael acceptor. So far I told you about the polarity reversal of aldehyde, and in the last 5 minutes, I spoke about the polarity reversal of Michael acceptors. And next, we know aldehydes, Michael acceptors, these are very good electrophiles in organic synthesis. And carbenes can be used to reverse their polarity. And we asked this question, can we extend this concept to reverse the polarity of an aldimine using carbene catalysis? So specifically, what we wanted was an aldimine which is connected to a Michael acceptor using a benzene tether. And as you can see, the imine carbon is inherently electrophilic in nature. So what we envisioned was the use of carbene to reverse the polarity of this imine carbon from the inherent electrophilic nature to the nucleophilic character. And if successful, this can add to this Michael acceptor to form a carbon-carbon bond, or eventually a 2,3 disubstituted indole derivative. And if this has to form, or if this indole has to form via the polarity reversal, the reaction should proceed via the ASA analog of bristol intermediate. So remember, in the case of aldehyde, it was simple bristol intermediate. The Michael acceptor case, the deoxybristol. And now with aldimine, it may proceed via the ASA analog of bristol intermediate. So this was our concept. So my student Attanu Patra, a PhD student, worked in this project. So he came up with a simple acrylate, beta substituted acrylate substrate, and he tried a variety of conditions and finally found that with an N-phenyl trisodium salt and potassium terbutoxide for carbene generation, this reaction eventually resulted in the formation of 2,3 substituted indole derivatives in high yields. And we can see that a wide variety of substituents are well tolerated, and the 2,3 indole formation was quite successful with very high yield. So this reaction proceeds via the generation of free carbene. The free carbene adds to the imine moiety, generating a tetrahedral intermediate and a proton transfer as usual. And in this case, it is a ASA analog of bristol intermediate. At this point, we also considered the addition of free carbene instead of to the imine. There is a possibility the carbene can add to the Michael acceptor, generating a deoxybristol intermediate. And if that case, the deoxybristol intermediate can potentially add to this imine electrophile, generating the same product. However, our initial mechanistic studies supported by DFT calculations by my friend Kumar Vanka from NCL Pune, we have sufficient grounds to believe that this reaction proceeds via the ASA bristol intermediate. And once this ASA bristol intermediate is generated, it could add to this carbon-carbon double bond to generate an enolate, and a subsequent elimination of carbene could result in the formation of 2,3 disubstituted indole derivative. So far, we use this strategy, the polarity reversal of imines, for the synthesis of indole derivative. So we thought, can we extend this beyond indoles? Can we use this concept, the polarity reversal of imines, for the synthesis of a 6-membered ring? So again, my student Attanu Patra, he came up with an imine which is connected to an alpha trifluoromethylated styrene derivative, and the idea was to reverse the polarity of this imine carbon, and he was eventually successful with the construction of a 2,4 disubstituted quinoline derivative, or specifically a 4-difluoromethylated quinoline. So the Asa-Brislow intermediate generated here makes this carbon nucleophilic. It adds to this moderately electron-poor carbon-carbon double bond with an elimination of a fluoride, resulting in the formation of this difluoromethylated product. As I said from the very beginning, we although believe that this reaction proceeds via the Asa-Brislow intermediate, we cannot rule out the initial addition of carbene to the Michael acceptor. To prove that this reaction is essentially happening via the Asa-Brislow intermediate, we designed a substrate, a bis-imine, where we believe that the carbene adds to one of the imine moiety, generating the Asa-Brislow intermediate, and the second imine moiety acts an acceptor of the nucleophilic Brislow intermediate, and this reaction eventually afforded the dihydroquinoxylin in 98% yield and 98% enantiomer ratio. These are the selected examples of the dihydroquinoxylin synthesis. So in the last part of my presentation, I hope I was able to tell you the way one can do catalysis using carbenes to reverse the polarity of aldimines for the construction of wide variety of indoles, quinolines, and quinoxylin derivatives in high enantio enriched form. And to conclude in the last 45 minutes, I hope I was able to give you a take-home message on the chemistry of carbenes and heterocyclic carbene, how this catalytically active species can be utilized for the polarity reversal of aldehydes, where the reaction proceeds via Brislow intermediate. It can be extended to Michael acceptors, where the deoxy-Brislow intermediate is the key intermediate, and finally the aldimines, where the Asa-Brislow is the key intermediate. And I should thank all the students. I'm very happy to have extremely talented group of PhD students. So today I highlighted the work of Sauman Barik. So he is the key for axial chirality, and Balanna, he's about to move for his postdoctoral studies to Münster. So he demonstrated the NN axial chirality. Shilpa Barik, she demonstrated the Umpalung of Michael acceptors, and half of the students, the remaining students, are working in the area of benzene chemistry, which I spoke in the same space a few years ago. So also we are supported generously by various funding agencies, the startup grant from NCL Pune, and the continued support from IISc Bangalore. And at present, most of our research is supported by the funding from SERB, and I should acknowledge the continued support from SERB for most of our ongoing projects. And once again, Franklin and Ramakrishnan for the kind invitation to SH. Coming to SH is always a nice feeling, back to home feeling, because this is the place I learned chemistry, specifically organic chemistry. Wonderful group of teachers during my time. I still remember, so I met Joseph Jones Saturday morning. So it's a fantastic department, and this is for all students. You are blessed to have a group of teachers which are hardworking to teach you organic chemistry, and fantastic. So yeah, once again, thank you very much for listening to me, and I will be very happy to take questions. Thank you.
Biju, that was really good. A couple of questions. You use a variety of NSCs. Okay, how do you fine-tune the reactivity of these NSCs? Because you can see that, there are a variety of reactions you are talking about. One of the questions that I have is, when you start with aldehyde, and you have an alpha-beta unsaturated carbonyl compound also, the reaction proceeds in such a way that first one of the carbonyl groups of the thialaldehyde reacts, then a Michael type addition, etc., etc. Yeah. There of course will be a possibility that the first Brislow intermediate that has been generated, that will react with another, thialaldehyde molecule, so that you can get sort of a benzoin benzoin condensation there. That's not happening here. And of course the question which arises there is, which you have demonstrated later, NSCs react with alpha-beta unsaturated carbonyl compounds also. But in the first case, the NSC, the one that you selected, that really reacted with the aldehyde very preferentially giving extremely good chemoselectivity there. I'm not talking about the natural selectivity. And I noticed that the NSCs that you are using are quite different. So obviously this is an issue that you have looked into, but I'm curious to know, how do you fine tune or how do you really decide this is the NSC that I have to use to get this specific reaction in the specific chemoselectivity that I am looking for? Okay.
So yeah, to answer this question in a sincere way, to my knowledge it is still unknown. There is no literature as of now in carbene catalysis where one can confidently say that this carbene can do this particular catalysis. We just have a feel based on the literature knowledge. So we know that a sort of set of carbenes can be utilized for the acyl anion reactivity or the alpha beta unsaturated acyl sodium reactivity. So coming back to your first question, the phthalaldehyde case, where one can easily consider the benzoin pathway, but what we have seen, or not only we, many others observe that this benzoin formation is reversible under the reaction condition. So once the benzoin forms, the carbene acts to the carbonyl and it comes back to the aldehyde and another molecule of Brislow intermediate. So we feel that this is also possible in our case, although we did not experimentally prove it, but this is a knowledge in literature. And to the second question, there are ways one can tune the reactivity of an alpha beta unsaturated system. So for example, if you take a cinnamaldehyde, one can get the acyl anion reactivity or the reactivity at the beta position. So if one uses a less sterically demanding carbene, for example a thiazole derived carbene, then the carbonyl carbon will be less nucleophilic, more nucleophilic in terms of sterics. But if we want the reactivity at the beta position, we need to use a sterically demanding carbene, such as IMS hydrochloride derived carbene, where the carbonyl carbon reactivity will be significantly lowered because of the sterics of the carbon. Or in other words, the nucleophilicity is pushed from the carbonyl to the beta position by tuning the sterics. So by tuning either the sterics or the electronics, again as I said, this is not conclusively proved. At least we have a feel that a given carbene can catalyze a given transformation to a certain extent. But what we normally do in our lab is, we have a library of carbenes in our lab and we have to test the reactivity with all carbenes. Yeah. And even the painful thing is that unlike all other areas of organocatalysis, where one starts with the catalytically active species, for instance, proline catalysis or hydrogen bonding activation, we start with the catalytic active species. But in carbene catalysis, we start with the azolium precursor and we generate the carbene in solution. So we have an additional parameter, which base will be used, the solvent. So it's, we have to try everything. Yeah. Thank you.
Yeah, I want to ask a question. I follow your demonstration, and your synthesis was very good. But my question is this. I saw you doing the synthesis, you use some compound that have similar, I don't know, they have similar reactivities. But when I was following you, I never hear anything like those groups that have similar reactivity that you protected any of them, but the reaction just flows smoothly. I don't know if I made a mistake, but I was following you. And some compound you use, they have almost similar reactivities. But during your synthesis, there were no group that were actually protected.
Pardon me, can you please repeat once again? I did not get your question.
You are doing the synthesis, and some group have almost similar reactivities.
Absolutely, yeah.
And none was actually protected, but the synthesis just went on smoothly. That's where I have doubt.
So if I understand your question right, you are talking about this CN acylichiral molecule, right?
Yeah.
Yeah, so what we do here is, so first of all, we don't use any protecting group. Okay, so here what we basically do is we follow a reaction which is known in the literature, a simple Stetter-Aldol reaction, which is demonstrated by Ye and co-workers for a diastereoselective synthesis of this compound. So we adopted that knowledge for the synthesis of acylichiral molecule. So no protecting group, nothing. So we use a strategy, a cascade reaction, which is known in the literature. So the Stetter-Aldol cascade reaction, and by simply changing the acrylate, which is known, we use a maleimide, and essentially we could construct a CN acylichiral molecule. So there is no need of any protection or deprotection in this. A single step reaction, you start with a ethylaldehyde, generate the nucleophilic Briscoe intermediate, add to the second Michael acceptor, and an intramolecular aldol reaction. I hope it is convinced. Thank you.
Thank you for the great talk. My question is an extension to Professor Pratap's question. You showed an example where in one case the carbene acts as a reagent rather than a catalyst.
Yeah.
So is there a way to predict whether carbenes would act as a catalyst or as a reagent? And the second part of the question is, if you had used another carbene in that place, could you make it an example for catalysis?
Yeah. So I understand what the question what you asked. So actually we started with catalysis. Okay. So here, so our original idea was to reverse the polarity of this chalcone at the beta position. But with the catalytic amount of carbene, we obtain only traces of this compound with the incorporation of the carbene. So that indicates that the carbene addition to this chalcone is irreversible. So to make sure that this compound is formed, then we did a stoichiometric reaction. So we originally started with catalytic reaction, and in this case, say for example, we started with 10 more percentage of this, and we got just a hint in high resolution mass that this oxidized product generated. So with that knowledge, we tried the reaction with one equivalent of carbene precursor and isolated 60%. So this, it's all based on the observation from an experiment. And in this experiment, we understand that the carbene precursor or the carbene is incorporated in the final product. So to enhance the yield of this product, where the carbene is acting as a reagent, not a catalyst. So we use stoichiometric amount. And this reaction, since this oxygen is coming from the dissolved oxygen present in THF, we did the reaction in air, which gave the product in slightly excess yield. Okay. And this gave us a hint that this procedure cannot be utilized in catalysis, because once the Brislow intermediate is oxidized to a thermodynamically stable diketone, there is no possibility that this can be used in catalysis. So that's how we found that in the next species, with this cyclic 1,3-diketone, we found that this Brislow intermediate is isolated, so there is no oxidation at the alpha position. So this gave us a hint that this concept can be utilized in catalysis. And this experiment again, it is a stoichiometric reaction. We use one equivalent of carbene precursor and slight excess of this 1,3-diketone and obtained about 89% of this deoxy-Brislow intermediate. And with this knowledge on this Brislow or deoxy-Brislow intermediate, then what we did was, we want to make this reaction catalytic, but for that we need to add another electrophile that's why we added isotin as an electrophile, and now the carbene is 20 more percent. So it all started with catalytic amount of carbene. With the observation, we probably in some cases to isolate the adduct, we set up the stoichiometric reaction and then come back to catalytic reaction. I hope I was able to convince. Thank you.
How easy it is to remove the catalyst after the reaction? Because these are also organic. Is there any method to completely remove it off or destroy the catalyst? Because this looks everything almost of same.
To be honest, we did not try. So what we did was, so you can imagine, so in most of the catalytic reactions using carbene, the catalyst loading is 10 to 20 more percentage. So the turnover frequency is pretty low. So what we do is, in all the reactions, at the end of the reaction, we filter the reaction mixture through a pad of silica gel. We remove all the inorganic impurities, including the carbene precursor, because everything will stay at the top. Only the organic materials will come down, which makes purification much easier. And there is, as I said, unlike other modes of organocatalysis, where one can start with a catalytically active species, carbene catalysis is an area where we generate the carbene in solution from a precursor. So to answer your question in a different way, the isolation will be not the carbene, because carbenes are very unstable. So the way to go back is, at the end of the reaction, we need to add an acid to get back to the azolium precursor, and then probably it can be isolated. But we did not try. Okay? Yeah, Professor Tamaki.
So you are using the covalent bond for the catalytic reaction, right? So not different from the for example transition metal catalyst, right? So catalytic reaction need to be reversible, right? First, for example, complex between metal and the ligand, that is the formal way, right? So in your reaction, covalent bond is made, right? Yeah. So for me, it's a little bit how can I say it's a little bit difficult to use the covalent bond for the reversible reaction. So how can you explain energetically? So using the covalent bond is not so bad in comparison with using the metal ligand complex.
So very nice question. So it's not really on the covalent bond generated. Say, for example, carbene catalysis is basically covalent bond of activation. But I can tell you in a different context, enamine catalysis, for example, the famous aldol reaction demonstrated by Professor List and Barbas. Take an aldehyde, a ketone, add proline. So the proline will add to the ketone having an alpha hydrogen, form a covalent bond and generate the enamine, covalent bond formed species. So the key to success in that reaction is the reversibility between imine and enamine and a subsequent hydrolysis, which leads to the regeneration of the catalyst. So in our case, in carbene catalysis, the first addition, I will take you to the Briscoe intermediate. The addition of carbene to aldehyde, converting the planar aldehyde to a tetrahedral intermediate. This is reversible. The second proton transfer step to generate the Briscoe intermediate, again reversible. So the first irreversible step in the event is the nucleophilic addition of the Briscoe to a carbon-carbon double bond in a Stetter reaction. That is the first irreversible step. So I think regardless of the bond formed, it is more likely the ease of regeneration of the catalyst by breaking the bond. That is also feasible under the reaction condition. So this is known. This is a simple enamine catalysis. So the reversibility between imine and enamine, although the bond formed is a covalent bond, it simply forms and breaks. And similarly is happening in carbene catalysis.
Yeah, Biju, one last question from me. In the case of aldimines, you have looked into the aspect of the interaction of carbenes in the Michael acceptor part, aldimine. But have you looked at that kind of aspect in the case of aldehyde also? I don't remember exactly. I think you have detailed studies carried out with the aldimines only. Yeah, here you have explained the things, and what about the possibility in the previous two cases in such kind of, is there any possibility?
There is no possibility. We have only one electrophile.
Yes, but have you ever looked at this kind of possibility can be created there by the extension of the substrate or something like that?
No, the thing is that in all the cases with Michael acceptor unpolum, there is no question of other electrophile in the reaction system. So only in the case of aldimine,
yeah,
we have a carbon-carbon double bond where this can be also reversed.
Yes.
So that's why we considered this, and we have some hint of reaction. So if we generate an imine with a highly electrophilic aldehyde, so this carbon will be less nucleophilic because we have strongly electron withdrawing group. So the reaction will not work.
Yeah,
And that is what we observed.
Yeah, it's okay. One more last question. And one reaction you have showed that one fluorine or something is eliminating, and you have not given much details about the mechanism. I don't, if I follow is correct.
Right.
Yeah, could you please comment a little bit on the mechanism part?
Again, it's the same thing. So the imine polarity, the carbene adds to this imine. I'm sorry, the pointer is not really working. Can you change it? Sorry. Okay, so the idea was to reverse the polarity of this imine carbon. Okay, so the carbene adds to this imine carbon, generates a tetrahedral intermediate and an absorbed slow. And once this carbon is nucleophilic, it can add to this carbon-carbon double bond with a release of fluoride. And this will lead to an exocyclic double bond, which will be subsequently aromatized and generate the quinolane intermediate.
That time I missed this slide, that is why I asked.
So I did not show the mechanism.
Yeah.
Because it is obvious that it will undergo aromatization.
Okay. Thank you. Since we are running short of time, let me please join all of us to congratulate Biju for the excellent talk and wish you all the best for the future.
I hope you all had a valuable time of knowledge, and I owe my sincere thanks to Dr. Biju A.T. and Dr. S. Anas for handling the session. Now I request Dr. S. Anas to present the memento as a token of love to Dr. Biju A.T. and I invite Dr. Biju A.T. to receive the same. Now I invite Dr. Ignatius Abraham, Assistant Professor of Department of Chemistry, Sacred Heart College, Thevara, to hand over the memento of appreciation to Dr. S. Anas. A gentle reminder, the oral presentation will begin sharp at 3:50 at Malicious Hall, and the participants please stay back here itself. The participants of the poster presentation are requested to hang their posters on the respected poster boards nearby the Department of Chemistry, and the valuation of the same will begin at 5 o'clock. Also, I'm very happy to inform you all that our eminent artists and alumni are all set for a visual treat. The same will begin at 7 p.m. followed by dinner. I welcome you all on behalf of Department of Chemistry for the cultural meet. Now it's time for a tea break. The tea and snacks are arranged at the Marian Hall. Participants are requested to bring your coupon along with you and have your tea and kindly be back before 3:50 for the upcoming session. Thank you.
Good afternoon, everyone, and welcome back to 21st Professor K.V. Thomas Endowment Seminar and 4th International Symposium on New Trends in Applied Chemistry 2023. Now, it's time to start the oral presentations. Oral presentations play a crucial role in any conference as they provide a unique platform to exchange ideas and knowledge among peers. I hope that all attendees are as excited as I am to hear all these informative and insightful speeches. I welcome all of you to this session. The judges for the session are Dr. Porphyrins Matthew and Dr. Inaugural. It's my pleasure to introduce our esteemed judge, Dr. Porphyrins Matthew, Head of Research and PG Department of Chemistry, St. Thomas College, Thrissur. He successfully completed several projects funded by UGC and have several publications in internationally recognized peer-reviewed journals. Dr. Porphyrins is also a recipient of Boytas Fellowship from DST. He is a well-known research guide of Calicut University, and his current research interest includes organic synthesis and green catalysis. Now, moving on to our next judge, Dr. Inaugural. She is an assistant professor in chemical oceanography in the Department of Aquatic Environment Management, Thrissur. She has 17 years of experience in both teaching and research. Dr. Anu handled several projects funded by DFT, Ministry of Earth Science, and ISRO. She has publication in several international journals and has been awarded several fellowships, including Commonwealth Fellowship in 2012. I feel very much honored to welcome our judges to the session. And now, without further ado, let's begin the presentations. Please note the rules for presentation. The duration of presentation is 7 minutes and 3 minutes will be given for Q&A session. After 6 minutes, a short bell will ring and the final bell will ring after 7 minutes. Now, let me invite the first speaker, Deepak Patra, code number APB01 for the presentation.
All of you. It's my great pleasure to present our work in this platform. So thanks for joining us. So I want to discuss about our work, how responsive small molecular skills for molecular application. This work is carried out under the sponsor of Dr. Leon and Dr. Smith at Scale Scientist. When we talk about the energy smart materials, so actually the carbon neutrality in the 21st century, the world's largest mission. So there are many different government policies to push the energy conservation and reduction of energy. So there are like particular the study the production of energy is used for ventilation, air conditioning and cooling energy purpose. So how can we save this energy? So cooling is the most important part of the building block. So there are different energy materials based on the calorification, electrolysis and thermocooling. But among these thermocooling materials have great potential due to automatic acid controllability, zero energy input, high cost effective and easy implementation. So, 2013 from Berkeley Lab, they first published the basic IQ of smart windows. In their article, they actually explain how the smart window can switch to dark mode and that can block the light and heat, and it can also switch to bright mode and in the fully transparent mode. And when we talk about the thermocooling in the solvent reaction, the phase diagram, there are 3 different kinds of systems against CST. So these below that point, the systems are completely visible. And in case of UCST, above this point, the systems are completely visible. Like water, tidal, and system, and some kind of systems of both LCS and UCST, like water and hydrogen system. So we are working on based on LCST kind of system. So what is the LCST? So LCST is the intermediate phase science and behavior of binary solvent mixtures at acidic temperature due to the non-polar interactions. So we already know that the P-type of units, so the LCST temperature 32 degrees Celsius. So when at low temperature, that P-type of unit is hydrated by water molecules, and due to the free energy calculation, like that time the delta H value is predominant than enthalpy factor, that's why it is like a clear solution. When above that temperature, that time enthalpy factor is predominated, and it shows that Carbene solution, it shows that renal effect. So in our lab, we actually working on based on homogeneous material. We have synthesized and designed and synthesized one small molecular system based on thermosensitive unit and photoresponsive unit that is completely water soluble, and it's a very fast photosynthetic process, and it's also so that have long thermal half-lives, and it is easy to synthesis. And when we talk about the photoisolation, so there are two types of photoswitch systems. A two-state photoswitch system and is the multi-state photoswitch system. In case of two-state photoswitch system, A, that is thermal stable, that can go to the beta stable state, and B cannot stable under thermal and photochemical condition, like azobenzene and dihydroethene system. Another kind of system is the multi-state photoswitch system. There A can go to the B to C, and C can also come back to the A under thermal and photochemical condition, like donor acceptor systems. Here, in this work, we showed that our molecule actually is cyanosilibin substituted naphthalene unit that show multi-chromic photoswitching behavior. So here, that it can go to the G2E2 cycloadduct form. There is no visual color change. All of these cases, it is like white in color, but there is the color change in the under 365 nanometer UV light. It shows the grid is to cyan to deep blue in color. If you see this is the absorbance and emission spectrum. There are different distinct absorbance spectrum and corresponding emission spectrum. And if you see that molecule 2Z going to the 2E state within 120 seconds, and that's why that n-pass transition, the 355 nanometer, it decreases, and pi-pass transition to 285 nanometer, it increases with respect to the isostatic point 301 nanometer. And this is the absorbance with time, so it follows the first order decay kinetics. And this is the corresponding protein spectrum. Here our molecule exists is 94:6 ratio. So here after 24 minutes, it's going to 22:78%. So this is the protein spectra of 3 different molecular states. All 3 different molecular states show aggregation into stimulation behavior in aqueous medium, and that's why 3 molecular states show 3 different color. And when we talk about the thermal back isolation, here 2E can come back to 2G at high temperature like 85 degrees Celsius. So we have calculated that rate constant value at particular temperature, and we calculated the rate constant, put that rate constant value at Arrhenius equation and RD equation. We got the different thermodynamic parameters like activation energy, enthalpy, entropy, free energy. From there, we have calculated the thermal half-lives at ambient condition like 25 degrees Celsius. It showed that very long thermal half-lives like 41 years. And this is the mechanistic pathway. Basically 2Z, it is thermally stable form. It absorb the 360 nanometer UV light. It going to excited singlet state, and under intersystem crossing, it going to the singlet state to triplet state, and it going to the 2E state. And after next reaction is that if you continue light radiation, it going to the 2C form. So basically it is an example of consecutive reaction. So A can go to B, and if you continuous light radiation for 12 hours, it going to that C. So, but C can't come to B to A. So how can we use this classical photochemistry for energy saving purpose? So basically when molecule is photo switches from 2Z to 2E, that time the cloud point temperature actually decreases because all molecular components are thermoresponsive. So when it's going to the 2Z to 2E, the temperature decreases from 37 degrees Celsius to 30 degrees Celsius. So here we made the train by train cross window prototype. So we make that 0.5 millimolar concentration solution of aqueous solution, and we pour that aqueous solution into between glass plate. So when at low temperature like 30 degrees Celsius, we can see that it is the clear, we can easily see the outside object, but at high temperature it is like opaque in solution, so we can't see the outside object. And we go through the DLS study. So there we saw that at low temperature it is like 10 nanometers particle size, but at high temperature particle size increases to 165 nanometer. And this is the transmission spectrum. So here at low temperature, so it at visual region it show like near to 100% transmissive. So we can easily see the outside object, but at high temperature it is actually block the UV visual region as well as the NI region. So it indicates that it can block the heat and as well as the visual light.
And this is the actual transparent opaque state. So in case of 1330 cross big window prototype, it also shows similar behavior at low temperature. So we easily see the outside object, but at high temperature, we can't see the outside object. And this is the video graph. So within like 30 to 32 degrees Celsius, it's going to transparent to opaque state, and it can also come back to opaque state to transparent state from 32 to 30 degrees Celsius. And our molecule absorb is actually in the UV region, like 200 to 400 nanometer. We have whatever actually the water solute dye we can use, it will show the same color only. So we are using a red, green, and yellow dye, and it also show that transparent to transparent to opaque state, and that you can see the video. So basically in this work, we design and synthesize a single photochrome system that shows multi-photoswitching behavior through the JD photoisolation followed by cycloadduct, and it shows distinct photophysical property, and due to high thermal half-life and stability in aqueous medium, we can use for that chemical response having material for energy for indoor light and heat management. And this we have actually fabricated 1330 cross big window prototype, and it showed high T solution, T luminescence, and T alpha for like 10,000 cycles for stable operations. Thank you for kind attention.
Now audience can ask questions.
So I couldn't follow your presentation well, but you first show us the photoisomerization of the compound having the CC bond and cyano group.
Yes, sir.
And then you showed us the thermosensitive material. So what kind of the molecule do you use for the opaque state and transparent state switching? It's the same one?
All molecules we are using opaque state, transparent state. So basically, here we have three different molecules, three different forms. So all molecules are water soluble and colorless solution. So and when we go through from that GIS1 to EIS1, the LCST temperature decreases from 37 degrees Celsius to 30 degrees Celsius. So all these cases actually the only molecular formation changes, but color temperature actually decreases. And all molecules show that LCST temperature.
Okay, so depending on the isomer state,
Yeah.
the clearing temperature is different.
Yes,
so
yes.
precipitate makes the opaque state.
Yes.
Okay, I see. Okay, and I couldn't distinguish the okay, so finally to C is a cyclized, so
Yes.
That molecule cannot go back to the.
It is stable form.
I see. Okay, thank you.
Okay, and then in the NMR, in which solvent you have taken the NMR spectrum?
CD3Cl, sir.
CDCl3.
CD3Cl, acetonitrile.
Acetonitrile. You conducted the experiments in water. Why you have taken D2O?
Actually, in aqueous media, molecule is aggregated due to piper stacking interaction and hydrogen bonding. We are not getting the clear protein spectrum. So that's why we choose to, instead of water, we choose the CD3Cl, and we got visual protein spectrum. But in UV study, we can easily understand here. So this study is under aqueous media. So 2Z, it showed that in-pass transition at 355 nanometer and at bypass transition at 285 nanometer. So when we irradiate light, so within 120 seconds, molecule undergo from 2Z to 2E in aqueous media, and that time that in-pass transition actually decreases and bypass transition increases with respect to the isotopic point at 301 nanometer. But in case of water media, in D2O medium, we are not getting the resolve porphyrin spectrum. So that's why we moved to D2O2 CD3Cl.
After isolating the compound.
Yes, sir.
During isolation, is there any cis-trans isomerization possible or reversibility of the product possible?
Yeah, that 2Z actually thermally stable form, 2E actually thermally unstable form. So 2Z within 120 seconds is going to 2E form, and it can also come back to 2Z under high thermal condition like 85 degrees Celsius. So that's why we are studied the kinetic study, how much it will take time from 2E to 2Z.
Okay. Okay, thank you.
Thank you.
Thank you, Phil. Let me invite Anjali Nirmala, code number AEM01 for the talk.
A very good evening to one and all. I am Anjali Nirmala, working under the supervision of Dr. Ajay Ghosh and the co-supervision of Dr. Srijit Shankar in CSIR NIST Trivandrum. And first I would like to thank the organizers NTAC-2023 for giving an opportunity for the oral presentation in this platform. And also I am thanking to the CSIR NIST and my supervisors for the approval for presenting my work here. My topic is I have briefly taken the topic, thermally stimulated metal organic complexes for multi-state logic operations and energy efficient smart window applications. As the previous speaker said about the smart window applications and its energy efficient applications, I am also having my work on this particular behavior. For this, we have taken temperature as a stimulus. That is, temperature is not often considered as a precision stimulus, unlike light, voltage, or chemical systems. But temperature, we have chosen the temperature as a precision stimulus, and we have achieved the quantized delivery of temperature, and we have got the accurately stable, reproducible, and reversible responses for every 1 degree rise in temperature. And this could definitely, we have got explicit and defined stages between the initial and final stages. And of course, they have the system provided a large window of operation, and we have performed the logic gate applications, and for that we have got the reversal only upon withdrawal of the applied temperature. And the temperature has not been used extensively for logic gate operations capable of treating multiple inputs. And with this realization of multi-state molecular logic operations using thermal input alone is much more demanding. And also we have performed the switchable dynamic windows capable of exhibiting thermally modulated light and heat window transmission, and such chromogenic systems performing simple employing the simple metal ligand complexes is rarely found. For this, we have chosen the simple metal bipyridine complexes, that is the 2,2-bipyridine as chosen as the ligand L1, and an electron donating group at the fourth position of the bipyridyl ring as an electron donating group, and a conjugated pyridyl group is attached to the fourth position of the bipyridyl ring as the L3. And temperature and the structure and the structure shows the structure shows the complex the structure of the complex. Temperature-dependent optical spectroscopy has been probed as a tool for exploiting the nature of our system. So from 25 degree to 90 degrees Celsius, we have taken the we have checked the thermochromic behavior for all the 3 ligands, and for all the 3 ligands, we have got every 1 degree rise in temperature as a response. And with this, we have the intensity of the MLCT band at different temperatures have reached the initial value, reached its half initial value at 38 degrees Celsius for ligand L1. And for ligand L2, it reaches at 41 degrees Celsius, and for ligand L3, it is 48 degrees Celsius. So from this, we can we understood that there is an effect on electron donating and electron withdrawing groups on the pyridyl group. And this is the photograph showing every 10 degree rising temperature for all the 3 ligands with the metal complex iron. That is, for L1, we can see the complete bleaching of the MLCT band is happening at from 25 to 90 degrees Celsius in 90 degrees Celsius. And for the electron donating methyl group attached to the bipyridyl ring, that requires much more higher temperature for the bleaching of the complete bleaching of the MLCT band. And the conjugated pyridyl group attached to the bipyridine requires much more higher temperature for the complete bleaching of MLCT band. And this is the proton NMR spectra. We know that at the 25 degrees Celsius, the characteristic bipyridine peaks were seen completely, but at the heated paramagnetic behavior of the DMSO disease solution is seen here at 80 degrees Celsius, and at 25 degrees Celsius, it has been reversed back to the initial state. And this concomitant change in redox potential for Fe2 plus 3 plus oxidation is observed for all the 3 ligands, and for L1 requires much lower voltage than L2 and L3. And from this we have achieved, understood that there is an effect on electron donating and electron withdrawing groups on the bipyridyl ring for the thermochromic behavior. And also we have done the CV using a glassy carbon working electrode, a platinum counter electrode, and AgCl as a reference electrode in DMSO, that is the 3 electrode system, and 0.1 molar TBAPF was used as the supporting electrolyte. So from 25 to 90 degrees Celsius, we have performed the cyclic voltammetric studies. And this is of course, there will be a question regarding the DMSO is a coordinating solvent, and also DMF also we have chosen for, and other polar and non-polar solvents were also tried, but the thermochromic behavior was observed only for DMSO for a longer period of time. That is, at 25 degrees Celsius, the absorbance was maintained for a longer period of time in DMSO, but in room temperature itself, the DMS, the system was not showing the stable behavior. And this is the heating and cooling cycles of the 3 ligand systems, L1, L2 and L3 for the metal complex. And from this we can see that the time dependent absorption spectra is recorded over a longer period of time, at least 8 hours. We have seen the we have not seen the changes, but while cooling back, it traces to the initial value, and while reaching back the initial values, there is a stability. And the generality of the approach was also checked with the other metals like cobalt and nickel. For this, we have shown the thermochromic behavior from dark cyan to blue, and also from brownish to green, and the intensity of the MLCT band increases with the increase in temperature. And the switching cycles for stability studies have been mentioned in the in this slide. For up to 5 cycles, there is no change for 25 and 90 degrees Celsius. And this is the MLCT band showed a complete stability for longer period of time. So we have considered this for a sequential logic gate memory operations, and we have performed the logic gate memory operations for binary, ternary, quinary, and quaternary and ternary sessions. And also we have studied the smart gel applications. For this, we have added CTAP to the system, and the transmittance studies have been changed up to 25 degrees Celsius. For 50 degrees Celsius, it is opaque, and 90 degrees Celsius, it is completely colorless. And the prototypes have been showed for both the systems, iron, cobalt, and nickel. It is stable after several months of application. Color change is simultaneous with changes in transparency, and multicolor possibilities by using different ligands and donor metal salts. Use of temperature as a precision input is thermally responsive systems. So we have demonstrated multi-step stable and reversible solvent-assisted thermochromics in bipyridine systems. Fine-tuning of the color coordinates have been achieved. Color changes with concomitant spin stage switching have been demonstrated. Stable, reversible, and reproducible optical states responses for even every 1 degree rise in temperature is demonstrated. Multi-step switching possibilities mimicking simple, volatile, reversible, and reconfigurable logic circuit up to 5 inputs and binary, ternary, quaternary states have been achieved. Fabrication of dynamic window prototypes having color and opacity have been demonstrated. Color of light and heat transmission using dynamic windows lead to energy efficient buildings. Thank you.
Okay, you have studied electron donating and electron withdrawing groups on periodic group.
Yes, sir.
Which one is the best one?
So without the electron donating or electron withdrawing groups. Electron donating groups require much more temperature for the bleaching of the MLCT band. The simple system, that is the 2' bipyridine and iron, requires lesser temperature.
Bipyridine is the best one. Then what information you have received from taking the CV studies?
So the CV, we got this simple, that is the iron 2+3+ oxidation is for the 3 systems, for the 3 ligands, L1, L2 and L3 we have studied. For that L1 we got the minus 100 millivolt, that is the simple 2' bipyridine, and but for the electron donating group it requires minus 130 millivolt and minus 180 millivolt was required for the electron withdrawing conjugated bipyridyl ring, conjugated pyridyl attached to the fourth position of the bipyridyl ring.
Solely for iron system. This is for iron system. Okay, okay.
We have studied the general behaviors in iron.
Okay. Okay. Thank you.
Any questions?
So I don't understand the mechanism of the diminishing the MLCT bands at high temperature. So
Yes.
what kind of the
So here the
change?
mechanism for these systems are, we believe that this kind of systems have room temperature, the bipyridine is coordinating to the iron, but in higher temperatures the bipyridine is changing and the DMSO is coordinating to the iron. DMSO is having oxygen based ligand, so it is coordinating to the metal center.
I see. So metal
It's a
makes
spin-state switching mechanism.
complex
At the
with
reversible.
solvent, right?
Sir?
Metal makes a complex with solvent. Ligand
DMSO.
exchange. DMSO solvent. Okay, I see.
Thank you, ma'am.
Thank you. Thank you for your patience.
Yeah, I just want to learn from you. When you were doing your experiment, your temperature range was from 25 to 90 degrees Celsius. Is there any scientific reason backing such?
Is there any? Can you repeat the question?
I want to learn from you, your experiment, the temperature range. You said you took 25 to 90 degrees Celsius. Is there any scientific reasons backing such? You said you choose from 25 to 90 degrees Celsius.
Okay.
I want to learn from you, is there any scientific reason backing such?
Not scientific reason, it is from room temperature to higher temperature, it was showing it's a polar solvent. DMSO is a polar solvent, so from 25 to 90 degrees Celsius, we check the thermochromics in that complexes.
Okay.
Thank you so much.
Thank you. The evaluation of posters are happening now. The participants are requested to reach near the Department of Chemistry now itself. Moving on. Now I invite Reba Mary Cherian, code number APB 03 for the presentation.
Good afternoon. Myself, Ribosome, working as a research scholar from Newman College, Thodupuzha, under the guidance of Dr. Sindel Jos and Dr. Sabu Thomas. I am here to present a first phase of my PhD work entitled as Extraction of Highly Functionalized and Stable Non-Cytotoxic Nanocellulose Crystals from Elateria cardamom using a novel green mildoxalic acid hydrolysis. On moving to the highlights, it includes the valorization of this underutilized matured stems of cardamom using soft organic acid hydrolysis, and it resulted in the extraction of rod-like cellulose nanocrystals with high thermal stability, high crystallinity, and highly functionalized with bacterial non-cytotoxicity. And it has shown an enhanced characteristic features when compared with other cellulose nanocrystals, and it could be a substitute for common mineral acid hydrolysis for the CNC extraction. And this is the extraction procedure which we have done for the extraction of cellulose nanocrystals. First, the stems is collected and it's crushed and washed, and it is grinded in using a mixture, and then the ECA fiber is subjected for the alkali treatment, and then it is subjected for the bleaching with sodium chloride in acidic buffer, and then it is subjected for the steam explosion using 10% oxalic acid. Later, the solution is homogenized in water, and the nanocellulose solution is obtained, and the yield is calculated using gravimetric analysis, and it is found to be 69%. The formation of nanocrystal is confirmed using the TEM image. On moving to the characterization of the cellulose nanocrystals, we have done the morphologic analysis using SEM microscopy. This is the microscopy image for the raw fibers, which shows the presence of hemicellulose and lignin, and the nanocellulose embedded in the matrix of hemicellulose and lignin. And on moving to the same image for the alkali treated fibers, we can see that some amount of hemicellulose and lignin have been removed by the reaction of OH minus on reaction with the acidic OH groups of the lignin and hemicellulose. And on moving to the same image of the bleached fibers, we can see a further reduction in the size of length and diameter of the cellulose nanocrystal, which again shows the removal of hemicellulose and lignin by the chlorine oxidation, and also some amorphous regions have also been removed from the nanocellulose. And this is the same image for the nanocellulose obtained. This 500 micrometer scale and 300 magnification is not good to distinguish the cellulose nanoparticle, and hence we carried out transmission electron microscopy to confirm the cellulose nanocrystals. And we have seen that individual rod-like crystals are formed with some sort of aggregation, which might be because of the intensive hydrogen bonding between the cellulose nanocrystals, and we have calculated the length and diameter of the cellulose nanocrystals using ImageJ software, and histogram is shown here, and also through Gaussian curves we have calculated the average diameter for each samples, and we can see that a decrease in the diameter can be seen, which attributes to the removal of hemicellulose and lignin. And average diameter of ECNs is found to be 16 nanometer, average length around 309 nanometer, and average aspect ratio around 17.80, which is quite high when compared with the other cellulose nanocrystal, which aids in its use as a polymer reinforcing agent. This is for the atomic AFM images, which further confirms the cellulose nanocrystal formation. Here we can see distinctly the cellulose nanocrystal, unlike the TEM image. Here we can see distinctly the cellulose nanocrystal, which might be because of the sonication which we have done before, and also the very dilute solution taken for the AFM image. And this is the dynamic light scattering DLS study and theta potential studies to characterize the cellulose nanocrystal, and here we got a unimodal peak with a polydispersity index of 16.5%, which shows the effective extraction of cellulose nanocrystal. And this is the theta potential study where we got an average theta potential of around minus 34.24 millivolt, which is high surface charge functionalization, and this may be the surface charge functionalization that occurred in nanocellulose, where the OH groups of the hydroxyl group undergo Fischer-Spira's purification to get an oxalate substitution with the nanocellulose. This surface charge functionalization is visibly given by the nanocellulose by the complete dispersion of nanocellulose in water. This is the Fourier transform infrared spectroscopy for the functional analysis, and here we can see that some peaks when goes from raw fibers to ECNC have been disappeared, like the peak around 1730, which attributes to the CW water stretching in lignin and hemicellulose, and the second one 1510 centimeter inverse CW stretching of lignin, and 1250 CO stretching of alkyl neither lignin is absent. While as we can see a intensity increase in the intensity of the OH stretching, which attributes to the intensive hydrogen bonding present in the nanocellulose, here around 3333. The peak around 1639 shows the OH bending vibration of the adsorbed water in nanocellulose. The peak around 1152 shows the CO stretching band of oxalate ester group, and the peak around 1170 shows the CO stretching of the secondary alcohol of the nanocellulose. Or moving to the thermal analysis of the which we done with the TG and DTG, we can see the ECNC is said to possess some degradation temperature of very high degradation temperature of around 339.07 degrees Celsius, which is also comparatively higher when compared with the other nanocellulose crystals derived from other agromas residues. On moving to the X-ray, we can see the crystallinity has been increased to 72.81% with increase in crystallinity side 3.89 nanometer. This is the comparison studies, and we have yielded nanocellulose crystals with a high crystallinity, high characteristic features when compared with the other derived. This is the potential application of cellulose nanocrystals, which we can conclude with the proposed attributes. It can be used in edible coating, polymer reinforcement, biomedical engineering, water purification. We also have done cytotoxicity studies, which helps show that there is no antimicrobial activity against the 4 bacterial strains which have used, and it helps to pave the way for the idea of One Health, and also it can be used in conjugation with the antimicrobials with a non-antibiotic agent. These are the conclusions. These are the references. Thank you.
Riba, is it the first of a kind of work done using cardamom?
Yeah, yes. Yeah.
Okay. So you have shown a slide of, related to comparative studies. All the studies have been done by you.
No, that's other works, other reported works.
Reports comparing with what?
In all other cases, they have used either sulfuric acid for the CNC extraction, and here we have used mild oxalic acid for the CNC extraction. Successfully, we have resulted in the extraction of cellulose nanocrystals.
So the first phase of your PhD work.
Yes.
So you have for the applications as the next stage. So what are the applications that you are planning?
Biomedical applications.
Biomedical applications. Okay.
Okay, you have mentioned that the yield is 69 initially. In which context you have mentioned 69 from the starting material? Which is the starting material?
From the ECF fiber itself. I have taken 50 grams of ECF fibers to calculate the corresponding.
After the experiment.
Whole experiment.
Okay. Okay.
Thank you, ma'am. The next speaker is Ruba Rajan, code number APB04.
myself Rubarajan, Research Scholar from Kerala University of Fisheries and Ocean Studies. I am here to present the structural, thermal and antioxidative potential of biopolymer composites incorporated with the green macroalgal extract as a sustainable approach. We know that the synthetic polymers dominates the food packaging industries, and several environmental as well as food safety concerns are related to this in the current scenario. So as an alternative to this, bioderived packaging solutions are gaining much interest, specifically biopolymer composites that involve two or more components together which contribute to the enhanced properties of the biocomposites. So our aim was to develop a biopolymer composite in which we used the chitosan as the base material, to which we added the green macroalgal extract, for which we selected Halimeda macroloba, a green seaweed species, and its extract was prepared to and incorporated into the chitosan composite, and also it was reinforced with the silver nanoparticle. So the objectives of the study were to prepare ethanolic extract of the green macroalgal species and to develop chitosan based composite film and to characterize its structural, thermal and antioxidative capabilities to be used as a food packaging material. So the work protocol involves the sample collection, then its pre-treatment and drying, pulverizing to a size of less than 1 mm, and the ethanolic extraction was done with 70% ethanol, and the crude extract was lyophilized until further use. Then the extract was mixed with the 1% chitosan solution, and 2 millimolar of silver nanoparticle solution was added to this, and the film preparation was done following the solvent casting and drying method. So the result states that since the Halimeda macroloba is a highly calcareous species, the ash content was a little bit higher in the proximate analysis, which might be due to its calcareous nature. And the drying yield was 35.25% for the Halimeda macroloba, and the extract yield was 14.2%. The GCMS profile of the Halimeda macroloba extract revealed the presence of some bioactive compounds like cyclohexene, phytol, methyl ether, etc. that having bioactive potentials, including the anti-cancerous, anti-diabetic, immunogenic applications. As well as it helps to enhance the structure when it is incorporated in some food packaging applications. Then the NMR spectra revealed the proton shifts which corresponds to the carbon atoms in the alpha position in the unsaturated groups of allelic carbonyl immunogroups, as well as the aliphatic protons on carbon atoms that simply bound to the oxygen atoms in alcohol polyenol groups. Then coming to the properties of the biocomposites developed, the thickness range was nearly 0.11 to 0.125 mm. Then the old biocomposites were slightly yellowish in appearance, which might be mainly due to the presence of chitosan as the base material. And the color evaluation also satisfies the corresponds to the same observation. Then the FTR spectra of the biocomposites revealed that the broad characteristic peaks were observed with the increased hydrogen bonding, and which might be due to the composite effect of chitosan, the seaweed extract, as well as the nanoparticle incorporated in the film formulation. The bands at the amide 1 bands were located at the 3392 as well as 1633 wave numbers, and the OH and carbonyl stretching were shifted to a lower range upon the incorporation of the seaweed as well as the nanoparticle. Then the thermal analysis was conducted based on the differential scanning calorimetry, and the glass transition temperature was observed at 93.4 degrees Celsius. Then the same morphology revealed that the chitosan based controlled chitosan films have comparatively smoother appearance, while the incorporation of nanoparticle as well as seaweeds were caused a clog-like an agglomerate effect, which seen as the patches in the SEM images of the both composites. Then the barrier property of the composites were improved upon addition of the seaweed extract as well as the nanoparticle, while also coming to the mechanical characteristics, the tensile strength got improved upon the incorporation of the seaweed as well as the nanoparticle. Coming to the antioxidative analysis, the DPPH radical scavenging activity got increased upon addition of the seaweed extract. However, it got slightly reduced upon incorporation of the nanoparticle to the composite. The total phenolic content was improved compared to the control chitosan films. So to test the packaging efficiency, we have packed the biocomposite developed biocomposites with shrimp nuggets, and upon shelf life analysis were conducted at refrigerated conditions for 9 days, and we have observed that a change in pH values as well as increase in total volatile base nitrogen, and the peroxide value were getting slower due to the incorporation of seaweed extract as well as the nanoparticle to the chitosan formulation. So to conclude the study, we have found that we can develop active food packaging material by using the chitosan biopolymer with the seaweed extract as well as the nanoparticle. However, the physical appearance of the film as well as while incorporating seaweeds, we have to while incorporating seaweeds as well as nanoparticle, we must note on the migration of these particles to the concerned food product. So these aspects must be covered to scale up its opportunities to be commercialize the food packaging sectors. So these were the references. Thank you.
Why is it the synergic effect or combined effect contribute to the antioxidative property?
Yes, sir.
But in the slide number 14, you have mentioned that addition of nanoparticles can decrease the property like that.
The ppH only just get reduced, sir. Along with the seaweed, it was nearly 40 plus. But for the nanoparticle incorporated composite, it gets slightly reduced, 38 something, 38 point something. A slight reduction was observed.
Is it contradictory to your result?
No, sir. The thing is that only slighter variation is there. So the seaweed extract was more concentration compared to the nanoparticle.
What about the proportion?
For a 200 ml film solution, we added 180 ml of chitosan, then 20 ml the composite material. 10 ml seaweed extract, 10 ml nanoparticle.
So is it the first work reported from Halimada?
Yes, ma'am. Along with the other biopolymers, this is the first work.
So what is the importance, or why you have chosen green algae rather than the brown seaweeds which have been widely used in these type of works?
Currently, so many seaweed species are already utilizing for developing different packaging material. This Halimada species is less exploited, but having good properties like antioxidative, like having film forming abilities, etc. So that's why.
And in your GCMS analysis, you have identified a lot of compounds. So what is the interest of that identification with this particular thing? So GCMS analysis of what you have shown actually, the extract.
Yes, ma'am. The GCMS of extract.
Ethanolic extract
Yes,
of this Halimada.
Yes, ma'am.
So this extract, simply that you have taken it for GCMS?
Yes, ma'am. The ethanolic extract of seaweed species.
After purification?
Yes, ma'am.
So how did you purify this?
Actually, it was outsourcing.
Okay.
Thank you, ma'am. Now let me invite Fatima Ashraf, code number APB02.
So, Good evening all. I am Fatima Ashraf, PhD scholar from COFOS. I am here to deliver a presentation on the topic spectral evaluation and structural characterization of acid and pepsin-soluble collagen isolated from the skin of marine eel. So, this fisheries sector is considered as a sunrising sector with an annual production of 178 million metric ton according to the latest report of FAO. And around 20 to 50% of the total catch is considered as only the edible portion, and the remaining is just discarded. So it is very essential to recover the very valuable nutrients from this non-edible portion, and in order to minimize the environmental concern associated with this disposal. So one effective strategy is the conversion it into a value-added compound, and one such compound is collagen. This collagen is basically the most abundant structural protein that is present in the human body, which is having a triple helical structure with a repeating amino acid sequence of glycine, proline and hydroxyproline. So why marine collagen means basically the collagen that is available in the commercial market is derived from bovine and pig, etc. that causes so many disease outbreak and also the religious barriers. So in order to overcome this one, we can use marine collagen, which possesses so many good properties. So the main objective of this study was to isolate the acid and pepsin-soluble collagen from the discarded skin of Muranosaurus bagio, which is a most abundantly available eel or marine fishes in the Kerala coastline, and also to evaluate the various structural characterization of this isolated collagen. This is the extraction process. The raw materials were collected and pre-treatment were done using NaOH and butyl alcohol in order to remove the non-collagenous matter, and then acid-soluble extraction was done using acetic acid, and the pepsin-soluble was using both pepsin and acid. After that, centrifugation was done, and then precipitation was done using the NaCl. Then the precipitate was dialysed in order to cut out the molecular weight, and after that lyophilized the sample. So from the study, it was found that both the acid-soluble collagen and pepsin-soluble collagen was found to be a good yield of 29% and 40% on dry basis, and 11% and 16% on weight basis. And coming to the amino acid composition, both of these collagen is having a good amino acid profile, with glycine is the predominant one, and which constitute 1/3 of the total residue, and the amino acids such as proline and hydroxyproline, which is very important to maintain the structural stability of this collagen, which is also present in a good manner. Next is the SDS page was done to determine the protein pattern of the molecule. Here you can see that A1 and A2, which are present in the molecular range of 1000 kilodalton, which indicated that both of these collagen is type 1 collagen. Here comes the scanning electron microscopy in order to analyze the structural surface morphology. Here you can see that both of these collagen is having a rough and highly porous structure, which indicated that it provides a suitable environment for the cell migration, cell absorption, etc. This is the FDR spectra of the extracted collagen. Here you can see various particular spectra, means various characteristics peak at the all the respective bands, and which indicated that it maintains a structural integrity of the triple helical structure. Then coming to the denaturation temperature, which is a very important parameter of collagen. Here fractional viscosity was analyzed, and from that it can see that by increasing the temperature there is a reduction in the viscosity, which may be due to the denaturation of the collagen structure. And it is the HNMR structure. The peak at the 4 ppm is due to the presence of hydrated water, which react with the collagen surface and along with the structural molecule that provide integrity to the collagen structure. And here is the X-ray diffraction pattern. There is a particular band which maintains that the collagen is in its native structure without causing any denaturation. So the salient findings of this study is that the collagen that is derived from the discarded skin of eel is an effective alternative to mammalian collagen. Also, the higher yield and the higher thermal stability of the pepsin-soluble collagen as compared to the acid-soluble collagen make it an effective polymer for potential application. Okay, thank you.
You just go back to that FTIR. What is that amide A, amide B?
Yes, ma'am.
What is that?
These are 5 bands that are present in there. That amide A is related to the NH stretch coupled with hydrogen bond that indicate the presence of hydrogen bond in that molecule. And amide B is associated with the CH2 asymmetrical stretch. And amide 1 is CO stretch. And amide 2 is COO asymmetrical stretch.
Why actually this has been interpreted by you?
Yes, ma'am. Yeah. In all the research papers, this is like that for collagen.
You have done the interpretation using research papers.
Yeah, it's cross-checked with a research paper and this
Not
is
with
for my collagen.
Actually, I think while interpreting this, rather than going through this papers and arriving at conclusions, try to sit with some persons with chemistry background and interpret in a proper way. Otherwise, there can be some misunderstandings.
Also from the XRD pattern, what information you have received?
So this is mainly done to analyze the structure, secondary structural characterization. So in the peak at this 7 indicate that it is in native stage. That is, it is having a triple helical structure without any denaturation.
Peak at 7.
Yeah, 7, sir.
That is 2 theta value?
Yeah, 2 theta value. No denaturation is happened for that one.
Crystalline nature.
Yeah, crystalline nature.
Okay, thank you.
Thank you.
Thank you, ma'am. A general reminder, the evaluation of POF2 will begin at sharp 5 p.m. All the participants were requested to reach near the Department of Chemistry now itself. Now I invite Dr. Bhagya Tifi, code number AEM 03 for the presentation.
Shall I start, sir? Good evening. Myself Bhagya TC,
doing postdoc under the mentorship of Professor S.M.A. Shibli, Department of Chemistry, University of Kerala. I am here before you to present my work entitled as a ternary porous heterostructure with anatized hematite heterojunctions as an efficient photocatalyst system for hydrogen generation. The irrational use of fossil fuels and the consequences of global warming have increased the global energy demand to a greater extent. It is therefore necessary to find an alternate energy source which should in principle be pollution-free, economic and storable. And hydrogen is considered as a clean and emission-free fuel and has the potential to replace the fossil fuels. The full benefits of hydrogen as a clean, versatile and efficient fuel may be realized only if it is produced from renewable sources, which contribute to the green hydrogen production. The current status of green hydrogen production is shown here. All this shows the scope of a hydrogen fuel in our country's economy. Among the available green hydrogen production methods, photocatalysis of water is considered as one of the most promising technology, as hydrogen can be manufactured from the abundantly available solar light and water. So we are also concerned with the photocatalytic hydrogen generation by water splitting, and background and motivation of my present work is that I have targeted on iron titanium photocatalysis system, and being a well-established system in the laboratory scale, we targeted it as a potential candidate to be attempted for practical implementation stage. So throughout my study, we have adopted various strategies as shown here, and as our next approach, we decided to attempt on structured porous materials to ensure good stability and towards hydrogen generation. In this context, we proposed a game-changing strategy of loading small photocatalysts onto relatively large-size co-catalysts, and the design merit lies in efficient charge separation and abundant active sites for surface reaction. So the main objective of my present study is to explore the design strategy of loading photocatalysts onto porous nickel oxide co-catalyst structures for promoting rapid charge separation. So the synthesis of the present methodology includes the synthesis of ternary heterostructures by a two-step synthesis strategy. The first step is the synthesis of a porous nickel oxide co-catalyst structure, and the second step is the loading of the iron titanium photocatalyst into the porous nickel oxide structure. And the synthesized heterostructure are labeled as shown here, and they were subjected to various characterization techniques followed by photocatalytic studies, and finally a probable mechanism for the photocatalytic hydrogen generation is also proposed. Results and discussion. Initially, the phase structure analysis by X-ray diffraction patterns were identified. The figure shows the X-ray diffraction patterns of the different composite loaded ternary heterostructures in comparison with that of the bare system. Besides characteristic peaks corresponding to hematite and anatite, common peaks corresponding to nickel as well as nickel oxide were also observed, but with negligible intensities. Among all these ternary heterostructures, gradual shifting diffraction peaks corresponding to anatites and hematite were observed with regard to the 2% heterostructure, which reveals the close interaction of the active material with the porous nickel oxide, and this also evidences the probability for generating more heterojunction. Hence, the 2% heterostructure is expected to show enhanced performance towards hydrogen generation. The crystalline and phase characteristics were also confirmed by means of Raman spectra. The bonding characteristics as well as the presence of surface active species were evidenced from the FTR spectra. The morphological and elemental characteristics by means of FESM-ERAX analysis indicated the effective incorporation of the composite active material in the porous structure. The presence of mesoporous nature of the synthesized samples were evidenced from the PET analysis. The ternary heterostructure formation was confirmed from the TEM analysis with regard to the corresponding lattice fringes corresponding to rhombohedral Fe2O3, tetragonal TaO2 and cubic nickel oxide. All these indicated the good crystallinity, enhanced electronic interaction and effective charge transfer at the generated heterojunctions. And further, the chemical composition as well as the electronic states were identified from the XP spectra, where character shift in binding energies corresponding to Fe2P, Ti2P and Ni2P confirmed the presence of electronic coupling between the components, favoring for an enhanced photocatalytic performance. The visible light harvesting ability of the synthesized heterostructure were confirmed from the UV-Vis analysis. The recombination efficiency by means of photoluminescence spectra confirmed the lowest PL emission intensity corresponding to that of the 2% ternary heterostructure, and the band gap analysis also revealed the most favorable band gap corresponding to that of the 2.31 electron volt band gap was observed, which is the most favorable, and that was observed in the case of the 2% heterostructure. So all these inferences and observation confirmed that the 2% heterostructure evaluated. Initially, the activity was analyzed in aqueous solution. Obviously, all the ternary heterostructures exhibited enhanced performance when compared to that of the bare system. The order of photocatalytic activity is shown here, and as expected and as predicted in our previous inferences, the 2% ternary heterostructures have exhibited the highest performance, and further decrease in activity could be attributed to the blockage of the internal porous active sites. So the 2% heterostructure is confirmed as the optimum photocatalysis system. That is, in that case, the photocatalysis particles were able to interact effectively with the co-catalytic nickel oxide structure. And the next strategy is that photocatalytic activity was also evaluated in presence of previously screened amine-based proton source in our lab, which is diethyl amine-based hydrogen chloride, and the activity was found to follow the same order, but the activity was significantly enhanced, and the enhanced activity is attributed to the pronounced Hall scattering action of the proton source. And finally, the purity of the photocatalytic evolved hydrogen gas was also confirmed by means of GC analysis. The long-term stability and recyclability was also confirmed. In addition to the structural stability was also confirmed. And finally, a probable mechanism was proposed, which is a combination of direct research scheme and NP junction charge carrier transfer mode. And further to support the evidence, lifetime and charge transfer efficiency was also carried, and it was well supported the mechanism. So to conclude, the ternary heterostructure photocatalysis, which we synthesized, promoted the rapid charge separation by providing abundant active sites for more surface reaction. The present photocatalysis system, the activity was found to be competent with some of the recently reported photocatalysis as well. These are some of my selected references. List of publications in various international journals. My deep sense of gratitude to my mentor, Professor S.M.A. Shibli, Cliff University of Kerala, for the analysis facilities, and I would like to express I'm forever grateful to NTAC-2023 for providing me this platform for presenting my work. Thank you.
Bhagyam, you have selected nickel oxide for generating heterostructure.
It's a pro-catalytic heterostructure.
Okay.
And
What about other metals, metal oxides like the zinc oxide? You haven't tried?
No, sir. Actually, we were targeting on the iron titanium photocatalysis system. It's already well established, so we wanted to make it. We wanted to make it whether it is feasible for large-scale application or not. It is already well established, so we need to further enhance its activity. So if it is loaded to nickel is already a well-established co-catalytic structure. So we have tried with the other also. My colleagues are already investigated with regard to the previous transition method series. But I have established this as the best system.
But in the literature you have mentioned zinc oxide is also in the literature last slide, 3 slides back. That's it. Zinc sulfide.
Yes,
Last.
that is also reported, but I I wanted to convey that it is also competent with the reports as well.
Okay. When only 2% to 10% you have tried?
Actually, that 1 to 10% is based on the content of naphthalene added in it. We have synthesized a porous structure from the nickel powder and by mixing nickel powder with that of naphthalene. Then it was subjected to slow simmering. So we will be getting porous in that. And corresponding to that naphthalene content, the 1 to 10%. I made that.
Okay. Which nickel salt you have used? Nickel chloride.
Actually, nickel powder I have to mesh size 2 mm size.
Directly.
Yeah.
Okay.
Thank you, ma'am. The next speaker is Shabiba AK. The code number is AEM04.
Good afternoon to all. Myself Shabiba AK, doing PhD under the guidance of Dr. Ehia AA, Department of Chemistry, University of Calicut. The topic of my presentation is macromolecular motors sensing both electrical and thermal working conditions. The development of sensing motors capable of sensing their working conditions through the same two connectivities remains as a challenge to the scientific world. Here we are exploring conducting polymers as multi-sensing supercapacitors. Conducting polymers are multifunctional polymers which conduct electricity due to the presence of conjugated double bond along their backbones. Some of the examples for conducting polymers are given here, out of which polypyrrole is promising due to its outstanding properties. Amazing properties of conducting polymers like low operational voltage, simple fabrication, lightweight, etc. fascinated both research and industrial communities in various fields of application such as supercapacitors, sensors, artificial muscles, etc. But the major drawbacks associated with conducting polymers are poor mechanical strength, brittleness and poor processability, which limits their application in devices, and it can be compensated through making composites or hybrids with other suitable materials. Hydrogels are efficient candidates for this purpose due to their biocompatibility, swelling, diswelling properties, mechanical strength, etc. Here we are fabricating polypyrrole chitosan hybrid films, for which first we have fabricated chitosan film through solution casting technique. Then polypyrrole is coated on the surface of the chitosan through in situ chemical polymerization of pyrrole using ferric chloride as the catalyst. And the figure B and C showing the photographs of pure chitosan and that of the hybrid film. And the figure D showing the flexibility of the hybrid film fabricated. On moving to the characterization part, the FTIR spectra of the hybrid film shows both the characteristic peaks of polypyrrole and chitosan, suggesting the successful coating of polypyrrole on the chitosan film. And then the conductivity studies were done by BDS analysis, and it is showing a conductivity of 4.55 Simon centimeter inverse. Mechanical studies were done by UTM analysis, and the parameters are given here. FSM analysis are given here, and it shows an agglomerated granular morphology of polypyrrole grown on the surface of the film. EDX spectra and the elemental mapping images also confirms the coating of polypyrrole on the chitosan film. On moving to the electrochemical studies, we have conducted the CV in 1 molar NaCl by 3 electrode system, and it shows a broad anodic peak corresponds to polypyrrole oxidation and a broad cathodic peak corresponds to the reduction. And during the electrochemical oxidation, electrons are extracted. Therefore, in order to balance the charge and osmotic pressure, anions and solvent molecules entered into the solution, and thus during oxidation the polymer swells and during reduction the polymer shrinks. The CVs were conducted at different scan rates, and as the scan rate increases, the peak current increases, and by plotting the peak current versus square root of scan rate, a linear fit is observed. Thus, according to Randall's equation, the process is diffusion controlled up to 200 millivolt per second. And moving to the application part, we have conducted the supercapacitive studies first by the 3 electrode systems, and the supercapacitive behavior from the charge discharge. Charge discharge is shown in figure A, and it shows a capacitance of 559 farad per gram at 0.2 ampere per gram. As the current density increases, the specific capacitance decreases, and it's showing a retention of 86% of its capacity after 1500 cycles as shown in figure C. And the capacitance was also calculated from the previous CVs, and it shows a capacitance of 717 farad per gram at 5 millivolt per second. And as the scan rate increases, the capacitance decreases, which is shown in figure D. And the impedance spectra is also given here. It shows a semicircle in the high frequency region and a nearly straight line in the low frequency region, and the impedance fit is also given here. And on moving to electrochemical reaction sensing, the sensing is due to the electrochemical reactions of polypyrrole, and during the oxidation n electrons are extracted, but the extraction does not occur through a single step, it occurs through n consecutive steps. Therefore, the system is thus working outside the equilibrium condition, that is, it does not obey the typical Le Chatelier principle. Hence, the Notero's principle formed by reformulating the Le Chatelier principle is applicable, which defines that any physical or chemical perturbation, whether it is concentration, temperature, etc., of the electrochemical reaction rate, should promote a shift in the reaction energy to fit the new imposed energetic conditions. Therefore, any reaction energetic condition must be sensed by the reactive material. Thus, according to Otero, the evolution of potential is given by this equation, and this equation gives the effect of applied current and temperature on the potential evolution. And based on this, we have studied the reaction-driven sensing properties by using chronopotentiometry. And first, current sensing, that is sensing working electrical conditions. Figure A and B shows the chronopotentiograms of the hybrid film at various currents for the anodic and cathodic processes. And here the sensing parameter is electrical energy, which is given as U is equal to I into integral EDT, where integral EDT is the area under the chronopotentiograms. Then by plotting U versus applied current, we get linear fit for both anodic and cathodic process, suggesting it as a current sensor. That is, the material can sense the working electrical conditions. Next one is temperature sensing, that is working thermal conditions. Figure A and B shows the thermal chronopotentiogram at various temperatures, and as the temperature increases, the potential evolution takes place at lower values. Again, consumed electrical energy is the sensing parameter. By plotting it versus temperature, linear fit is observed, which suggests the material as a temperature sensor, or the material can sense the working thermal conditions. As a proof of concept, we have fabricated solid-state symmetric device, that is a sensing motor based on the chitosan polypyrrole hybrid films, and the symmetric solid device is simply fabricated by sandwiching PVA plus NaCl gel electrolyte between two identical pieces of the hybrid film formed, and its capacitance is calculated from both CV and GCD, and from GCD it is found to be 195.5 farad per gram at 0.2 ampere per gram, and showing a capacity retention of 89% after 1500 cycles, and from CV it shows a capacitance of 342 farad per gram at 5 millivolt per second. The impedance curve is also given here. And for the fabricated symmetric device, we have verified the current sensing capabilities. Conclusion: highly electroactive, flexible polypyrrole chitosan hybrid films were fabricated, and which shows a conductivity of 4.55 Simon centimeter inverse. The semi-wedges revealed a kind of granular morphology, and the hybrid film shows remarkable supercapacitive properties. Under galvanostatic condition, the electrical energy consumed is a linear function of working electrical and thermal conditions. Thus, the reaction involving macromolecular motors as reactants sense or responds to the electrical and thermal energetic perturbations of the reaction ambient by modifying the consumed electrical energy to self-adapt the newly imposed energetic conditions. As a proof of concept, a solid-state symmetric capacitor is fabricated with remarkable capacitance properties, and its current sensing capability is verified. This part of work is published in Journal of Energy Storage. Acknowledgements. Thank you.
What is the role of chitosan here?
Sir, actually our group is also focused on artificial muscle applications. For that, swelling and the swelling properties of hydrogels are very effective.
So you have studied?
We are actually demonstrating the multi-functional properties of polypyrroles, sensing, actuating and supercapacitance properties.
What about the stability of the resulting system composite?
The system is stable, sir, and the films are flexible too, and the CVs also shows that by repeated the CVs there is no additional peaks due to over-oxidation, or which indicates the stability of the film.
Have you taken the thermal analysis?
Yes, sir, we have taken.
Not shown here?
Not shown. It's due to lack of time.
Okay.
Thank you once again.
Any questions from audience? Thank you, ma'am. Next we have Dr. Shamna M, code number AEM05 for the presentation.
Good evening all. Myself Shamna, I am from Sacred Heart College, Devara, and I am going to present the work entitled Catalytically Active Silver Nanoparticles Stabilized on a Thiol Functionalized NU1000 for Hydrogen Evolution Reaction, and the work was carried out under the guidance of Professor Sugendu Mandal, Indian Institute of Science Education and Research, Thiruvananthapuram. The realm of coordination chemistry came into existence by the emergence of Werner's complex by Alfred Werner in 1893. In the subsequent years, the scientific community spectated the emergence of many new molecules like ferrocene, Hofmann-Cladrate, coordination network, etc. In 1995, a few baby steps were taken in the field of metal-organic frameworks by the discovery of a cobalt morph by Professor Omayagi of Northwestern University. In 1998, Omayagi and co-workers were successful in the discovery of the first stable 3D morph termed as morph 5, and it consists of zinc metalline clusters and terephthalic acid ligands. Now we shall define metal-organic framework. Morph is a porous coordination network consisting of metal ion clusters and organic ligands containing potential voids. And morphs can be used in a plethora of applications like sensors, drug delivery, photoluminescence, catalysis, composite systems, purification systems, energy storage, gas storage, and separation. Now I shall convey the objectives of my work. We all know that metal nanoparticles have high surface to volume ratio and large number of active sites per unit area. But the downside of metal nanoparticles are they are highly thermodynamically unstable due to high surface energy, and there is a tendency of aggregation during catalytic reactions, which result in the loss of catalytic activity and recyclability. So how to overcome this issue of aggregation? Utilization of high surface area materials with porous structures is a good alternative in this regard. Spatial confinement of the metal nanoparticles on these porous structures restrict the growth of nanoparticles and thereby prevent aggregation to a large extent. Since morphs have high surface areas and pore volumes and tunable porosity, we have chosen morphs as an excellent candidate for the spatial confinement of metal nanoparticles, and we have chosen NU1000 for that. NU1000 is a zirconium morph termed after Northwestern University, and it consists of zirconium oxyhydroxy clusters connected to the ligand H4-TBAPY, which is 1368 tetrakis parabenzoic acid pyrene. And it also consists of 2D extended Kegong network with a trihexagonal tiling pattern, and it has hexagonal and triangular channels of 3 nanometer and 1 nanometer respectively. Moreover, NU1000 is a very hydrothermal stable morph, and it is resistant to many chemicals. Moving on to the synthesis of silver at NU1000 SH, initially NU1000 is synthesized by the reaction between zirconite fluoride, benzoic acid into EMF, and the ligand H4-TBAPY at 100 degrees centigrade for 12 hours. NU1000 is functionalized using a post-synthetic modification technique which is called solvent-assisted ligand incorporation, named as SALI. Using 2-mercaptobenzoic acid, thiol functionalities are introduced in this NU1000 by the replacement of hydroxyl groups on the zirconium cluster. These thiol moieties act as the anchoring groups for the incorporation of silver nanoparticles, and in our work we have done three different loadings of silver nitrate, that is 0.1, 0.2 and 0.25 millimolar of silver nitrate. Subsequent reduction by sodium borohydride resulted in the formation of silver nanoparticles on the thiol moieties. Hence, the silver nanoparticles incorporated thiol functionalized NU1000 morph is utilized for hydrogen evolution reaction. Moving on to the characterization studies, the powder X-ray diffraction studies reveal that the pattern of NU1000, thiolated NU1000 and the catalysts are in line with the simulated NU1000 pattern, which indicates the phase purity of the synthesized materials. Moreover, the peaks of the standard metallic silver are not present in the materials, which indicate the low loading and small size of silver nanoparticles. And the presence of silver nanoparticles is further evidenced from the UV visible diffuse reflectance spectroscopy, where we can see a surface plasma resonance peak of silver at 451 nanometer. The TEM images shows that the silver nanoparticles are embedded on the NU1000 morph with a lattice despacing of 0.23 nanometer, and the average size of the nanoparticle is found to be 8.6 nanometer, and the TEM elemental mapping images are shown here, where we can see the silver nanoparticles are homogeneously distributed throughout the morph. BT surface area measurements were done using nitrogen adsorption-desorption studies, and where we can see the microporosity and the mesoporosity of the materials are retained even after the incorporation of sulfur and the silver nanoparticles, which indicates the structural stability of the morph. To learn about the coordination environments between the sulfur and silver, X-ray photoelectron spectroscopic studies were done. We can see the sulfur 2p spectrum of the thiolated morph consists of two peaks S2p 3 by 2 and S2p half, whereas in the silver incorporated material, the sulfur 2p spectrum consists of two peaks again S2p 3 by 2 and S2p half, but with a shift in binding energy values, which indicates the soft acid soft base interaction between the sulfur and silver in accordance with the Pearson's HSAB principle. And the silver 3D XPS spectrum shows the presence of two spin orbit pairs where each peak is deconvoluted into two, indicating the presence of silver 0 and silver 1. The electrochemical studies for hydrogen evolution reaction is done in collaboration with Professor Subrata Kundu of Citri Karaikudi, and all the electrochemical measurements were done using the conventional 3-electrode system, and the electrolyte was 0.5 molar sulfuric acid and the catalyst loading was 0.1 milligrams per centimeter square. The linear sweep voltammetric curves are used to determine the overpotential of a catalyst. Overpotential is a parameter which determines the efficiency of an HER catalyst. Lower the overpotential, higher is the HER efficiency. Here we can see the 0.25 silver Nu offers a lower overpotential at a current density of 10 mA per centimeter square. The Taylor slope analysis is used to determine the kinetics of the reaction, where we can see 0.25 AgNu offers a very low Taylor slope of 53 millivolt per decade, which exhibits a quick charge transfer at the electrolyte interface, and the rate determining step is Volmer-Herovsky step. From the Nyquist plot, the charge transfer resistance of the catalyst is determined, and we can see 0.25 AgNu has a low charge transfer resistance of 3.04. Mass activity is another parameter which determines the goodness of the catalyst, and here we can see with a very low loading of 0.1 milligram per centimeter square, the mass activity of 0.25 AgNu is higher than other catalysts. The dynamic stability is studied from the acceleration degradation studies, and the catalyst is stable up to 2000 cycles, and the static stability is measured using chronoamperometric measurement. From here we can see the catalyst has an incessant activity for 24 hours. In conclusions, a post-synthetic modification method called SALI was utilized to introduce thiol moieties Nu1000, and silver at Nu1000 SH was synthesized with nanoparticle of size range 7 to 8 nanometer with 3 different loadings, and the catalyst showed HER activity with an overpotential of 165 millivolt, and the sustainability of the catalyst is proved by the performance for 24 hours. And the work was published in Nanoscale recently. I thank God Almighty for this wonderful day, and with immense pleasure I thank Professor Sugendu Mandal, my project supervisor, School of Chemistry, ISA Trivandrum. I thank my group members and whole ISA-TVM family and CSIR for funding. And I extend my sincere gratitude to my current colleagues, Department of Chemistry, Sacred Heart College, Tevra. Thank you.
Why it is named as SALI?
Solvent assisted ligand incorporation.
Okay, and you have 3 loadings of silver, no? So that is, how will you differentiate the activities for 3 loadings?
Yeah, it is shown in the linear sweep voltammetric curves. Yeah, here. From the bar graph, it will be more clear. That is 0.25 AgNu will give a lower overpotential of 165, whereas 0.2 and 0.1, and this is thiolated without any silver nanoparticles. So
Okay.
it offers a very high overpotential.
Okay.
Regarding the silver nanoparticles, how you have prepared?
By solution impregnation method, actually silver nitrate is added to the Nu1000, thiolated Nu1000 morph, and then reduction by sodium borohydrate.
Okay. What percentage of silver at the end?
Actually 0.25 AgNu will have 2% silver, 2 ppm actually, 2 milligram per liter, and correspondingly 0.2 is lesser, and 0.1 is also even lesser than 0.25. Even though we added 0.25 millimole, some of them is leached out, like while washing, the silver nitrate itself is leached out. But after we have actually characterized after the catalytic studies, once the nanoparticles are formed, there is no leaching, because we couldn't see from the ICP-MS data.
So you can reuse the catalyst.
Yeah, actually we have reused the catalyst for 2000 cycles. The catalyst was stable up to 2000 cycles.
Okay.
Thank you, ma'am. Now, let me invite S. Vijay Kumar, code number AEM06 for the talk.
Pointer just moved. Okay, good evening everyone. So first of all, I like to thank the organizers for giving an opportunity to present this work titled High Capacitive Two-Dimensional Networks from Aliphatic Monomers, work carried along with Professor Ajay Ghosh and Dr. Srijit Sankar in CSIR NIST. Before moving into two-dimensional networks and the significance of preparing them from aliphatic monomers, let us look about porous carbon, which we are all familiar with. I hope all of us may come across at least any one of these, like beauty products or the porous carbon filters or the gas absorption mask or any intoxication products like tablets or food products. So what makes them to have a wide application means there are different sizes of pores in this activated carbon which absorbs small molecule and gases. But it is not just that is what the porous carbons can offer. It can work as adsorbent for ions or a catalyst or in energy storage. Particularly we are concentrating on energy storage, where in a capacitor if we use that as an electrode material, the small electrolyte ions can be trapped in the small pores and through electrostatic interaction, which in turn used for energy storage and releases. But we can improve the property of this porous carbon, since we can see there are some voids which are non-accessible to these small ions. In that case, we can incorporate 2D materials with defined pores, as well as we can incorporate different atoms like nitrogen, boron and others. So particularly we are concentrating on two-dimensional material like graphene, in which we are trying to incorporate atoms like nitrogen in a systematic space. So there are, through chemical methods, there are three electroactive linkages which are triazine as well as quinazoline as well as Hatton workers. We can see all of them capable of undergoing redox activity. But when we see the number of nitrogen atom, especially in this case it is 1:1 and it is varying in the other two networks. What we are going to do is we are planning to increase one nitrogen atom compared to this Hat network, which in turn we expect that to undergo a redox reaction like this in the presence of proton. So the origin of idea came from this work, where we can see they have made triazine units from both aliphatic and aromatic diketones. So we have taken a hexaketone with this tetraamine. We expected there are 4 possibilities of reaction with respect to the reactivity of each of them, but in one case we can expect same kind of triazine formation. So even though there are two chemically inequivalent amines, we can expect 2 different networks. One is where the nitrogens are in cis fashion, and another one is it is in trans fashion, which in turn gives 2 types of pores. One is circular and another one is triangular. So coming to our initial characterization of the reaction, what we see in IR is there are peaks for residual amine as well as residual carbonyl, which gives an idea that there is a formation of triazine ring as well as unreacted amines and ketones in the network. When we go for XRD, we are getting only one peak for the pi-pi stacking, which is giving the 2D spacing, as well as in the solid state NMR spectrum, we see a broad peak centered around 150, giving an idea that though there are 3 chemically inequivalent carbon are there, all are centered here in this position. And the thermal stability of the resulting network is higher than the corresponding monomers, And when we went for the stability state, we put them in different solvent and acid, and we found that the residual solids are having the identical spectral and XRD features. But initially we thought they are digested, but in fact they are not digested, they are exfoliated in solvent, which is confirmed by Tyndall, and also the 2D layers are found by the atomic force microscopy. In SEM also we can see different type of morphologies like seeds as well as spongy balls, and the elemental distribution is also seen here in the elemental mapping. In TEM we can see elemental distribution equal, and also we can see 2D layer with the same spacing as we observe in the XRD, and here also the elemental percentage is given by Yields technique, and here we can see this is showing a feeble crystallinity. And coming to the X-ray photoelectron spectroscopy, through which we analyze different types of atoms present in this, which is giving the presence of carbon, nitrogen and oxygen, and porosity also found by around 14 meter square per gram. So though our hypothesis is having two different type of spores, considering all the above results, we come to a conclusion that the plausible network may be with unreacted site as well as both the different pores crumbled to each other. So coming to the capacitance behavior of this material, after all we are organic chemists, so we have given this to Anjana PM and Dr. Rocky of our institute. They checked the electrochemical capacitance of this material, both in 3 electrode as well as in device state. In 3 electrode we can see the redox behavior of this one, which is we are correlating with the triazine units percent as well as the residual carbonyls. And in 3 electrode measurement we are getting around 600 farads per gram of energy storage. In the device state, in a symmetric capacitor, we see around 333 farads per gram. And in summary, what we anticipated is to create one nitrogen extra, whatever is already studied in the literature, and that is from an aromatic monomer, which is in turn synthetically challenging as well as in the purification also difficult. But in our case, we have achieved the triazine unit around the benzene ring, which is synthetically easy and purification wise also it is easy. And in this case, this is the highest organic material reported with this much farads per gram of capacitance, and our material is competing with that one, and it is outperforming in the case of symmetric device. And I thank you for your attention.
What about the solubility of this
This
material?
is insoluble materials. That's why we did all the characterization or in solid state, IR, XRD, and mapping through SEM10. Everything in solid state.
Insoluble in.
Yeah, they are insoluble and highly stable in 9 normal hydrochloric acid also.
Then what application you are expecting from this type of products?
So we are trying to create a graphitic azo network, and we are planning to use it in energy storage material. That's what is demonstrated also. They used in a symmetric capacitor, where it is shown a comparatively good performance with the literature.
Similar to the work of Alder condensation type to form graphene type structure.
Yes, yes. Graphene with azo graphene.
Okay. Okay.
So you synthesize the poor two-dimensional material, right? But so I imagine that another possibility is that just the highly branched material. So what is your proof to have the pore structure?
Yes. So though the hypothesis is like this, we have a trigonal pore and a circular pore. We say the plausible network is this. There are unreactivity, and there are some places cyclization is not complete. In such cases, when you have a free amines or the carbonyls in the tethering sites, there are possibilities of interdigitation also. That's why in the porosity measurements, we see the porosity is ranging from micro to meso and macro. It is starting from few nanometers to few hundreds of nanometers, which it reflects in very less surface area. So if it is a perfect 2D material, they may stack perfectly. But there are imperfections which leads to interdigitation, because here it is drawn in 2D, but in solution, when they are forming, the other network can approach from this way and enter through that. It's like catenanes, they can interdigit one within another. At some places, we see 2D, that's what we see in these lattice structures. But it is not extended. That's why it is not reflected in XRD. We do not have much peaks other than pipette stack. So the crystallinity is very feeble.
Thank you, sir. The next speaker is Dheeraj BDF, code number AMI01. I invite Leah Turakel, code number AMI02 for the presentation.
Very good evening to one and all present here. I am Leah from IIT Palakkad, working under Dr. Minju Porel. My title for the talk is Functionally Tunable Thiocarbamate based Organic Polymer for Ultra-Fast and Efficient Removal of Diverse Contaminants. I would like to start my presentation by quoting a renowned scientist, George S. Hammond. He said, "The most fundamental and lasting objective in synthesis is not production of new compounds, but production of new properties." So what if we have a compound or a few compounds with a lot of properties? My work is based on such a compound, dithiocarbamate-based organic polymer or DTCOP, which can actually be used for performing all these applications listed here. I'll be discussing one by one. For a material to have these many applications, the first and foremost thing to have is this, the balance between economy and performance. To make it economically balanced, we made the synthetic strategy so simple such that it can be completed in a two-step reaction using inexpensive starting materials, and the total time taken for the synthesis is just 1 hour, even in a highly scalable manner. And to make it performance better, we have introduced tunability in functional groups and ring size, and also incorporated a important functional moiety dithiocarbamates in its backbone. These materials were stable, porous and water insoluble so that it can be used for water remediation processes. By looking at the synthesis, it is observed that both these reactions has been completed within 15 minutes and no high temperature, high pressure, no inert atmosphere, nothing is required. If you have an RB, add your reagents and your polymer is ready, and this purification is also very simple by washing in different solvents. And we have synthesized these many compounds by changing its functional groups of our interest like benzyl, methyl, and this chain size also been altered. And these materials has been characterized by using IR, NMR and elemental analysis. And morphologically, this DTC OP1 and 2 were fine powder, whereas OP3 and 4 were little different. It showed a glassy or shining appearance, and this change in this appearance were also observed from the SEM image, as you can observe here. And this change was evident from the powder XRD pattern, and you can see that it is OP3 and OP4 were actually amorphous, but interestingly OP1 and OP2 were semi-crystalline in nature. And this material also showed porous properties with a tunability in the pore size, and also they were stable at a higher temperature and have high glass transition temperature as well. As this material has a lot of sulfur in it, we exploited the soft-soft interaction between the sulfur and heavy metal mercury for the removal of mercuric ions from water. And we observed that 99.9% removal of mercury over the concentration of 5 to 400 ppm was observed even in a high uptake capacity of 413.7 milligram per gram, which means 1 gram of the material can take 413.7 milligram of mercuric ions. And we organic chemists like to separate everything by column, and we used our material as the stationary phase in the column, and we observed that mercury removal was happening, but the system was very slow. So we introduced a very new technique which has never been used for this kind of application, which is spin column, where we have loaded our material in this plastic tube which is fitted by a fret, and we added our mercury contaminated water and just spinned in a mini centrifuge tube for 30 seconds, and all this water was down, and we found that this mercury has been removed from the water completely. And this material has also been regenerated up to a couple of cycles, and no removal of essential metals like calcium, potassium and sodium was observed, and it is effective in different pH and in real water samples where another contaminants are also present. Iodine is also another contaminant, and our material has a lot of heteroatoms such that it can capture iodine from various sources like air, water and organic solvents. And we can observe that this material has captured iodine from its vapor state, and it has been regenerated for the next use. And iodine capture from cyclohexane was also possible within 5 seconds, and iodine from water as I2 and I3 minus was also captured within a short time, less than a minute, with high uptake capacity. As our material has a lot of aromatic groups, and it can make pi-pi interaction with organic dyes. And these many organic dyes, we have tested the removal property, and we observed that our materials can remove these multiple cationic and anionic dyes, and with a high uptake capacity. Especially for Congo red, it was about 1000, and exceptional result was observed for eriochrome black tea, which was 4000 milligram per gram, and this is the highest ever reported till date is 1200. So not only the high uptake capacity, kinetics is also important. There is a video over here. If you play it, it would be great. Sorry for this technical glitch. So without wasting time, I'll mention what is happening. We have loaded our material in this syringe and we have added EBT solution in it, and we can see that within no time this color has been removed from this material and the water is crystal clear. And to show that this this filter has no effect, this control has also been done without adding material. So the kinetics is very fast, and the removal was also observed from the mixture of dyes except for two dyes, methylene blue and rhodamine B, so that we have exploited this for the purification of these two dyes for the from the mixture of other dyes. And this material has also been applicable for the removal of pharmaceuticals from water, especially diclofenac and also oxoanion permanganate ions within 5 seconds with good uptake capacity. And volatile organic compounds are one major contaminant in the air, which is also contributed by our chemist, and that has also been taken up by the material with good uptake capacity, and this material also showed antibacterial property as well. In a nutshell, I can say that we have developed a novel class of dithiocarbamate based organic polymers with tunable properties, and it has been observed that it is compatible for various applications. And in future, we'll develop the concept of lab to the market. And I acknowledge IIT Palakkad, my group members, funding agencies and facilities we have used. Thank you so much.
Regarding dye removal, is it an absorption process or okay? Can you reuse the polymer?
Yes, we have been
How?
Regenerated by after a methanol wash, and it can be reused many times. Methanol wash is enough.
Then again dye will move to the environment.
No, we can save it and reuse further. These dyes has been used for as indicators, so we can reuse it as indicators if required.
Okay, you mentioned that the soft interaction between sulfur and mercury. There are several other metals like zinc, gold, etc.
We have
you tried?
studied the interaction of various other heavy metals like cadmium, lead and all, and we observed that removal is also happening, but not in this high uptake capacity. So this removal up to 99% was observed, but only in a low concentration. In a higher concentration,
In that case also you can recover the polymer by HCl wash.
in the metal case, the polymer has been recovered by HCl wash, concentrated HCl wash.
Okay.
So you have shown one spinning technique. So what will be the reason for this more removal using that particular technique?
Spin column is actually based on the spinning technique, as you rightly mentioned, and when spinning this water or contaminated water has been in touch with all this material in a very short time, and it has a centripetal force which is actually like this. So the contact with this materials for the water will be very high in this case, rather than doing the normal absorption method or this column method.
Thank you, ma'am. Now we have our last presentation. I invite Dheeraj BDF, code number AMI01.
Okay, good evening everyone. First, thank you for your patience. Firstly, let me thank the organizers for giving me opportunity to present our work here. I am BDS Dheeraj. I am working with Professor Kuruj Joseph from Indian Institute of Space Science and Technology, Trivandrum. We are a group working on polymer nanocomposites, but we started working on converting this polymer to carbon nanofibers and increasing its applications. Okay, let us see this. Here my presentation topic is preparation of porous carbon fibers via electrospinning approach. What are carbon fibers? Carbon fibers was the highly used aerospace material having high mechanical properties, tensile properties, electrical properties, thermal properties, etc. And how we are using carbon fibers is, carbon fibers we are incorporating in a polymer, making a polymer composites, and these laminates we are using for structural applications. And there are so many applications of polymer. Since my institute is a space institute, we mostly focus on aerospace materials and how we are making these carbon fibers, right? There are 3 different ways of making carbon fibers, but we focus on electrospinning approach. What is electrospinning? Electrospinning is a simple approach in which we are taking a polymer and mixing it with a suitable solvent and making a fibers out of it. Okay, this approach can be like extended to your applications in different ways. And how we are making these carbon fibers from electrospinning? Let me take an example of PAN. PAN is one of the most used polymer for making carbon fibers. You can say 90% of the carbon fiber in the world is made with PAN fiber, but PAN is a non-graft agent polymer. You can see the structure here. To make this PAN as a carbon fiber, you have to do 2 steps. One is stabilization and carbonization. During stabilization in oxygen, this PAN structure will change into a ladder type of structure, and during carbonization, it will convert into a complete carbon structure. Depending on the temperature, the properties of the fibers will be there. So, like this only, in our lab, we made PAN-based fibers. First, we have taken electrospinning of PAN, thermal stabilization and carbonization, and we got some nicely flexible carbon fibers out of it. Then we want to improve the functionality of this fiber, so we thought of porous carbon fibers. There are 2 things that we consider here to increase the porosity of carbon fibers, to increase the surface area and increase the application. And the second point is the cost of PAN. PAN is very expensive, so we want to use some other polymer instead of PAN to get carbon fibers. That's why we have taken PVDF for our work. PVDF is a thermoplastic elastomer and a fluoropolymer having many advantages. And what will happen in a PAN-based system is, when you're taking PAN, for making it porous carbon fibers, we are doing an extra activation step or physical or chemical activation. But if you take PVDF, PVDF will directly give you porous electrospun fibers. This can be converted into porous carbon fibers directly. One, you are decreasing the material cost. Second, you are decreasing the production cost. So you can make carbon fibers at low cost. And what we have done is, like this only, we have made the 3 different sections. In step 1, we have taken PVDF of molecular weight and this combination of DMF and acetone, and we have made beautiful carbon fibers at different flow rates of electrospinning. You can see at all the flow rates, we can see nice fibers. And with respect to this flow rate, the diameter increase, like from 300 nanometer to 1 micron you can make. And then if you can see in high magnification, these fibers are porous. And next step is dehydrogenation. And why we are doing is, in PAN we are doing thermal stabilization, but here thermal stabilization will not work. When you take PVDF fibers and keep in thermal stabilization, all the fibers will melt. Once there is no fiber, there is no need of carbonization, right? Then we have used some dehalogenation technique in which we are keeping this in a strong base so that the fluorine will go out and the conjugated double bond will form and it will arrange to form this carbon fiber. And then we have seen this is carbon but stability is not there, which means here there is so problem with the stabilization step. It is effective but the parameters has to be optimized. For this we have taken 2 things. One, we have done the stabilization in an autoclave. Second, we have taken a surfactant like a phase transfer catalyst for increasing the reaction speed. And what we were expecting is this PVDF is white, it has to convert into black or dark brown, then we can say that it is completely stabilized. We have taken 2 bases. In 2 bases we have seen this concentration is optimized one and color was completely changed to black. So we have taken this one, we have set this stabilization parameters and carbonization we have done at 900 degrees in argon climate, and then we have got beautiful porous carbon fibers. You can see when we are using NaOH as the base, at different flow rates we got some beautiful fibers, and all these are having good carbonyl, and at low dimensions these are carbon nanofibers. And when we are using KOH, we have got the same thing. The porosity, these are all porous fibers at different flow rates of PVDF, and these are all carbon fibers with good carbon yield. And then what we have optimized is, then we have set parameters for PVDF and then stabilized it and got porous carbon fibers with good carbon content. Then while the surface area is more than 1500 meters per gram, which is very significant in case of a porous carbon fibers. And when you compare with literature, our carbonation temperature is only 900, but we got a very high surface area than reported, right? And then what we are doing now is we are improving these parameters of electrospinning and stabilization, and we are trying to improve the surface area of porous carbon fibers. Now we want to see whether this autoclave we are doing now is working or not. So we have taken at similar conditions 2 samples with autoclave and without autoclave, and we have seen that surface area of the fibers with autoclave is more effective. So autoclave is working here. So because of the stabilization step we adopted, we are getting a high surface area porous carbon fibers. Then what we have done, we want to increase the porosity further. So we want to add a lignin as a sacrificer polymer, which means lignin will be with PVDF. During carbonization step, it will sacrifice itself and form a porous nature there. Like that we have seen with different compositions of lignin, you can see 5, 4, 3 and different combinations, and we got nice porous carbon fibers, and these porous carbon fibers are having good surface area. Right. And now we conclude that this PVDF is a thermoplastic elastomer and very cheaply available. If you consider the cost of PAN and PVDF, instead of buying 1 kg of PAN, you can buy 2.4 kgs of PVDF, not accurate based on Google searches and all. And India is not having a carbon fiber commercial manufacturing unit. We are depending on some other countries, right? So India don't have a commercial grade carbon fiber. So research in carbon fibers and searching for a low cost alternatives is very useful to set up a carbon fiber plant and supply carbon fibers to our domestic needs. And I have to acknowledge my supervisor, Surya Joseph from IISc, or the director, Dean HOD and VSSC Center. They have done a lot of help for this work. And CSA, Saif USAT and CSA NIST and the fellow students and organizers of this conference. And thank you all for your kind attention. Thank you. I'll be happy to answer your questions. Thank you.
Have you compared the chemical applications of these 2 compounds?
No, our application is EMI shielding. We are working on that.
Okay.
With the PAN-based things, we have done all the work. We want to compare that with this porous carbon.
But it is very costly. PAN.
Very costly, so we are decreasing it.
What about the cost ratio?
When I have seen in Google, like common searches and all, instead of buying 1 kg of PAN, you can buy 2.5 kgs of PVDF.
Commercially?
Yeah, commercially.
Questions from audience?
Instead of using PVDF instead of PAN, what are the applications you are expecting in a
EMI shielding application with porous structures.
And regarding the porous structure, you said you used lignin.
Lignin as a sacrificing material.
Yeah, and you made a porous material, and that pores are, how deep are that pores?
That's what we are deeply investigating on that. But at 97:3 ratio, we are seeing good surface area. We are doing studies on that ratio now.
So what would be the fiber diameter of those fibers?
So we are getting 400 to 600 nanometer range fibers.
And this lignin particle diameter?
Lignin particle diameter very less, like we have seen that 3 to 5 nanometers small thing, dispersal phases.
So in that case, the lignin nanoparticles will not make the deep porosity, right?
Yeah. Actually, the way in which what people will do is, when 2 polymers are there, this dispersal phase will be very less and it will be used as a sacrificing template. So that is the way I tried it. And it is not that much working. PVDF alone is giving 15, 20 something, and this is giving 50, 60 something. That is not effective that much, but very small improvement is there.
So is this, that concentration of lignin has any direct effect on the PVDF concentration?
Sir, we have started with less amount. When we are taking 50-50, I am not getting this much porosity. Very small amount only, we are getting this much amount. What is happening is, when I am taking good amount of lignin, the surface is getting broken from this fiber. So I am not getting increase in porosity, a macro porous structure will form. So we are not going for high ratios of lignin, we are using for less amount.
Okay, thank you.
Thank you.
Thank you, sir.
And that was an end to today's oral presentation.
Thank you, judges. And I would like to thank all of you for your attentive listening and participation during this session. A gentle reminder, we will have 2 oral presentation sessions tomorrow. The first session will be held at 8:30 a.m. in the Malicious Hall, and the next session will commence at 1:30 p.m. in Nobel Laureate Hall on the first floor. I repeat, we will have 2 oral presentation sessions tomorrow. The first session will be held at 8:30 a.m. in the Malicious Hall, and the next session will commence at 1:30 p.m. in Nobel Laureate Hall on the first floor. The cultural program will begin at 7 p.m. in the auditorium, and the dinner will be served thereafter. It's my pleasure to invite everyone to be a part of this event. Thank you.